হ্যাঁ, কথা বলেছি। Hello, good morning, our respected teachers.
Good morning to you all. I welcome on behalf of APCRI Government College uh, for this special lecture delivered by Dr. Madhumita Manna, our Additional Director of Public Instruction, Education Directorate, Government of West Bengal. Within a few minutes, we'll start our session. A few participants are going to join. So we'll start our lecture very soon. Hello. Hello. Am I audible? A very good morning to you all. Today is the second day of Interdisciplinary International Web Seminar on Modern Trends in Humanities, Science and Technology and Social Sciences for Sustainable Development. The program is going to from 23rd and today is the 24th September. It is a 2-day program. Last day we had with us in the inauguration session Professor Subiresh Bhattacharya, Dr. Anjan Chakraborty and other delegates. Today's session will be started by the special invited speaker Professor Madhumita Manna, Honorable Additional Director of Public Instruction, Administration, Education Directorate, Government of West Bengal. After the special invited lecture, we will go to an invited speech, international speaker, Dr. Indrajit, Disaster Preparedness, Mitigation and Management Section, Asian Institute of Technology. On behalf of Organizing Committee, APCRI Government College, this is my privilege to introduce our special guest, Dr. Madhumita Manna, for opening the session. I request this is my privilege to introduce our special guest, Dr. Madhumita Manna. I request all the participants, please mute your microphone and video. Thank you. Dr. Madhumita Manna, our eminent speaker, has endless contribution in her field of work. I am here just giving a glimpses of her ornamental qualifications. At present, she is holding the chair of ADPI Administration. She was former head of the Department of PG Department of Zoology, Bethune College and Varsha Government College. She had also brought Bidhannagar Government College as former principal in WBACS category. She is involved in hardcore level of research. Her contribution in the field of tropical disease and parasitic diseases will be remembered by all for a prolonged period. 39 research papers and so many publications are there. Due to shortage of time, it's a very tough job for us to elaborate all the things. We welcome our special guest for special speech, Dr. Madhumita Manna, Additional DPI Administration, Education Directorate, Government of West Bengal, India, in this international web platform to deliver his speech. Madam, over to you, please.
Good morning to all, and thank you for introducing me so nicely. At the outset, I want to give thanks to Dr. Mayuk Sarkar, officer in charge of APC Roy Government College, Siliguri, for inviting me in this international webinar on modern trends in humanities, science and technology, and social sciences for sustainable development as a speaker. I am also thankful to the organizing committee. I extend my gratitude to Honorable Vice Chancellor, Professor Subidesh Acharya, for extending collaboration with one of our government colleges. I am thankful to all dignitaries, teachers of the college, my ex-colleagues. Many of my ex-colleagues are here, and present colleagues also are here, and all the participants for joining the webinar. And I wish every success. I know it is the second day of the seminar, and first day I asked Mayuk how it was. He told it yesterday I was extremely busy. He invited me. He gave me the link. But as you know, the vaccination program for the college university students are going on. It will start. So the preliminary work, that hard work is now going on in the department. So I couldn't make time to join that inaugural session, and I'm sorry for that. And I wish every success of today's webinar. And now the topic of my today's, let me give some time. কোথায় সিলেক্ট করব? আমি স্লাইড শো করে দিই। So the topic of my today's talk is colors are neglected tropical diseases. And before slide এইটা তো স্লাইড ছিল না। Sorry. For almost last 30 years, I am working in the field of molecular taxonomical characterization of the clinical isolates of Indian culture. And before going to detail of a part of my work, it is actually I have three aspects, three areas of research going on in my laboratories. In one field, it is the molecular taxonomical characterization of the clinical isolates that has been collected from the field, and that was also my work at the PhD thesis at Indian Institute of Chemical Biology. I think many of you know that Indian Institute of Chemical Biology works hard on Indian colors are in different issues, that is their immunological aspect, their genetical aspect, the molecular biological aspects, and also the epidemiological part. And that epidemiological part was given to me. It was a part of UNDP project that time when I was a student, PhD student under the then director, late Professor Amarnath Bhaduri. And my another research area is the vaccination prophylaxis and vaccination program for the disease, and still another is the phytotherapeutics using the nanoparticles. So today I'll be talking as the heading of the seminar is the modern trends in humanities, science and technology and social sciences for sustainable development. I think the science and technology for sustainable development, Moyuk told me to select my area, my topic. So I thought that I should say something about the sustainable development or the scientific development using the technology in the field of parasite identification, characterization, and epidemiological works. So I have selected a part of my research work, and before going to that research work, I want to brief something about the disease, about the background, about the history. So historical, in the historical background, we know that for the Kala-Azar or black fever or dum fever, whatever the name, it is the disease called known as leishmaniasis. To zoologists, this is the visceral leishmaniasis, and in Africa and India in the mid 18th century, the disease is called it is named variously, and the scientists, two doctors who discovered or who described the parasite rightfully, Dr. William Leishman from London, he described the pathogen and published his paper. Within few weeks, Charles Donovan in India, actually he was working in then Madras. He also published his discovery about the pathogen a few weeks after Dr. Leishman. And that time, it was that for naming the parasite, it was Dr. Ronald Ross, the Nobel laureate and the stalwart protozoologist, named the species as Leishmania donovani in 1903, honoring both the scientists. Many of us we know, those who are from the zoology, that the parasite is a kinetoplast protozoa. Am I audible? Everybody hear me?
Yes, ma'am. Yes, ma'am.
Okay. As I am not seeing any of you, so I got confused. So if we look back, that Kala-Azar, history of Kala-Azar in India, and also the current status, that on the basis of the existing medical records, we Asian are very lazy in keeping the records. That is the worst part of us, that we don't keep records. But it was that British personnel, they used to keep the records. So we got the information with their help, that on the basis of that record, the first epidemic of Kala-Azar erupted in 1824. It is the first report, first written report, 1824 in Jessore district, now in Bangladesh, killing about 45,000 person in consecutive 3 years. Records of epidemic of Kala-Azar from Garo Hills, Assam have been reported in 1885, 97, 1913, 1925 and 1944, showing a mean inter-epidemic period of 10 to 20 years. Similarly, Bihar has been devastated with Kala-Azar till the date year 1882, and followed by in 1891, 1917 and 1933, again that inter-epidemic period, span of inter-epidemic period is 10 to 20 years. Now, the disease that time was killing lives of hundreds of thousands, and that time it was not known that which is the vector of the disease, how the disease is propagated, what is the pathogen is known, that it is the kinetoplast protozoa, but the exact mode of transmission, it was not known. by the time that disease it started killing thousands of lives. And that time, even Brahmachari, you know, Dr. Even Brahmachari's magic bullet, the savior and the unsung hero who came up with the magic bullet, the urea stivamine, that helped, that saved in British India lives of thousands of persons. And for his great contribution in science in the life of human being, he was twice nominated for the Nobel Prize. But as you know, all of you know that he didn't get that prize, that prestigious prize. But then also, we should not forget, we should not go on oblivion the name of even Brahmachari, who came up with the magic bullet, that is the urea stivamine. If you see the graph, you could see that the in the report is in my right hand. In 1925 to, in the span of 1925 to 1936, that death number of cases treated, and the death reduced to from 6365 to 753. Now, the current status, regarding the current status, in post-independence decades, it is considered, the disease is considered to be eradicated, has been eradicated from the eastern part of the country as a part of DDT spraying program of new government for eradicating malaria. That DDT spraying somehow kill that vector. That vector of the disease is the female sandfly. So at that time, after independence, a few years, we noticed that there is almost no Kala-Azar cases coming up. But in late 70s, the disease came back with full vengeance. The disease broke out in epidemic proportion in the state of Bihar and adjoining district, that West Bengal and the other Uttar Pradesh. That was also, as they share the common boundary, that human migration is between among the states. So that also badly affected. In 1977-78, Kala-Azar caused death of 4,000 people in India alone. In early 90s, again, it has been seen, it has come back again, and it has been seen that about 2 lakhs to 2.5 lakhs people were contracted with the disease. The situation has become worsened further due to the emergence. In the meantime, there is an emergence of the drug unresponsiveness among the patients toward the first line drug, sodium stevogluconate. The second line drugs used for that insensitive or unresponsive cases of the patients, that amphotericin B, pentamidine, paromycin, were toxic, costly, and less effective. And along with the disease, along came HIV. Co-infection with the HIV also made the control strategy inadequate. Now, that leishmaniasis is present all over the world. It is a disease of the tropical and the subtropical countries, and the diseases are broadly divided into 3 major categories. One is the visceral leishmaniasis or Kala-Azar, caused by Leishmania donovani complex, species complex. Cutaneous leishmaniasis, manifestation in skin by L. tropical or Leishmania tropica species complex. Mucocutaneous leishmaniasis by Mexicana complex and Braziliensis. PKDL is another, it is a peculiar sequel of Kala-Azar. It has been seen that 10 to 20% of the treated cases of Kala-Azar patients are showing the emergence that the disease called the post-Kala-Azar dermal leishmaniasis, that is a macular or nodular patches in the skin, and that is present peculiarly here. And it has been considered that as there is no reservoir found here in India, that PKDL cases are considered as the anthropogenic reservoir for the disease. So if we look at the global distribution of the leishmaniasis, and if we pinpoint only visceral leishmaniasis, then we will see that 90% of the VL cases are confined to the following countries: India, Bangladesh, Nepal, Sudan, and Northeast Brazil. If we come back, if we come back to Indian scenario, if you could see that in India, Bihar is always devastated with the disease. 31 district of Bihar, 11 district of West Bengal, Jharkhand, and 11 district of Uttar Pradesh are currently affected with the disease. Now, for prescribing any drug for any parasite, it is very much required, it is must to know the species status of the parasite, because the drug meant for one parasite, one pathogen may not work in other species. So before giving any drug regimen, it is required that its identification should be known. So for that identification purpose, for parasite, those who are morphologically similar, we know there is morphological taxonomy where morphology act as a marker. But here, marker for identification or recognition. But here, parasites under the microscope all look same. The amastigote or the promastigote, they look same. So clinically, they can be isolated depending on the disease profile, depending on the vector choice, depending on the host parasite interaction, they may be classified. But for the case of unicellular parasite, it is very difficult to distinguish them morphologically. So other techniques, along with the development of the technology, other techniques came into account. So with the molecular development of in the 60s, the purification techniques for enzymes and the proteins, of course, nucleic acids and other complex molecules, new molecular criterion for taxonomic classification of leishmania became available. Among them, multilocus enzyme electrophoresis is based on isozyme profiling, where a population is defined by this profiling, and this population, when is defined by a group of enzyme, then it is called, the population is designated with a zymodim number, and that is a species specific profile. So isozyme profiling is done by scoring the mobilities of the crude extract of parasites in electrophoretic field of native gel and stained for specific enzymes. And as I told, it is a species specific attribute. So here I'm showing you the migration of the some enzymes. The name of the enzymes here: nucleoside hydrolase, 6-phosphoglucose dehydrogenase, phosphoglucomutase, and isomerase, glucose phosphate isomerase in the panel A. They are showing that migration in the electrical field in cellulose acetate plate, and the strains or the WHO standard strains use is L. tropica. K27 is the WHO standard for L. tropica. DD8 is the WHO standard for Donovani, and other two lanes, one is UR6, other is AG83. These are the strains collected and characterized and kept stabilized in Indian of Chemical Biology. That UR6 is a difficult isolate for us because it has started showing some differences from it with the classical L. Donovani. So we have taken a step to identify that strain at molecular level, and we ultimately came up with the conclusion that it is neither L. Donovani nor L. tropica, though it has been collected from a confirmed Kala-Azar patient. So MLEE at least solves a problem of strain identification. And that strain, as I told you, it is widely used in Institute of Chemical Biology and some other laboratories for leishmania related work. So if we start with a wrong species, we will end up in a wrong conclusion. So that is the beauty of the identification process, taking the help of the technology. This is called the zymodeme profile. That, as I told, 10 to 20 enzymes will develop that profile, and we have to take many enzymes. It is not the single enzyme we will take and we'll do the gel running and we'll say that that is the zymodeme. It is not so. It is 10 to 20 enzymes we have to take, and we have to run the gel, and we have to see the band profile and score it, and see the similar band, see the dissimilar band, and calculate the Jaccard simulated index. And then you can say for a newly isolated clinical isolate that it belongs to L. donovani, or it belongs to L. tropica, or it belongs to some other species group. Now, with the advent of recombinant technology and other emerging tools and technologies of modern molecular and cell biological level, research in protozoan parasite has seen a magnificent change. Introduction of PCR technique based strategy has brought paradigm shift in the field of biological sciences, especially in the field of species identification, epidemiological surveillance of the disease, and the evolutionary biology, etc., molecular population genetics, etc. PCR and DNA based techniques, among them immediately PCR came into light, and it got Nobel Prize, Carey Mullis. That PCR technique has been started exploited in various fields, modifying the basic technique, and RAPD is emerging as that technique, which is known as randomly amplified polymorphic DNA. Unlike PCR, classical PCR reaction, here random primers are used for amplify the genome at certain point, and that amplification, it has been seen that amplification is done in a specific manner. That means RAPD is a species specific marker. So our lab started using that technique. Actually, I also started doing my PhD work that technique, and using that technique, several clinical isolates, say about 30 to 35 clinical isolates are typed and characterized. At that time, during the time of my research, Sachs et al. Claimed that Indian Kala-Jar can be caused by L. tropica, which is an impossible statement, because this species never related with the never related with visceral leishmaniasis. It causes the cutaneous leishmaniasis. So that time there is a great controversy that how come the L. tropica will be never. It has been reported from the eastern part of the country. It is present in India in the western part, and causing the cutaneous leishmaniasis in Rajasthan. And how come it come into here in eastern zone, where the L. donovani is in the endemic for L. donovani, the reason is endemic. So that was one claim, came out in 1995 in Lancet. And there is another peculiar occurrence of both L. donovani and tropica in the localized cutaneous leishmaniasis LCL patient from the northern part of the India, and that has been reported by Sharma et al. In 2005. So we have started when we have started, we have collected so many clinical isolates going to the field. That is, it is in the Jharkhand, it is in the different district of the West Bengal, like Birbhum, Dinajpur, like that. From there, we have collected so many clinical isolate, and in this process of collecting that isolate, we came across one clinical isolate that started behaving wrong, or started behaving, not it is wrong, differently in our RAPD or isozyme procedure. So we have checked that parasite. If you could see lane number 11 and 12, where 12 is K27, that L tropica, who recognized reference strain for L tropica, and immediately before that in lane 11, that T5, that T5 is given in lane 11, and if you see the profile, you can see that it is different from rest of the other clinical isolate who belong to L Donovan, who have matching with DD8 in lane 2, that it is matching with DD8, and T5 is matching with K27. So doing this, and also the RFLP, I am coming to that. Khanra, she is my PhD student, Supriya Khara. She reported the association of L tropica with the disease by this profiling. And with also that is RFLP. The RFLP is another technique that use the restriction enzyme to cut the genome in a specific site. If the site that cut site is mutated, then it will not cut that internal DNA sequence. That means that cleavage site is mutated, and it has been seen. It has been used for long in the species identification and characterization and strain identification before the PCR based technique came along. And that RFLP technique also showed very clearly, if you can see that lane number 17 and 18 here, and 17 and 18 in both the gel picture, you will see T5 is matching with K27, the standard strain of WHO, and all other clinical isolates matching with Donovan. So with this, phylogram is constructed, and where it has been seen that all the clinical isolates we have collected that time and characterized, they are clustered in one group, while T5 is present in some other place. Then another technique called single stranded. Would you please give me 1 minute? I'll take one sip of water. It is another technique known as single-stranded conformation polymorphism, SSCP. The worker in the field, they know the technique. It is simple and sensitive technique for mutation detection and genotyping. And it discovered and the genotype of the single nucleotide polymorphism, SNP. And the principle of the SSCP analysis is based on the fact that single-stranded DNA has a defined conformation, and altered conformation due to a single base change in a sequence can cause single-stranded DNA to migrate differently under non-denaturing electrophoresis condition. Based on that technique, my lab, they started genotyping through SSCP, and our results suggest that the resistant Leishmania isolate could be differentiated from the sensitive isolates by SSCP by profiling by doing that SSCP of ITS1, ITS2. These are the spacer, internal transcriber spacer, and the one anonymous marker L0114. Still another technique then we have used, it is the multi-locus typing, MLST. Multi-locus typing is a technique of typing the multiple locus of the isozyme, the enzyme we have taken, that enzyme of product of the housekeeping gene, and that gene loci may be typed for the characterization of the parasite. MLST appears based in population genetic study. The technique involves the PCR amplification followed by DNA sequencing. and it directly measured the DNA sequence variation in a set of housekeeping genes. Because housekeeping genes, why we are taking housekeeping genes? Because they change lately. They do not change so quickly. They are almost conserved over the period of evolution. If they change, that means the species changes. So that characterizes the change by the unique allelic profile, and the workflow is like that. It is not required for all of you. And we got that phylogenetic analysis of 6 housekeeping gene, that aconitis, alert, enol, and PGM, all that spermidine synthase. And we got from there, we got for alert and enol, we got 45 unique sequence typing ST3, and we also got ST6, where the T5 shares is shares the sequencing with 2 other clinical isolates, that T3 and T7. These are confirmed L. donovani strains, and T5, as I told you, it is a L. tropica, though it has been collected from a confirmed Kala-Azar patient. And then we are into another very ultimate of my dream, that is the sequencing of the whole genome of the parasite. For that, I have a collaboration with Professor Parthu Saha of Saha Institute of Nuclear Physics, and he gave me the chance to do the whole genome sequencing of all the clinical isolate we have collected so far. And the result of the sequencing analysis is that we have taken SSGR, that means drugs, SSGR resistant, SSGR sensitive, SSGR sensitive paracardial strains. Paracardial strains is the picadial strains collected from the visceral organ, not from the skin. And one peculiar isolate we have, that is the miltevosine resistant isolate. And from that sequencing analysis, we prepared the dendrogram, and aneuploidy, by the aneuploidy study, we could group our clinical isolate in 3 different zones. That is the aneuploidy profiles of the L. donovani strains obtained from whole genome analysis of the clinical isolates, and the chromosomes are seen. They are basically diploid, but tetraploid, triploid, pentaploid also seen in our study. And based on that list of genes mutated in the paracardial, when we are searching over the database, and that what are the genes mutated in case of paracardial strains, then we have come up with this report that these are the genes that are related to the cellular. Then we check, that is also through the database. That is another tremendous progress in the field of technology, that database bases are available for doing many work. All are it is the repository and the information base, and it is the dry lab work. So cellular process, those cellular process that with the genes, that is we see that cellular process involve metabolism, ion transport, repair, phosphatase, helicase, all that thing. On the basis of that pathway analysis, we have started doing the pathway analysis of the 61 genes, which is all over mutated in the all strains. We among the thousands of genes, we ultimately came down to 61 genes, and that all thing we have done through the dry lab and publish a paper. And then the predicted secondary structure of the mutated genes for calcium translocating P-type ATPase ion transporter. All we are looking for, actually looking for surface protein or the transporter, because these are all related to, we are looking the what are the genes modified or enhanced or anything for the drug resistance that can be associated with the drugs resistance. And drug resistance is a serious problem here nowadays. All the drugs are now insensitive to the SAG, as I told earlier. And miltevosine, we are using the miltevosine, oral drug miltevosine. That is also that is a cancer drug, oral cancer drug, that in the when we have started using, Indian government gave permission to use that miltevosine, but after few years of using that drug, again the resistance, it is the common phenomenon, we all know, the resistance again and again came out, and we got isolate, that is miltefosine resistant. So our target was to see these things and the predicted model. And in doing so, in the analysis of the 3D protein structure of the predicted secondary, it is all the dry lab work where we could identify that if we align the wild type protein with the mutated protein, and if we align, we could see that there is a deletion in that, the arrow showed that there is a deletion in the coil from the in the mutated protein. These are the softwares and programs used in the study. So in conclusion, I may say that species identification of any parasite is a must for epidemiology and taxonomy, for proper drug regimen, and for phylogenetic relationship analysis. And hierarchy of techniques employed for such studies in my laboratory is microscopy, isoenzyme profiling, polymerase chain reaction, SSCP analysis, MLST, and ultimately whole genome sequencing. They are my PhD students and the research fellows. Actually, today's talk based on the work of Supriya Khara, who is now in University of Glasgow doing her postdoc, and Srimati Nivedita Rani Saraf. She is my PhD student. All whole genome sequencing work is actually her contribution to my lab, and Shonchita and they are doing other works. My collaborators are Professor Shamul Rai, Professor Parthu Saha, Professor Ranjan Kumar Das, Dr. Anindita Chakraborty, and Arghya Adhikari. Thank you. কোথা থেকে আবার আসবো আমি, এটা কেটে দেবো?
Thank you, ma'am.
না, কোথায় আসবো?
Thank you, ma'am, for the speech.
On behalf of Seminar Organizing Committee, I, Akash Chaudhary and Vishwajyoti Roy, Organizing Secretary, express our heartfelt gratitude. Ma'am?
Yes, I am here.
She has the supremacy in both the fundamental molecular biology and medical biology research, as well as in administrative ability. We salute such an eminent international scientist, as well as the excellent administrative officer, Government of West Bengal, India, who expend a beautiful time excluding the extreme busy schedule with us, and we are very happy and express our heartfelt gratitude to our honorable invited speaker, respected ma'am. Now I request our officer in charge, Dr. Mohik Sarkar, as well as president of this program, to summarize the lecture of our honorable additional directorate of public instruction, education director, Government of West Bengal, India. Over to you, sir.
Thank you. Thank you, madam, for this excellent speech. The main thing is that when we are going for these any lectures, particularly we have to go for our history, and then we go for the other things. Any type of research we are going to do, first of all, that is the identification. So, excellently you have identified that Listonia donovani and Leishmania tropica, because this is one of the major disease-causing agent in our area. Particularly, I saw that one in the Uttar Dirajpur area. This Kaladar is very much prevalent over here. Still now, it's very much prevalent. And you have briefed up that particularly in 1824. We don't know also that in the Jeshore district of Bangladesh, it has happened first. And then in the India, it had came. And about this, the students know that UN Brahmachari's contribution, this ureastibamine, particularly the ureastibamine, one of the magic bullet. Yes, definitely it was a magic bullet at that time. And we don't know anything about that matter, about this sand type, about this Kaladar, how it's happened. And this thing is beautifully explained, probably. And particularly I saw that when you are going to do any type of research, then this type of things should be designed first. One thing is that one that you have to identify your things. Then first one is that's why it's a microscopy. Then you have to go for the isozyme study. Definitely, when we have nothing in our system, so we have to go for the isozyme study. This is the first and foremost thing, that protein detection is very much important. Then after that, this PCR technique has came. So automatically the PCR technique and the RAPD method, that randomly amplified polymorphic DNA analysis, is very much important components for analysis of anything. Then it's a RFLP, and then SSCP and the MLST. And then you have done the whole genome sequencing. It's an excellent work, because this is the technique from lab to land technology, because we need this type of technology in the lab to land, otherwise we cannot go for the things. So students must learn from this type of research, that how we can approach. When the PhD students will join in our any laboratory, they should know that how to approach first. So for the 1 or 2 years, they have to first go for this type of things, that identification. Because in our system, as a entomologist, I know that identification is the most important component in our system. In the biological science, first you have to identify which one is a positive agent. So that positive agent, is most important. Then you have to step by step you have to do. And in your lecture, we have saw this is a complete picture of a Kala-Azar major disease, and we have studied in a very elaborate way. So thank you, madam, for this excellent speech. Because main thing is that students learn we have learned lots of things. We don't know anything about that matter also little bit, but still it's a very good lecture as that one. And anybody has any questions? So madam is busy, I know that one.
Actually, my madam is calling. I cut her phone. She is calling.
Okay, thank you, ma'am. Thank you very much.
Okay. If somebody has any question, please come to me over the phone or any way, and I can help if I can. If anybody does research in this area, and not in the Kala-Azar, any area, if anybody needs any help in doing this sort of work, I'm there. I'll be there. So thank you, Moyuk. Thank you all.
Thank you, ma'am.
I'm leaving, though I have a desire to listen to other eminent speaker, but as madam is calling me, so excuse me, and thank you all.
Thank you.
Bye. See you.
Thank you.
In this second day of International Multidisciplinary Seminar on Modern Trends in Humanities, Science and Technology and Social Sciences for Sustainable Development, we already, in the first session, one special invited speaker was there, Professor Madhumita Manna, Additional Director of Public Instruction Administration, Educational Directorate, Government of West Bengal, India. She delivered her speech. Now the session is for invited speech. We have different invited speech from different country. First speaker is Professor Indrajit, Disaster Preparedness, Mitigation and Management Section, Asian Institute of Technology, Thailand. Next, we have the technical session designed for life sciences, as it is being introduced into the schedule, and I think all of you are going through the schedule. Specific links are there, and you will enjoy or you will present your oral presentation to the specific link. And technical session 3, that means for life science, it has major 3 sections: section 3A, section 3B, and section 3C. And another part, there is a after that session we have a break, and today's second technical session, which is designed as 4A, 4B, and 4C, is specially designed for physical, chemical, technological, and mathematical science. So this is the overall briefing of the today's program. We have another invited speech, international speaker, Dr. Kaushik Das, Department of Physics, University of Maryland, United States of America. And then the valedictory session, and in that section we have some important declaration about the feedback form, about the certificate, and all other things. Presenters who are in this session and all the delegates who are in this session are humbly requested from the part of the organizing committee to bear with us. We are looking for the entry of our invited speech, international speaker, Dr. Indrajit.
Akash sir, Indrajit sir will join soon, within 12:25, I think.
Vishwa sir, I think we got Professor Indrajit Paul.
Okay.
Sir, Indrajit Paul, sir, are you in web?
Yes.
Okay, thank you, sir. Thank you, sir.
Okay.
Today is the second day of today's international interdisciplinary web seminar on modern trends in humanities, science and technology, and social sciences for sustainable development. In this section, we had a special speech from Professor Madhumita Manna, which has already been completed. Now the session is for invited speaker is Dr. Indrajit Paul, Director, Preparedness, Mitigation and Management, Asian Institute of Technology, Highlight. Professor Paul is a visiting professor of disaster risk management in Philippine School of Business Administration, Manila. Integrated research on disaster risk, China, in which section he is a young scientist. He was a former faculty of Center for Disaster Management, Lal Bahadur Shastri, National Academy of Administration, India. We have with us, this is our privilege on behalf of the organizing committee, seminar, APCDA Government College, West Bengal, India. On this international web platform, I heartily welcome Professor Indrajit Pal to deliver his invited speech. Now it is over to you, sir. Professor Indrajit Pal. Professor Pal, your presentation is visible.
Yes, sir. Visible, sir.
Oh,
Your presentation is visible.
my presentation is okay?
Yes, sir. Yes, sir.
Yes, sir. Yes.
Okay. So thank you so much. It's my privilege also to be with you, and I always love to be present the perspective of the Asian sort of countries in India as well as our sort of in West Bengal. So I used to also deliver a couple of lectures in other universities in all across India.
Sir, you are not audible.
I'm not audible?
Audible, but the volume, less volume.
Better now?
Yes. Yes, sir.
Yes,
Please.
sir. Yes. Yes, sir.
Okay.
Please.
Yeah. So actually, normally we are using, Zoom, and Zoom is taking default. There are 2-3 mics, so it's taking other mics also. Anyways, I'm a full-time faculty member at Asian Institute of Technology, Thailand. We have a disaster preparedness, mitigation, and management program. This is an interdisciplinary program located at Thailand, and I'm also faculty member in other 2 universities as a visiting in China and in Philippines. So I have a couple of projects right now going on in this region. One of the project is under UN University on the higher education, trying to look into the higher education is also in the different universities and how we can develop the multidisciplinary course curriculum. So I am also helping in developing course curriculum for the disaster management for multiple universities. I'm also a co-principal investigator for UK funded one of the largest project in this region for Delta resilience. That's one part is also in Sundarban, India, and Sundarban, Bangladesh, and Vietnam. So, and some of the projects also I'm involved in World Bank with disaster resilience perspective in Sindh province, Pakistan, and some of the projects also in Myanmar and other countries as well. So first, actually, I'll give you some sort of perspective where I am from and what actually we are doing in terms of the disaster management, because disaster management subject in India and also in West Bengal as a degree program, not very much sort of popular. So we have not many universities who are offering degree program on disaster management in India and West Bengal as well. So here actually we are sort of higher education organization. So it's only masters and PhD we are offering. So we don't have any bachelor degree program here. So this is our global outreach to different countries. Like the students are from all these countries plotted here. So it's about 24 countries across the globe. So we are trying to highlight some of the issues. I'll show you some of the diagrams as well after that, after a few slides. So on the disaster governance, policy and management, how the disaster management policy should be looked into, what are the analysis we can do on the policy, remote sensing application and GIS for the disaster mitigation, community-based approach to reduce the disaster risk, multi-hazard vulnerability, flood and drought specifically, and also climate-related hazards and climate change-related issues and adaptation. This is the hierarchy actually we are following in the higher education curriculum. So I think it's also useful for the universities like in some of the universities as well. So we are following the normal course curriculum as well as like the projects, and the students are actually giving opportunity to the projects to work on as a sort of intern, so that they can get, during their study, they can get the sort of exposure to work on the project and how actually on the real world field they should go. And then also short programs and other international programs also we are organizing. So this is a brief about the different type of degrees actually on the disaster management you are offering. So anyways, these are some of the highlights about the student activities on the field. So apart from the class teaching, we're also taking students to the field to look into the different perspective. We have some exchange program with Japan. This is one of the program we're organizing with Kyoto University, Japan, to expose. It's a visiting program with Kyoto University. So students are coming from Japan to here. There are 4 or 5 countries actually together moving around in 2 countries. But for last 2 years, because of the COVID, it's difficult to move for the travel. So we are doing online this same thing. This is another program I started last year, 2019. This is the first batch with one of the university in China, Central South University. So about 6-7 students we have sent as an exchange program to China, and accordingly their part also sometimes comes. This is the recent international program with 21 partner university across the globe.
Sorry to interfere. The slide is not moving.
Not moving?
No, sir.
Which slide right now you can see?
Sir, first slide.
First slide.
Yes, sir.
Now it's moving?
No, sir. Actually, it is not displaying. Actually, no.
It is not visible, sir.
Visible, sir.
Now?
Yes, sir. It is visible now.
Again, the first slide, or it's moving?
No. It is changing.
It is changing, sir. No.
Maybe there are lag time. So, okay. So what I was saying, this is the footprint of our education program, like on the disaster, from where all the students are coming to our department for the degree, like about 24 countries. I'm not putting full screen. I think somehow there is a problem. So.
Not an issue, sir. It's okay.
Is it okay now? Is it okay now? Full screen?
Yes, sir.
Okay. Thank you.
Yes, sir. Yes, sir.
So these are the areas actually I just highlighted about the key research areas we are focusing on the research. So the research is not. The research on the disaster management is a contemporary studies right now. Whatever is required for the locality or the community and the government, we're trying to do that type of research, which is actually will be useful for the policy making and other things. So this is also I showed the diagram, like how we are progressing in the different steps of the students also in the education, higher education, and how we are involving students in the fellowship program, in the international movement, in exchange program, and also apart from the regular degree program. So within the degree program is embedded degrees, and some of the field visit I'd like to show you, like how students are actually giving opportunity to visit the disaster impacted areas or the departments who are controlling the disaster management. This is the exchange program I was showing with Kyoto University, Japan. And this one is with one of the Chinese university, Central South University in Changsha, China. So So it started recently with our department. My slides are moving, I believe.
No, sir. Now it is still.
No,
Still?
sir, it is still now. Yes, sir, it is not moving now.
Yes, it's changed.
Yes, sir, it is moving now. Moving.
Okay, okay, there are something wrong. Okay, problem with the Google Meet maybe. Okay, so this is the international program actually we organized in AIT recently with 21 sort of partner university across the globe. So these are the partner university actually working in developmental field, and also some universities from UK and Australia on board. We also called executive secretary of UN ESCAP as a chief guest for our program on disaster resilience and sustainable development. So hopefully, I mean, almost we are organizing yearly basis or maybe 2 years gap. So if you are interested, maybe I will also invite all of you for the next program. This is the area actually right now. Maybe whenever we are talking about the disaster management, these are the different terms coming up. So maybe we'll discuss some of the terms here in the context of the recent report published in 2020. If you see the different dots, extreme temperature, floods, there are 127 floods in this region, 59 storms or cyclones, 25 landslides. So there are all, if you see like the hydrometeorological hazards are more in this region. So our concern is like this part. So if the slides are not moving with my talk, so please let me know, because then I'll sort of check.
Now we see the world map, I think, again.
World map with the dots, right?
With the dots,
Yes. Thank you. So these are the area actually in the Southeast Asian part. If you see it's dotted with different colors, that means actually that this is the record of the different disasters happened in last few years. So blue and green color actually signifies about the meteorological and hydrological events, that is flood and cyclones and like this, which is impacted like by the climate change, more or less. So in this part of the world, like Southeastern Asia, if you see, is too much of disasters happening. So we need to be sort of more sort of mitigational measures to be taken. So if you see on the perspective of the loss, disaster related loss, it's a huge loss. I mean, we West Bengal, we know very much very well about the loss of the disaster, because very frequently West Bengal also facing a lot of disasters. I'm very much aware about this. Between 2004 to 13, about 43% of the total record global disasters occurred in the region, like in the Southeastern Asian region or Asia Pacific region, and the total cost is about $73 billion US dollar. So this is an enormous amount of money, and a certain percentage of GDP also you can consider. Asia Pacific record, like if you see with the two things here, one is the economic loss, another is the occurrence. So economic loss is very much with the occurrence. So here we have a huge economic loss in a big spike. That means like in 2011, this is 2011 point. In 2011 in the Southeast Asia, there are two major disasters happened. A very, very big size disaster. All of you know about the Japan tsunami 2011 and also nuclear meltdown. So that is one. And second, also one of the largest flood happened in Thailand. So almost 90% of the area was inundated in Thailand. So 2011 was the highest sort of loss in this time, if you see both the disasters together. So these are the global data. Actually, I have given sort of plot. I mean, tried to show you to have some sort of understanding about like how important disaster management is in the region. We should also think about more educational program as well to have more knowledge about this, so that we can do more research and get more understanding, and accordingly we can prepare ourselves. So this is about like the region. If you see the region wise, the blue color signifies about on Asia. So Asia is highest in terms of the affected and also in terms of the damage. Number of killed may be less compared to Africa, because Africa there are some other disasters like drought, so that killed a lot many people. But in terms of the affected and damage, there are a lot many happening in the Asian part compared to other part of the world. It's a regional map. In terms of the occurrence of the different type of disasters, drought, flood, earthquake, and storms, also we can see like the epidemic is taking a large number here. And also if you see the storm, this is the storm number, that also increasing in the last few years. So this is the data from 2019, all the recent data. Again, like there are two things actually we normally see for the disaster management and for the country perspective. So all the countries are plotted here to look into which country is exposed to what type of disasters and which country is actually having more vulnerability. So in terms of the disaster, sort of split into two parts. So x-axis here is about the vulnerability index, and y-axis is exposure index. Some countries too much exposed to different type of activities, different type of events, different type of sort of hazards, like say Indonesia. Indonesia is here located. Its index is moderate compared to Nepal and Pakistan. If you see Afghanistan, Pakistan, and Nepal on the exposure term, they are on very high index because the potential of the hazards are very high in these countries. On the other side, if you see the vulnerability index, so there are some other countries which are vulnerability index are very high. Okay, for some sort of for the correct calculation of the indexing, we need also some sort of specific indicators. This one is very interesting one, like from which time we are using what type of technologies. We all know about the satellite imageries. We all know about the UAV. Now it's popularly known as drone, sensor-based approach or IoT, Internet of Things. LiDAR data is also very high resolution, three-dimensional data we can get. Simulation, computer simulation, all other spatial data. Crowdsourcing is also another part, but it started, if you see the color, it started very recently, like in 2013, 14, it's coming up like this. And social media is also another way. So all these things actually we can see which year, what type of technology actually right now we are using and for what purposes we are using. Now I am showing one pie chart. Is my slides moving accordingly?
Yes, it is moving.
Yes, sir. Yes, sir.
Okay, so this is a simple comparison. If you can look into, it will give you some idea why we are not able to sort of mitigate the disaster too much in India, and why other countries also able to mitigate or not that much of loss or death for the disaster. So we have compared like three countries, three different economy. One from India, another from Thailand, like it's middle income country, and the third one is the Japan, high income country. So we have low middle income country, high middle income country, and high income country as per the World Bank categorization. So if you see Japan, they are putting about 6% of their GDP, 6% of their GDP for the disaster risk reduction related activities from the national budget. Thailand is spending. So as the percentage of expenditure will increase, per person expenditure also will increase. So the per person expenditure for the disaster management will increase. If it increases, then obviously we have more sort of mitigational measures, more other preparedness activities can be done. So these are the scenario we can also do for other countries as well. But this is for the comparison of three different economy where we are placed. Although it's a little bit of old data, but you can get the pattern. So the percentage may vary a little bit based on the national budget document. So it's a 2016 document, but you'll get the pattern how we are expenditure is allocated. This is very popular cycle actually called disaster management continuum. we are in different phases, like in the disaster. So we have broadly divided into pre and post disaster phase. So in post disaster phase, actually the crisis management and need assessment, response, recovery, on all these things are going on. And when there are no disasters, we call the pre disaster or less disaster, or after the recovery of the disaster. So then that prediction of the prediction and early warning system, mitigation, preparedness, capacity development, all these things can go on. Eventually, what actually the point is, we are trying to move from the crisis management phase to the risk management phase. If we start preparing ourselves when we got the cyclone warning, then maybe it's not a good idea to have a good preparation, because we don't have anything to prepare for. In two days or 3 days, we cannot prepare ourselves, although we get the information. But if we can prepare beforehand, like if cyclone comes, then what are the cyclone shelter we will use, what type of sort of methods we will apply for protection. So all these things should be pre-planned. So we cannot, that is called the risk management. So we can plan early so that it can be mitigated. When the flood comes, then putting some boats and other things will not help us. So if we know how to sort of predict the flood with the rainfall, like if we get the rainfall pattern and try to understand, okay, there is a high possibility of flood in next 2 months, then the government and the local authority can prepare themselves. So we can develop this type of model as well. So these are the area actually right now global community is working on, including India. India, we have very sort of a good step actually we have taken from the crisis management process to the risk management. Now I think all of you are aware about the disaster management law. Before COVID, nobody was very much aware about the disaster management law, but it started in 2005. But only I think 100% people in the country possibly know about the disaster management law and act, because it's exercising its power through different process. These are the broad dimension how we calculate the risk of a particular location. So we can understand the hazard, what are the hazard actually can come into that location, what are the exposure of that particular location, and the vulnerability of different types. So these are the few area actually we can calculate the risk. This is very interesting diagram actually came from IPCC AR5. Recent one is IPCC AR6, came few in last month actually. IPCC AR5 is actually 2014 release, and there on the climate change context, these are the definition or the way of risk assessment in terms of the hazard, vulnerability, and exposure. I'm not going to the theoretical background right now, just to give you some sort of overview how the calculation has been done. I think all of you know about the, now I'll explain about 2-3 slides in a very simple manner, the meaning of the resilience. What is the meaning of the resilience? The community or the person or the country or the locality which can come back to the normal situation after the disaster faster, that process is called the resilience. If some community or some person is having high resilience, they can come back faster. And if the country or the individual or the group of people or the community having less resilience, they will take time to come back to the normal situation. This type of toy, I think all of you have we have seen. It's called it can come back to the knees. They believe like bamboo, and that's why like in Feng Shui also bamboo is small bamboo tree is also sort of China and Japan also putting it as a worship. So this is that the idea is like they also think like bamboo is a great example of a resilience. Like the bamboo tree during the winter time, there are huge snowfall in Japan in some majority of the places. So with the snow, the bamboo tree will bend, right, with the weight of the snow, but it will not die. When the snow melt after a few months, then the bamboo tree will come back to the normal situation and grow. So this is also another natural example of the resilience. How if this is very hard and we have no capacity to survive during that time, then the tree will die. But if you go in a theoretical perspective, so this is the resilience looks like. We have normal curve with the time. is going on, when there is an event like disaster or any type of shocks, it will go down. Then it will try to come back to the normal line again, in the same line. Like this is a linear one. So it will come back, trying to come back to this line. But if the resilience is high, then it will come, it can come here, like it will take less time to recover and come back to the normal line. And if the resilience is low, then the same line will take more time. Same resilience process will take more time to come back to the normal situation. So this is, as a whole, is called resilience curve. It can be drawn for any other situation also. This is another example, like to understand like what is the meaning of hazard, what is the meaning of exposure, what is the meaning of risk in a simple term. So this is a coastal area, coastal sea beach. So in sea beach, all of you know, like UV ray is higher. So that's why we put the sunscreen with SPF 50 and something like this with the sunscreen protection. I think sort of men possibly in this group not very much aware about SP 50 and other numbers, but I am sure like others will know. So the time shows here is 11 o'clock or 10 o'clock in the morning. So there are two couples taking sun bath. So if you see, like one are prepared with umbrella, another is not prepared with umbrella. But the exposure of UV ray or sun rays are same for both the groups. So after certain time, like about 1 o'clock, means about 3 hours, they are exposed for 3 hours in the same situation. Now without the umbrella and without umbrella and with umbrella. So without the umbrella, that couple got sunburn, and with umbrella they are also having same exposure, but they have no sunburn. So this umbrella is called here in terms of the mitigation. So they have tried to protect themselves through that process of mitigation. So if there is a flood possibility, we know there is a flood possibility, we can take some sort of mitigational measures to reduce the impact of that particular hazard. So here the hazard is the UV rays or sun rays, and exposure is the area they are exposed to, and the risk is that sunburn. This is the definition, but if you go in the left side, it's a cartoon, but I think you will also get some understanding about what is the meaning of the hazard. So there are many hazards roaming around, and with that hazard, many type of accidents or disaster can also happen. Okay, in class maybe we can have more time to identify how many hazards you can identify, something like this. But since it's not the class, maybe we can skip. Now I'll show you some sort of example. This is the example from the Tarai area, the lower part, southern part of Nepal. Two satellite imageries of the same areas. So if you see, like in 2004, this is a river, and this is the area is floodplain. So during monsoon time, there is a possibility of flood, but maybe due to the low water flow or some reason, there is no flooding for many years. So people think like, okay, this is a dry area and there is no flood happening, so we can construct. So if you see the date here, 2009, 2005, 4. So in 5 years, you can see like the area is completely habitated. So there are many construction happened, but obviously now maybe it's more. It's 2009. You see, like just to compare the two part. So this area is now more and more exposed. These people and this whole establishment is exposed to the flood hazard. This is the natural channel. If some high amount of rainfall is happening in the upstream, the water will flow through the natural channel through this. So it's not the fault of the river, not the fault of the rainfall also. We are constructing our houses on the river, right? So once this river will for the flood, the entire area will be washed away, then the whole community will ask for the compensation and also for the other things. But and also there will be a huge amount of life loss and property loss as well. So who is on the fault? I am not going to discuss that. That is a governance issue, and it's the implementation of the policy or ignorance of the policy or what, that we have to also. But as a whole, we call unplanned urbanization. There are some other examples also I'll show you from other parts of the world. And these are the similar example when they are constructing these houses on the river. Then you can see like they are more and more vulnerable for the flood hazards. This is also I think the university is located in Siliguri, and I think Kalimpong and Darjeeling and Sikkim is also very close. This is not from Sikkim and Darjeeling. This is from the northern part of India. But I think we can see the similar type of structure in our other places also. So this is nothing but an unplanned land use. So we can see like the mountain area is prone for the earthquake and landslide, and there we really need to take precaution or measure to have this type of construction of multiple stories. If not, then what are the consequences? We have seen the Uttarakhand incident in 2013, that flash flood. So flash flood has not followed the other routes. It simply followed their own river channel. Only thing is, if you see the location of the houses is completely located on the river. On the river. So all these are washed away like this. There are government establishment even. So that means like we have to have some sort of proper plan with the risk mapping. We need to understand like if something goes sort of high level of water flow, this area obviously will be flooded. But it's possible to know that. It's not a very sort of complex issue. It can be sort of predicted to some extent. Finally, this is the line actually I need to explain a little bit. The hazard impact people depends on their vulnerability. Means like if the high water flow or the flood water is the hazard, it depends on the person's location or the vulnerability where you are located. So based on your location, that hazard may turn into a disaster or may not turn into a disaster. Say for example, I'll going back to the same picture. The people who have constructed houses here or on the upper side, they are not exactly on the riverbed. So they are less vulnerable to the flood if something happens. So this is actually the same thing, like those who are exposed to the location of the hazard, they are more vulnerable, and that whole thing can be disastrous. I'll show you now a few examples what actually we are doing in terms of different activities. This is also one of the things we can develop a dashboard where we can understand what type of vulnerability that particular location having. So we have used like geodata and some satellite based information. This is one of the provinces, one of the state actually, one of the district in Thailand, very close to Bangkok, about 60 kilometers from Bangkok. So this area, this is actually the old capital of Thailand, Ayutthaya. So there we have tried to see like different houses, different type of houses, and which house are vulnerable to what type of condition. So we have tried to map into 3 segments. The houses with sort of low height, houses with little bit of higher elevation like 2-story, and houses with stilt. So the underground, I mean the first floor of the house is actually void with the pillar. So that area is frequently flooded. Almost every year there are floods. So if you see the roads also nearby, so we can have some parameters to compare. And then there are some dashboard created to understand like which are the area will be inundated if there is a flood and which area will be less inundated. So we can also calculate. So there are some link actually on the Google, S3 story map. If you want, I can share this link also. You can directly go online and check how what will happen if there is a flood, which area will be flooded and which area will be not.
Yes, sir. It will be helpful for us. Please share the link in chat box.
Yes. Yes, I shared already.
Thank you, sir.
Yeah. Can you see my
Thank
screen
you, sir.
also? The same dashboard
Yes,
screen?
sir.
Yeah.
Yes,
So
sir.
this is okay. So this is one of the example. Actually, this is not a full scale project. This is a student project. This is a master student project, master student project, thesis project actually. This is the project actually I was talking about as a full scale at the country level. So here we are trying to see the 3 countries and 4 delta. So the project name is Living Delta. Newcastle University from UK are the principal investigator. I am one of the co-principal investigator from the regional side. To understand the complex development challenges, like what are the challenges that Delta people are facing in terms of the multiple dimensions, not only disaster, plus other things as well. So eventually we are trying to check about the socio-ecological system of the Delta. So this is the area actually where we are doing our study. It's close to Hanoi. It's called Red River Delta, starting from China, that river. Another is Mekong River, which is also starting from China, across going across 5 countries. And Vietnam is the last country. And this is the delta actually we are trying to look into. And Ganga, Brahmaputra, and Meghna Delta, GBM in short. So that delta is actually spread across Bangladesh and India, both sides. So we are trying to look into both the sides actually. So these are the study area we are right now focusing on. The research is still going on. This is also another project, as I was mentioning, with 4 other universities, 2 from Japan and 2 from Indonesia, trying to look into like the international framework for the disaster management, that is called Sendai Framework for Disaster Risk Reduction, that is in short SFDRR. And all of you know about the SDGs and the Sustainable Development Goals. So how we can integrate these 2 things, development perspective and also disaster risk reduction, 2 things in the higher education. Because if we have nothing with the higher education, then who will take care about this sort of research component and how the things will go on? So we need to also develop that type of cadre or that type of group who know all these things. Okay, so this is another project right now going in to understand the risk profiling of the southern part of Pakistan actually. So this is to understand the erosion potential, and also if there is a sea level rise, then which are the areas will be inundated, and what will be the cost of the loss of that area. This is another activity right now going on after the Amphan, which are the area actually last year, which are the area inundated, and how that dual disaster, because right now we are facing not only in Kolkata, in other country also, like there are existing COVID problem with all the countries, and with the COVID problem, with all this restriction, if there are other disasters are coming, how the government and the people are reacting or helping or doing the other things like the response and rescue. We have seen some incident in Vietnam. We have seen some incident in Indonesia in last year and this year also. We call dual disaster impact. This is one study I was doing in Myanmar. So to understand the resilience assessment for the community level. So we have taken flood affected community. So what we have seen in that particular study, I will share the journal paper also if you want. If you have interest, you can read through that paper. So where we have actually seen the community having a lot of potential to protect themselves without any other type of helps. So if we can develop the community resilience within ourselves with a lot of things there, so we can see the natural capital, social capital, human capital, all these things can be looked into. So we have tried to see like how it is playing a role to develop the community resilience in Myanmar. But this type of formula can be also applied to other countries as well. This is the study of Amphan. I think you are very much aware about the Amphan incident, but The point here is after the incident, what are the areas actually we need to look into. So we also came up with some international publication after that, taking consideration about the pre-existing vulnerability of the human health in Sundarban, because already that particular area having some problems beforehand, before the COVID and before the Amphan also. But add on to that, there are two more disasters came. So now how the vulnerability of that particular location increased. So that means like the vulnerability of the Sundarban community in 2018 and 2021 is completely different if you take the existing vulnerability, pre-existing vulnerability. We have tried to come up with some sort of understanding, conceptual diagram, like how it is actually impacting in the human health. I'm sorry, like I'm going a little bit of faster because to give you some sort of insight of some of the activities.
Sir, you have enough time. Carry on. No problem.
Yeah. Okay. So this is the disaster management cycle actually, what we also showed before. So in all the parts, actually the problem is right now with the COVID, we cannot follow the normal pattern of each of the segment. Say for example, like you are aware about like during cyclone, we have very good system of pre-cyclone warning, right? So 3 days, 4 days before you got the warning information. Everyone got the information. But even you get the information, what you will do? We got the information, that's good. But what we have done after getting the information? Are you able to protect the tree to fall? I mean, I am talking about Amphan. We got information, we have only thing what we have done, like a large number of evacuation. That evacuation is also maybe not very much organized because we don't have that many number of evacuation center or cyclone shelters where it can be protected. But the challenge is for all the governments and all the countries right now is the shelter is meant for certain number of people. But now we have to follow the COVID protocol or the process also, all the hygiene and everything. So keeping all these things in mind, it's very difficult to sort of accommodate a large number of people in the designated location. It's not possible for any government to throughout the world. So that is very challenging area. But the point is, apart from the evacuation, if we are prepared, means like if we can anticipate, like if there are cyclone stuck in Kolkata or in some area where we have lot many trees, so there is a high possibility that tree will fall with because tree having some sort of capability to withstand certain speed of wind. After that, it cannot, that particular tree. So that is very much possible to understand. So if we have that understanding before, we can chalk out some plan like, okay, this area might have problems, so that alternative electricity or some water system can be planned. Okay, I think we are better planned in the next one. Like in Yas, we have a much better plan than Amphan because we have some experience. But every time we cannot wait for the experience, and then it will be too late for the next disaster. Okay, the red dots here actually signifies these are the areas where we have really problem during the COVID time. COVID time, addressing the evacuation plan is not possible. I mean, as per the normal time. Shelter is another problem. Evacuation drill also not possible because we cannot organize any physical training and physical activity. Health system capacity is also right now we are in much better situation, but in between, like during Amphan time, it was really bad. The health system is already overbooked. Then during cyclone or during some flood, if there are some more number of people affected or impacted by that particular disaster, then you have no place to go because health system is already full. So this is also another challenge. All red dots are temporarily overbooked or overwhelmed due to the COVID situation. Emergency response is also very much challenging. Continuing the basic services like the water, electricity, and other things, medical services, aid and relied distribution. So aid distribution is also a big issue But now with the changing situation, I think this is getting much better now. Medical assistance also, it was the challenge. So all this point we have mentioned, keeping in mind the Amphan situation, what are the exact problems, exact area of sort of impact we really faced due to the cyclone and Amphan together. Okay, maybe you can skip that. This is another example actually from the eastern part of India. So if you see, the eastern part of India is facing a lot of cyclones. So we all know about these names of cyclones. So these are the impact for the cyclone in the east coast of India. So the east coast of India is actually highly vulnerable for multiple tropical cyclone. In short, we call TC. So tropical cyclone is impacting here. Western part is not that much impacted by the tropical cyclone. It is impacting, but it's not that much. This is the flood inundation or flood risk analysis for Kerala. I think all of you are aware about 2018 massive flood in Kerala. So this is also some of the remote sensing based sort of analysis we are trying to do. This one I showed you about some of the examples in Pakistan, the erosion. So these are the examples, and these are the international partners right now. We are especially for the disaster management institute having lot many other partners, entire institute, but this is I am showing only for the disaster management group. We are partnering with these universities and organization across the globe, and these are the active partners actually. So the students are going either or working with that group, or we have some active projects, or something is going on with that partners. These are the few books actually we published, and there are some more, but I could not put it everything. It was very small. So these are the few books. With that, I'd like to thank you so much. So if you have any questions, I am happy to answer.
Thank you, Dr. Indrajit sir. We are very grateful to you as you are present in our seminar today. I also thank Tuhin Ghosh sir. Tuhin Ghosh sir is a professor, School of Oceanographic Studies in the University of Jadavpur. Actually, he give you your number and you are agreed to deliver a talk in our seminar. We are very much enlightened through your seminar. Especially, you are touching so many areas regarding the disasters like tsunami, economic laws, Asia condition of Asia, and the low, middle, high income countries who are actually how much investing their GDP in the high disaster management. So, and the sort of resilience you are talking about, we are very much enlightened through your speech. Thank you, sir. And one thing you show in one slide, that is Nepal situation of a river bed, which
Yes.
showing the satellite imagery of 2004, and you were showing the satellite imagery of 2009. We are seeing over there that habitants they are going houses in the river bed. They actually they are the vulnerable people, but this is not the fault of the. I think this is not the fault of the government. This is the fault of the people itself, because they know that this is the course of river, and since we are getting less land in less amount of money, so we are constructing our houses in that river bed area. So obviously they are putting themselves in vulnerable section. So far my knowledge is concerned, it is in Siliguri also, sir. If you see the Mahananda rivers, river bed, if
Yeah, I know. Yes.
if you analyze the satellite imagery, you can also see the scenario is the same, nothing, and they are blaming the political parties that what the political party are doing during this time. And you are sitting over there in a rich zone, and political party will do for you.
No, and the problem is like, like this is nothing new. I mean, this is not only, don't think it's only happening in Nepal or India. I can give you at least 15 example from 15 countries of similar type. The point is like we don't see our own fault normally.
Exactly.
But it's a both side actually. It's not only the people, it's also the government, because government's responsibility, whether it's a local government or the national or state, is their responsibility to govern. Means where you are doing wrong, so there are some organization or some authority, they should control. If I am doing something wrong in terms of the crime, then there is an authority to control me, like police. Same thing, if I am doing some wrong construction, there is an authority to control. So it's a two-way. We cannot only blame government, we cannot only blame the people also. So if someone is wrong, I think we have the responsibility also. We cannot sort of leave our responsibility. Look, okay, I have not seen that. Eventually, if that people will lose their life and the property, the government need to compensate, right? And the where from that compensation will come from your and my money, the taxpayers' money, right? It's not the anybody. So it's like there is also debate, like why I'll pay for someone other's mistake. This is the mistake from someone. But it's a quite long debate. Like those who are paying tax, why I'll you spend my taxpayer money to some wrong decision. Okay, so yeah.
Anyway, those who are present out here, if you have any questions, you may please ask two questions our sir, Dr. Indrajit Paul sir, and yes, this is open to house.
Yes, Join Ghosh, you can have your question to Professor Paul.
Yes, sir. So I was asking that same question as Bishoti Roy, sir. That it is the problem of the government, right? Is the fault of the government that they are not stopping the people from building houses and all that. The authorities are not stopping the people from building houses. The authorities are not taking authority. The authorities are not taking authority of the problem, and they are just sitting idle. It may be the fault of the government that they are not allowing the authorities to take action or to do anything for the people, or to remove them from their place and giving them a particular different place to settle them. It is kind of government's fault, right? So that they are not allowing, they are not allowing them. Like sir, in Dharabai, in Mumbai, Dharabai, like it's a one of the biggest slums in the in world, I guess. So they are like, they could have moved the people from one place to another, and they could have made some development programs over there, and they could have made buildings, and then they could have given them rent, and there can be a government profit. There can be a government revenue, but they are not doing anything such things, and the government are to be fault, right, in this situation, because it's the government's fault. They are not taking actions. They are the persons who we are choosing, and they are the representative who should take actions.
Join, you have specific questions, or otherwise Hina will, I think, ask something.
Yeah, just a quick. I mean, I'll respond to that. Before joining AIT, I mean, I'm here in AIT for the last 7 years. Before that, I was actually spent 8 years in IS Academy. So I was used to train all the IS officers in India, in Missouri. So here actually we have seen a lot of decision issues. Like the problem is not that simple. I mean, it's not the fault directly we can blame to anyone like this, because it's not very straightforward problem. While first place, why that people, even they know like that is a If you go to like Kalimpong or Darjeeling area, people know like this is a very critical slope. If I construct some house that maybe may face some sort of landslide problem in the future. So despite that, why the people are constructing their house there? So there are some sort of underlying root causes. We call underlying root causes. Unless and until we sort of solve the root causes, like with paracetamol you can stop your temperature, but the root cause for that fever you need to understand, then that particular things to be addressed. Otherwise with paracetamol you cannot stop your fever, right? We need to really address the real problem of that fever, why that fever is happening, right? So that root cause need to be understand and to be solved. Otherwise, like if we stop plastic bag tomorrow, it will not stop because we have no alternative option yet. I mean, if we put some alternative on place for use and effective manner, then we can implement like, okay, there is no plastic from tomorrow. But suddenly if we stop no plastic without any alternative option, then I think implementation will be very difficult. So these are the real challenge actually. Okay, I mean we can go to the next question also.
Yes.
Thank you, sir.
Hina Khatun, if you can raise your question to Professor Paul, please. Hina Khatun is there?
Yeah. Hina Khatun, you raise your hand.
Hina Khatun, are you available and you have a very specific question to Professor Paul, then please ask. Or any other questions from any participants or any viewers, please you can have, if you have any question, then please you can raise your questions into this international web platform, and Professor Paul is with us. Okay. Thank you, Dr. Indrajit Paul.
Yeah, thanks.
For your
Dr. Tuhin Ghosh, Professor Tuhin Ghosh also.
Yes, sir. Yes, sir.
Thank you.
Yes,
Thank
sir.
you, Tuhinda, for connecting me.
Thank you for your good presentation, information, and Professor Paul highlighted the hardcore session of disaster, the unpredictable profile of the environment, research opportunities in disaster management all through the world. What is going on? What would be the prospects, future and prospects of disaster management studies, which is not only useful for the students, but in this web platform, international section, so many works opportunities are there, research opportunities are there, and Professor Paul has highlighted the hardcore section of that problem, address the problem, and trying to mitigate the problem in international level regarding the disaster management. Excess out of his busy schedule, Professor Paul was with us with his excellent presentation, and on behalf of organizing committee, I Acharya and Vishwajit Roy, Episura Government Organizing Section, West Bengal, India, again heartily congratulate and thanks Professor Indrajit Paul to have with us, and it is our expectation in the future program when we invite Professor Paul, probably and surely he will with us in this session and in this section in that way. Thank you, Professor Paul, for your nice presentation and being with us. Thank you, sir.
Thank you so much. Thank you so much. Really, really pleasure to interact. Thanks. Definitely I'll try for the next.
ইন্দ্রজিৎ দা, এরপরে দেখা হবে আবার।
নিশ্চয়ই দেখা হবে। নিশ্চয়ই
হ্যাঁ, আমরা যদি মানে কোনো অফলাইন সেমিনার করি অবশ্যই আপনাকে ডাকবো এবং আসবেন অবশ্যই।
হ্যাঁ, আমি নরমালি আসি, কিন্তু এখন মানে এই কোভিড এর জন্য প্রায় অলমোস্ট 1.5 years যেতে পারছি না দেশে।
আচ্ছা স্যার। বলছি স্যার আমাদের একটা ছোট্ট প্রোমো ছিল, প্রোমোটা আপনি দেখেছিলেন আমাদের কলেজ রিলেটেড?
হ্যাঁ 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 দেখেছিলাম।
স্যার কেমন হয়েছে স্যার একটু
না
যদি
ভালো
বলেন।
হয়েছে, আমি দেখেছি ওটা ড্রোন শট ছিল ভালো, মানে আপনারা করেছেন।
ওকে স্যার, ওকে স্যার, খুব
তবে
ভালো
একটু
লাগলো।
একটু যদি ইয়ে দেয়া যায় কিছু, মানে কি বলবো, কিছু লেখা সাথে আরো ভালো
আচ্ছা
হবে।
আচ্ছা স্যার, হ্যাঁ
একটু
মানে
ভালো
আমরা
ভালো হতো।
অ্যাকচুয়ালি খুব শর্ট পিরিয়ড অফ টাইমে ইন্টারন্যাশনাল
বুঝতে-
সেমিনারটা অর্গানাইজ করেছি।
হ্যাঁ
মানে
হ্যাঁ।
মানে আমরা সত্যিকারের কথা এত শর্ট পিরিয়ড টাইমে যে করতে পারবো এটাই আর কি, মানে মানে এখনো ভাবছি আর কি করতে পারছি বলে।
না না হয়ে গেল তো, আমি যে কালকে আপনার ইনঅগ্রালে ছিলাম কিন্তু আমার একটা মিটিং ছিল, আমি আর থাকতে পারিনি।
হ্যাঁ এক্স্যাক্টলি, আমি আপনাকে
আমি
বলার
ঢুকেছিলাম।
জন্য, হ্যাঁ আমি বলার জন্য আর কি বললাম যে, কিন্তু আপনি দেখছি তখন বেরিয়ে গেছেন স্যার।
হ্যাঁ না আমি তারপরে আর পারলাম না, অ্যাকচুয়ালি আমার একটা এখানে মিটিং ছিল একটু আর্জেন্ট, তো ওই জন্য আমি থাকতে পারিনি। আমি আধ ঘন্টার মধ্যে ছিলাম, ভিসি সাহেব যখন শুরু করলেন তারপরে খানিকক্ষণ শুনে আমি চলে গেছি।
ওকে স্যার, থ্যাংক ইউ ভেরি মাচ স্যার, দেখা হবে।
হ্যাঁ হ্যাঁ হ্যাঁ।
ভালো থাকবেন স্যার।
ওকে ওকে, থ্যাংক ইউ সো মাচ, থ্যাংক ইউ সবাইকে। হ্যাঁ।
Okay, we are now we are in the technical session for life sciences and allied subjects. All the participants, presenters, delegates, and viewers in this session, there will this is designed for 3A section, whose link has already been provided with you, and all the participants who are there and who are designed for their presentation in this section are requested to be ready with that part. We are taking only 5 minutes to join our chairperson, honorable chairperson, and till then, please bear with us. Please give us some only 5 minutes. We can connect our honorable chair, please. kindly request all the participants to kindly I kindly request all the participants to hold on as we are waiting for our chairperson to join the meeting, and we'll start our session as soon as the chairperson joins. Hello, am I audible?
Yes, yes, audible.
Okay.
So, when the next session will start, sir?
Professor Chandra, sir, are you there?
Sanjay is there. Sanjay is there.
Good afternoon, Sanjay da.
Yes, sir, you are audible.
Welcome, Sanjay.
Hello? Yes, sir, আপনার কথা শোনা যাচ্ছে। Yes, sir, you are audible. Hello? Yes, sir, you are audible. Hello? Yes, sir, you are audible. Participant, please bear with us. Only 2 or 3 minutes, we can start our session, technical session. Hello, can you hear me? Please. Hello? Sir, you are audible. Hello? 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 Yes, sir, you are audible. আপনি কি শুনতে পাচ্ছেন? স্যার শুনতে পাচ্ছেন?
Is there?
Participants are earnestly requested from the part of the organization. Hello. Yes, sir. Yes, sir, you are audible.
Hello.
Hello, Akash sir. Am I audible? I am Pulokesh.
Yes,
Am
sir.
I audible, Akash? Okay.
Yes, sir, you are audible.
Yes, Pulak sir. Yes, Pulokesh sir.
Okay.
Good afternoon, Swapnadi.
Okay.
Good afternoon.
Okay. Sanjay sir, can we start? Can we start? Sanjay sir, can we start?
Akash sir, is my screen is visible?
Yes, it is visible.
Yes, Shrikant. হ্যাঁ শ্রীকান্ত দেখা যাচ্ছে। হ্যাঁ দেখা যাচ্ছে।
Professor Sanjay Poddar, am I audible?
কি হয়েছে একটু কি দেখ? না স্ক্রিনে না
হ্যাঁ স্যার, শোনা যাচ্ছে আপনাকে?
হ্যাঁ হ্যাঁ শোনা যাচ্ছে।
হ্যালো,
হ্যাঁ দেখা
শোনা
যাচ্ছে।
যাচ্ছে?
হ্যাঁ কোনো প্রবলেম হয়েছিল, সরি সরি সরি। হ্যাঁ
Okay, sir.
হ্যাঁ
Can we
হ্যাঁ
start? Okay. This is the second day of an international interdisciplinary web seminar. In this technical session, this session is designed for the life science and allied subjects. And in this session, we have with us Dr. Sanjay Poddar, Assistant Professor, Department of Zoology, University of Burdwan. He was a former professor, assistant professor of different government college. His research area of interest is allergology and medical entomology, international,
Akash,
national.
Akash,
Yes, sir.
I am not assistant, associate professor.
Sir, yes, assistant professor. I already mentioned that.
Oh, okay, okay. I have some technical problem is there.
His teaching experience and regarding the research experience, it will take lots of time to explain that part. And it is our privilege to have Professor Sanjay Poddar, an eminent scientist, international scientist, with us as in the chair of this session. I, on behalf of organizing committee, seminar committee, APCI Government College, heartily welcome Professor Poddar to chair the session in this international web seminar.
Thank you, Akash. Swapnadi is there, I think. Okay, so
Yes, I'm there.
How are you, Swapnadi?
একদম okay, I'm okay. Nice to see you. Good.
Right,
Carry on.
after a long time. Okay, okay.
Right. After
Okay, so we have a running out time, so I think we have to complete it within 3 o'clock. Is it right, Akash?
Yes, sir.
Okay, so we
Yes,
have 11 presentations, I think.
Yeah, exactly.
So can we allot 8 minutes, 7 minutes for presentation and 1 minute for discussion?
With the permission of our honorable chair, I want to declare one thing. As we are going through the scarcity of time, so presentation should be restricted only 5 to 6 minutes and 1 to 2 minutes for a question answer session, sir.
Okay. So
can we start?
Can I start? Definitely.
Yes, the first presentation from Dr. Srikant Guria, Assistant Professor, Barashat Government College. His research topic, pulmonary risk of arsenic poisoning and insight of environmental pollution. I request Professor Guria to present his research.
Am I audible? My screen is visible.
Yes, sir.
Audible.
Your screen is visible. Now
Okay.
over to Professor Sanjay Poddar.
Srikanto, start.
Yes, good afternoon Sanjay da, অনেক দিন পরে।
Good afternoon. Okay.
So my topic is pulmonary risks of arsenic poisoning and insight of environmental pollution. Before going to discuss, actually it was very much short work when I was the part of the parasitology and medical entomology special paper at Darjeeling Government College. I did this work along with the Professor Swapna Chetri and also my mentor Dr. Madhusudan Das. So I just represent some of the result of the total work, that is the pulmonary risks of arsenic poisoning and insight of environmental pollution. It is known to all that exposure to arsenic through drinking water, it is very much related with the respiratory symptoms, mainly the obstructive lung disease. And arsenic is an environmental chemical that is related to human health also. We attempt to evaluate the nature of changes of the histological changes of lung tissue by arsenic. So what we have done, that we treated the groups of the white albino rat with the arsenate, sodium arsenate, in a proper way, and maintain the rat. And ultimately, we perform the histochemical studies with using hematoxylin histological analysis, and showed the difference between the normal lung architecture of the normal groups of rat and the arsenic, that is sodium arsenate treated rat. So what was the finding? The sodium arsenate treated rat showed thickening of the interalveolar septa. Some eosin-stained mononuclear cells found infiltrating in the alveolar septa, and also found hypercellularity. The hypercellularity means some cells increased in number that included pneumocyte and also fibroblast. Some alveolar macrophage and pneumocytes of both type, that is type 1 and 2, were degenerated, and they have also showing some surface bleeding, that is the indication of the initiation stage of the apoptosis. And it is known to all that pneumocyte type 1, they are responsible for gaseous exchange in the lung, and pneumocyte type 2, actually they are the cell, they are the progenitor cell from which the Clara cell will form, that will produce the surfactant that maintain the surface tension in the lung. And also it is found that the pneumocyte hyperplasia occurs. It is the number of the cells of the pneumocyte increase, and it is the indication of the early response of alveolar wall to injury. So the upper picture is the normal histology of lung, that is very much known to all, and the lower picture is that some eosin-stained cells come into the lumen of the alveolar sac and the thickened of the alveolar septa. The left hand side picture is the control lung pneumocyte, type 1 pneumocyte that are thin, and type 2 pneumocyte that are larger inside. But in case of the right hand side picture, arsenic treated lung pneumocyte, where the interalveolar septa, it is the space between two alveoli, become very much thickened due to hyperplasia. And it is the yellow color cells indication is the normal alveolar macrophage. It is also known as alveolar dust cell, where some foreign particle come and it will be engulfed by the macrophage. But in case of the arsenic treated cell, it is found that the macrophage are pycnotic and surface bleeding were noticed. So we have also published this paper, that is pulmonary involvement of arsenic poisoning and effect of arsenic toxicity on peritoneal macrophage and spleen cells in rat. Some also experiment we have done in my SARS lab. So what is the conclusion? The general conclusion is that extravascular localization of leukocyte that is present in the lumen of the alveolar sac that indicate the vascular injury. There are some previous paper that also indicate that the arsenic treated cell transformation that leads to lung cancer. Actually, what is happening when the arsenic enter into the body, it will produce the excess oxygen or superoxide, that is known as ROS or reactive oxygen species, that is responsible for angiogenesis and also the apoptosis. Therefore, arsenic poisoning in environment may be an important cause for pulmonary dysfunction in mammal. I want to acknowledgement our PG student also, and also the Swapna Chetri ma'am, and who did a number of histological slides, and also we have done some apoptotic studies, though I have not showed these studies in this short period of time. And thank you for the attention. I also like to acknowledge our chairperson and also the organizer who gave me the opportunity to give the short lecture. Thank you.
Thank you.
Thank you, Srikanto. Very nice presentation. This presentation is open for discussion. Any questions? I think no questions. So I have just one to ask Srikanto. What dose you use for this arsenic induced treatment? The dose.
The doses was that is the sublethal dose. Actually, 1 milligram of you can see my slide. Yes, 1 milligram of sodium arsenate that was dissolved in 1 ml of vertebrate saline, and 10 microliter was injected for each rat for 2 weeks. It means 14 days.
No, I mean to say, is it normal in the environment, or you would just apply the hypothetically the one?
No, actually, this sublethal dose created the dysfunction of the organ. that has been published in previous paper. So I have selected this.
Okay, okay. Thank you, thank you. Thank you. Nice presentation, Sikanto. I think I move on to next my presenter, who is Akash. And one more thing, just I want to say that those who are present their paper should switch on their mobile video.
Yes, can you see Sanjay?
Yeah, 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 yeah. Thank you, thank you.
Okay.
Okay, okay. You can leave. Sikanto. Okay. Now the next presenter, Akash. Who's the next one?
So I think my name is in the list, Utpal Sinara. I think I was again.
Okay, then you can present. Okay, okay.
Okay, so I'm presenting from my mobile phone. Actually, I'm in college, so
Okay.
maybe there are some technical glitches. I'm trying, so please cooperate
Okay,
with me.
okay, okay, okay.
Just for So can you see my presentation? Sam?
Yeah, yeah, yeah. No, no.
Yes, sir. Can you see?
Yeah, 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 yeah.
Okay, thank you, sir. Respected chairperson, sir, all listening guests, a very good afternoon to all of you. Brief introduction to the topic that I am going to say for a few minutes now. What are wetlands? I will be saying a few words about that. What are migratory birds and their role? A specific study, I was about to say about 5 wetlands, but due to shortage of time, I have parted my presentation and I am only focusing on only a single wetland for now. So what are the migratory birds that can be found in that very wetland? What are the inferences that can be drawn from the study, overall findings, and what are the that we can say we will see. Now, wetlands, as we know, are the transition zone between land and water. Now, with material functions, but as you may know that they are very much fragile in nature, and almost 50% of the global wetland has already got destroyed. And as you know, India has got almost 58.2 million hectares of wetlands, of which West Bengal participates with 12.48% of its geographic area designated as wetlands. Disturbances have converted many wetlands to nearby state to degraded heavy habitats and has very adversely affected the wetland flora and fauna. The water not any
I can't hear anything. Sorry, I can't hear anything. I think there is a technical problem.
Is it audible now, sir?
Yeah, 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 yeah. Yeah.
Audible, sir?
Audible.
Yes,
Carry on.
yes,
Carry on.
you are audible. Please continue.
So whatever survey actually carried out by West Bengal Forest Department in this very year on February reported 1.23 lakh of water belonging to 65. So we can say that water birds of West Bengal are diverse and they will actually colonize these wetlands during October. Wetland and water birds are because and particular criteria and the biology as well as understanding species biology for management and conservation of the wetland. Now, the I'm going to focus on is Satragachi wetland, which is located in the Howrah district of West Bengal. So, the methodology that were applied for the present study were line transect primarily, and for a robust study, hand frame and binocular frame methods were also used, and suitable literatures were used for identification of birds. So, as you can see, the wetland is near crowded Satragachi railway station, and there which in lake the signals can see the like common teal. These are also found. Also, some wetland dependent migratory birds in this lake. So mostly from this study was done between October 2004 to March 2014, and these are the specific density of the birds that has been recorded. These are the most prevalent birds which were recorded, but this list is not exhaustive. And you can see that in this green highlight, the lesser whistling duck was the most dominant bird, and these birds actually started colonizing this lake from October, but the number reached its peak in December, January, and gradually they departed in February and March, and subsequently their number decreased. So other birds like fulvous whistling duck, gargani mallard, pochard, and purple heron, they were present in scanty numbers. So what were the general observations? There were these birds numbers were just decreasing in during the total period of study. Like important birds, stuffed duck, perigenous poacher, they stopped visiting this lake. There was water hyacinth menace all over the lake. Adding to this was solid waste and liquid waste and noise pollution all around the lake. So the comparative study which has been shown here, there was significant decrease in bird number if we compare 2004-2007 study with 2010 and 2008 and 10. There was significant decrease. At the same time, there was significant increase in number of water hyacinth actually. So you can see in these pictures how solid waste and waste water was there. So the actually take like 10 included in national wetland conservation recognized in PC, NPA, other 13. Whole free zone, and there has been dogs been placed all over the lake. However, taking all together, the number of the bird was coming down, and it was as low as 800 in 2008. So some steps were to be taken, and some citizen-led cleanup drive came up. I must mention one of them. This was Nature Mates, who came up wholeheartedly and manually cleaned this wetland to make space for this wetland and for birds. And actually, from 2019, birds have started increasing in this lake, and 5,000 lesser whistling teams was recorded in 2020 and 21, and other migratory waterfowls like Norpil duck, cotton-pygmy goose, gadwall, pintail, and perigenous duck have started to return to this lake. So overall, we can say that lesser whistling duck dominated this lake. However, by no means is a one duck lake. Urbanization negatively influenced the lake. Wetland degradation was caused by alteration of pollutant runoff. Hello. Aquatic weed actually can wetland if not cleared regularly, and functioning of the was found satisfactory, and conditions to some extent can be reversed with proper management. So each one of us can play important roles, like we can use the age-old rule of reduce, reuse, and recycle. We can conserve nature by protecting, preserving, and we can collect nature by outreaching programs like some citizen science initiative like eBird and blog and social media using. So I would like to acknowledge these people, and thank you for your hearing.
Thank you, Dr. Roy. Just tell me, there are lots of works have already been done in Satragati regarding avifauna. So what's new in your work? Is it a original work or review work?
No, sir, it's totally original work actually. And what I tried to emphasize is the citizen science program. I have tried to incorporate citizen sciences program. Actually, the abstract that I submitted to this webinar,
I
included 5 wetlands.
What's new in your work? Because there's a lot of papers that already published in regarding avifauna diversity and others. They are decreasing regions and other things. So what's new in your?
Exactly. Yes, so I'm coming to that exactly. You are telling exactly the right. You have pointed it correctly. That has not been done from this wetland. Actually, the work that I was going to present was a comparative study from 5 different wetlands. I had selected Satragachi, Purulia, Shayabad, that Ombuja wetland from Durgapur wetland, and from northern parts there was Rosomoti and Narakalibil. So that was my plan to compare the study, but within this 8 minutes I was unable to present that. So I curtailed the study. Actually, what was new was to compare between these wetlands and how they stand from the standpoint of pollution and anthropogenic interventions. That was my study actually.
Okay. Thank you, Dr. Roy. I move on to next speaker, the Dr. Bhutia. Are you there, Dr. Bhutia?
Dr. Dawa Bhutia?
Yes, sir. Yes, sir.
Yeah.
Yes, sir.
You just presented your paper. Okay. 6 minutes time. Okay. Switch on your video.
Okay, sir.
Okay,
Am I audible, sir, clearly?
you are audible.
So is my slide visible, sir?
Visible, visible, visible.
Okay.
Just enlarge it.
Okay.
Enlarge it. Go
Okay,
to the slide presentation mode, I think.
So is it visible, sir?
Yes, yeah.
Okay. Very good afternoon to the respected chairperson and to the organizers from APC Roy Government College. Today, my topic for the seminar is traditional ethnozoological practices among the inhabitants of Darjeeling Hills of West Bengal, India. So first of all, I'd like to talk about the introduction. Okay, as we all know that plants and animals have played a major part in human evolution. So based on that, humans have utilized both plants and animals for their welfare. So ethnozoology basically deals with the study of the past and present interrelationships between human cultures and the animals in the environment. So it is one of the main subdisciplines of ethnobiology and shares many methodologies and theoretical framework with ethnobotany. And currently the WHO estimates around 80% of the world populations rely primarily in folk medicine derived in plant and animal origin for immediate cure against illness, of which 8% are of animal origin. So Darjeeling Hill, okay, basically I'm talking about Darjeeling Hill, okay, it's a region in the Himalaya hotspots which is rich in plant diversity and as well as animal diversity. But plant diversity its richness and its usefulness as a medicinal plant has been properly documented, whereas the documentation for ethnozoological practices among the hilly regions and the people living in the hilly regions are still compromised and they are not still compiled till date. Hello?
Continue.
Okay. So the main objective of my of this study is to record the ethnozoological knowledge of different communities of the inhabitants of Darjeeling Hills before more faunal resources and the populations who are repositories of such knowledge disappears. So for that we selected a study area. Okay, the area was called a small place called Pokhrebung, which is located 29 kilometers away from the main town of Darjeeling in West Bengal of India. So Pokhrebung Valley usually contains 8 tea states and many sub-villages. And the prominent ethnic tribes that have been living in this small village are Bhutia, Lepcha and Tamang, along with Rai and Chetri as non-tribal population. So this is the study area, Pokhrebung. So methodology used was that the basic thing was that the questionnaire survey was conducted and the people selected in this survey were generally over 40 years of age. A total of 89 households was selected, and among them the key informants were also interviewed, like experienced village people, the practitioners of herbal plants and animals, faith healers like Zachary, and Buddhist monks like Lama. Along with that, the occupation of the people living in that area was also noted. And along with that, again, the younger generation was also questioned about the knowledge of ethnozoology. So these are the results. In the results, we found that 40% of the people in that village or small town around Pokhrebung depended mainly on agriculture. 22% were wage workers, 21% were following this service sector, 11% were students, and 6% carried out small local businesses. And the people also, we also found that the people practiced animal husbandry. So in animal husbandry, 68% of the population, they carried out cattle farming, 55% is carried out poultry, 11% goat farming, and 2% piggery. So coming to the main point, we found that the 19 different animal species were used in 25 different categories of medicinal or other religious purposes, with mammals representing about 58%, apes 32%, amphibia 5%, and fish 5%. Of the 19 animals which we found out, 11 were mammals, 6 was apes, 1 was amphibia, and 1 was fish. So 18 animal product or parts were used in zootherapy. Animal products like flesh, blood, urine, milk, etc. were commonly used as ethnozoological practices, along with indigenous herbs and plant products. So these are the tables which represents the animal species and the parts used along with the medicinal use. So coming to cattle, Flesh and tail, urine, milk, they all were used for the medicinal purpose, but mainly flesh and tail was used for tuberculosis. Coming to monkey's blood was used for tuberculosis. Donkey urine was used mainly for alcoholism, as well as to treat typhoid. And sheep, mainly the ghee was used as vulnerable for healing wounds. Coming to porcupine, the flesh and spine and were used, but particularly flesh was used to treat asthma. Coming to fox was used to treat, the flesh of fox was used to treat gout and arthritis, as well as to treat chickenpox. And humans, mainly urine and milk were used, but they were used mainly for treating eye infection as well as other vulnerability for wounds. So coming to goat, milk and urine were used to treat eye infection, measles, even tuberculosis, as well as the meat or the flesh of goat was used as nutraceutical. Hair, the brain and flesh was used to treat asthma. For pig, okay, the nose was used for treating nose bleeding. Himalayan black bear was used to treat for malaria as well as gout and arthritis, as well as some sprains in the body. Coming to the birds, okay, Indian people, the feathers were used for treating nose bleeding. Domestic fowl, they were used mainly for nutraceutical value as well as vulnerability value. Mainly flesh, eggs, and fats were used. Blue whistling thrust, okay, the flesh of blue whistling thrust was used for treating piles, dysentery, pneumonia. House sparrow, the flesh of house sparrow was used for aphrodisiac as well as for nutraceutical value. Pigeon, the flesh was used for treating asthma. And Khalid's pigeon was also used for treating the disease as a nutraceutical value. So coming to amphibians, okay, Levis frogs was there. The whole body part of Levis frogs was used to treat infertility, fever, measles, diarrhea, piles, and model, finally the fish. Okay, whole body and the blood was used to treat asthma as well as anemia. So coming to discussion, we found in the survey, we found that 70 to 80% of the inhabitants were directly dependent on agriculture and animal husbandry for their livelihood. The ethnomedicine was intensively used to treat ailments for infections, diseases like tuberculosis, conjunctivitis to wounds, arthritis, piles, etc. This is very important. The bond nose powder of pig to treat chronic nose bleeding possibly has not been previously reported in India, even from other tribal groups. Coming to blue whistling thrust, the use of blue whistling thrust as a traditional medicine has not been reported by other tribes of India. Fat of a number of animals of class Fs and mammals has been found to be mainly used as vulnerability and emollient, along with the other animals which were used. Okay, that was, some of them were common with the other tribal groups, but this blue whistling thrust, they were not common with the other tribal groups which we compared. The study also found that the use of domestic fowl, pig, and goat in magical religious form of worshipping family deity like Kul Puja and worshipping forest god Ban Devta Puja by local faith healers. But lastly, I'd like to say is that the younger generations interviewed in this study clearly showed the lack of knowledge regarding the ethnological practices. Thank you.
So, Dr. Bhutia, you work on a nice topic, but tell me how many people have surveyed? This is basically a survey report, I think.
Yes, sir. Yes, sir. So we surveyed around 89 households, sir.
89 households. Do we have
Yes,
any
sir.
correlated, or do you rely on their what they said?
Pardon, sir?
Do you rely what they said when they are using this type of animals for their defined healing purpose of their different diseases?
Yes, sir. Yes, sir. So
Is
they
there
basically
any
we found
literature
that,
regarding this?
no, sir. That's why I have mentioned in my previous slide, sir, that though the knowledge of these plant-based herbal medicines are well documented, but the ethnological practices, okay, their knowledge has not been well documented, sir. And the written literature is very few, sir. It's scanty. It's scanty, sir.
You can work on this aspect.
Yes, sir. So we have published the paper also, sir, regarding this paper, sir.
Okay. Thank you. Thank you.
Thank you,
So
sir.
we move on to the next speaker, Dr. Shukanto Mojumdar.
Yes, am I audible?
Yes, you are audible.
Okay, a very good afternoon to the respected chairperson, the organizers, and all the participants. Myself, Dr. Shukanto Mojumdar from Siliguri College, and I'd like to present the paper. Is it visible?
Yes, visible. Just go to the slide.
Yeah, I'm trying to. Okay. that is in vitro control of fish pathogenic bacteria Pseudomonas aeruginosa PKB113 strain by the extracts of pan and lobongo and collected from West Bengal. So before going to the main study, I would like to express the objectives of the study is to study the alternative way to retard the pathogenic growth by using these plant biomolecules, and also we have to assess the antibacterial activity of the plant biomolecules along with the determination of the effective dose, and also we are trying to commercialize this product and that's why it will be administered in the field level. So bacteria are one of the major factors causing very much damage to the fish producing country like India. India is the second highest fish producing country, and today some countries are also restricted to prevent this type of pathogenic bacterial growth by means of chemicals and drugs and antibiotics. So this study is basically for the purpose to find out some alternative to this synthetic and also harmful chemicals. And we are using the natural compounds like the secondary metabolites which is coming from the extract of the plant molecules, and in this present study we are try to find out the activity of the pepper beetle, that is the pan, and Syzygium aromaticum, that is the lobongo, against the Pseudomonas aeruginosa PKB113 strain, which was collected and isolated from diseased fish in West Bengal. And there are some materials and methods by which we are collecting and also identify the plant materials from the Department of Botany, University of Kolani, and also we make the extract by drying up the pan leaf and also the fruits of the lobongo, and after that we with the help of rotary evaporator we made the crude extract of these materials, and then we go for the antibacterial activity test by means of this diffusion method. And in that case we use the Whatman filter paper of 6 millimeter in diameter. At first we have to inoculate the broth with the help of 10 to the power 6 colony forming unit per ml of, that means the 0.5 McFadden suspension of this bacteria, and after that we have to spread culture this one. Along with this we are placed this extract soaked disc in particular area of the petri plates, and after that we wait for more than 24 hours or near about 24 hours. And another study is the bacterial growth curve analysis. In that case we are using the nutrient broth, and we studied for near about 24 hours and by means of just taking after a regular 2 hours interval the spectrophotometric data, and after that we are analyzing with the help of simple statistical analysis by arithmetic means and also the standard error. So what are the observations is that the crude extracts from the plant and they have some antibacterial activities against the Pseudomonas aeruginosa, which is in case a multi-drug resistant bacteria, and we are just assessing the inhibitory zone, and we found in that case that also we are taking the positive control like tetracycline as 100 microgram per ml, and negative control with just methanol, and methanol is showing no inhibition, and tetracycline is showing highest inhibition in when we are measuring in diameter. It is almost about 13 millimeter, and our extract that means the extract from pepper beetle and lobongo, these have got some antibacterial activities, and there are a remarkable that means the zone of exclusion is there. I am just showing this one. In 100 mg per ml concentration of pan, it is almost about 8 millimeter of zone of inhibition, and in 50 mg per ml it is showing 3, millimeter in zone of inhibition. Whereas in case of Lobongo, that is Isidum aromaticum, it is showing almost about 15 millimeter in 100 mg per ml concentration and 10 millimeter in 50 mg per ml concentration, and we are restricting this one in 50 mg and 100 mg concentration. In between also we are taking 80 mg and 60 mg, but in which we find out that the minimal inhibitory concentration is almost about 50 mg per ml, which is showing a clear zone of inhibition. So these are the Petri plates, and some methanolic extract is there without any zone of inhibition, and Caesium aromaticum, that means the extract from Lobongo, tetracycline for the positive control, and pepper beetle as the another extract. So this, and also we are taking this extract in the methanolic in methanol, and that's why these are the part which is showing the zone of inhibition. So in discussion, this is the bacterial growth curve analysis, which is showing that the normal growth curve of bacteria in nutrient broth, which is almost calculating in for 24 hours interval, and this is the pink color bacterial growth curve, which is for the pepper beetle, that means the pan extract, when we are administrating the pan extract in this nutrient broth containing bacteria, and also this yellow colored is the control by means of Lobongo extract. And in conclusion, we can say that in the present study, the antibacterial effect is very prominent in case of pepper beetle and Lobongo also, and also it has been shown that the Lobongo extract, the fruit from the Lobongo extract, is much more effective than that of the pepper beetle. And so it can be also used in field level, but it has not yet been done. What is the active molecules and what are the effective dose? It is still yet to be done in for future. So I would like to I'm grateful to late Professor Praveen Kumar Bandyopadhyay for his continuous support when during his supervision, and also thankful to DST for their financial support during the project. So these are some references, and thank you.
Okay, Dr. Majumdar. So have you you have already told that you have identify or isolate some bioactive compounds from PAN and E. So how do you conclude? How do you conclude that there is a different types of bioactive molecules are there in PAN and Lobongo? So until unless you can identify or isolate the things, I don't think so. You can say that this is helpful for control the this type of microorganisms.
No, actually, what is this work is pending, the isolation and characterization of the active biomolecules, and in most of the cases in the material review, we have found out that the eugenol and some triterpenoids, these are coming from the Lobongo extract, but in case of PAN,
different types of terpenoids and flavonoids are there. So until unless you are isolated the bioactive molecules, you cannot say definitely that this will act as an control agent of this.
Yeah, definitely.
Okay, however, nice presentation. Okay.
Okay, thank you, sir.
Okay. Next present speaker is Pulokesh Paroi, I think.
Assistant Professor, Hooghly Motion College. His title of the presentation is Career Systematic Studies.
Hello. Am I audible?
Yes, yes.
Am I audible?
Yes, you are audible.
Yes, yes. Okay. Okay. Very good afternoon to Chairperson Sir. My colleagues of previous colleagues of this college, actually very recently I have been transferred from this college. Only 20 days I have been joined in new college, UGC-HUMAN college. So very good afternoon to my colleagues. other participants and also can see my students. So very good afternoon to all. So myself is Pulokesh Paroi, Assistant Professor in WBS. Today's topic of my seminar is the Karyosystematic Studies.
Dr. Paroi, can we enlarge your presentation?
Okay, sorry. Okay, sorry. Now, yes.
It's okay. You can continue.
Okay. Karyosystematic studies on Gasteria obtusifolia, Gasteria variegata, and Gasteria glomerata. So, why, and the question comes, why actually I have been chosen this actually Gasteria. So, Gasteria actually is a very important genus, similar to aloe, but it is difficult to identify, and our general actually people, we always mixing up with the aloe. So this Gasteria actually a important genus within the family Hawarthiaceae, and this is a succulent and is cultivated nowadays in India, in the various parts of India for its great horticultural values, and some of cases it is actually medicinal value as well. And it is also rare succulent in India, so need to preserve conserve for the biodiversity purpose. And I have my objective is to taxonomic identification of these actually genus we have been chosen, mainly based on the cytological different parameters. So cytomorphological analysis software, which is more advanced, karyotyping type. So identification actually on the image analysis software, and finally the relationship among these species have been tested out on the basis of different parameters of chromosome and different types of cytomorphological parameters. So materials I have chosen Gasteria obtusifolia, Gasteria variegata, Gasteria glomerata. Methods actually followed by standardized method of Professor, my teacher, late Professor A.K. Sharma, the Padma Shri teacher. So now, regarding these the plants, these are the very plant Gasteria obtusifolia, which is labeled as A. Level B, that is the Gasteria variegata, and level C, the Gasteria glomerata, and level D here the inflorescence. The characteristic inflorescence actually the plant is morphologically identified by its tongue-shaped leaf, and it is known as the gaster lily or stomach lily of the desert, mainly as it originated in South Africa. So gaster, so mainly flowers are very bright in color, orange or red in shape, and stomach shape, stomach in shape. This is the Gasteria obtusifolia, this is the variegata, and this is the glomerata. Now, the karyomorphological studies have been based. So first I have been actually done the somatic chromosome study as well as meiotic chromosome study. The left one is the somatic diploid chromosome, and the right side is the pollen mitotic, which is haploid in number. This is the chromosome number of Gasteria variegata. The previous was the actually Gasteria obtusifolia. This is Gasteria variegata, and this is the Gasteria glomerata. So this is all in a single frame. And now, though most of the actually chromosome have usual normal in character, but during the meiotic study, only 2 to 3% meiotic abnormality has been noticed, which is very related to the speciation or adaptation on the survival of the species. Now this is actually the karyogram of these species, Gasteria variegata, Gasteria glomerata, and Gasteria obtusifolia. And this is a table where different karyomorphological parameters have been shown. SCN, that is somatic chromosome number, in Gasteria obtusifolia 2n is equal to 14, Gasteria variegata and Gasteria glomerata it is 2n is equal to 28, and other karyotypic formula has been mentioned here, shown here. But the very important to trace out the relationship and the cytotaxonomical parameter, the TCL, total chromatin length of the haploid complement, is very essential. So, TCL, total chromatin length in Gasteria obtusifolia, though it is deployed in nature, but it has been shown here that it is the value is 2015.14 mu, whereas in Gasteria variegata it is 2561.15 mu, and Gasteria glomerata it is 2337.08, and the respective actually coefficient of variation, statistical variation, coefficient of variation have been made. So according to the basis of actually study, another actually table is formed from where we can conclude that different stages in chromosome morphology have been noticed clearly, and total chromatin length shown here, the values prove that the Gasteria glomerata is most advanced in nature as compared to the Gasteria variegata, and then after Gasteria obtusifolia. So why it is actually obtusifolia is most primitive. So by image analysis software, it proves that though Gasteria obtusifolia contains the diploid chromatin number, but the total chromatin is less higher. So higher value of the total chromatin actually proves or indicates the primitiveness of the species, while the shorter length of chromosome indicates the most advanced species. So another actually way out to find out the speciation of this actually species. Now these are the different references whenever I am going to study, and with time limit. Thank you all. Okay, this is actually very deep actually discussion about my finding.
Thank you, Pulogar.
Thank you, Dr. Faroi. The presentation is open for discussion. Is there any questions? I think
Yes,
no questions.
thank you.
Thank you for your nice presentation. So we can move on to the next presenter, Sushma Chatterjee. Thank you.
Sushma Chatterjee.
Okay.
Fellow, Department of Lab Medicine, Calcutta School of Tropical Medicine. Her research, her topic of presentation is environmental pollutants and macrophages.
Am I audible?
Yes, you are audible.
Just a minute. Take some time. Is it my slide visible?
Oh, no, ma'am.
Maybe it's some technical issue or something else. Sir, still not?
No.
Okay.
I think she should join later. So next speaker, Dr. Nandini Bhattacharjee. Okay. So Sushma, you can join later. Okay.
Sir, I think right now my screen is presenting because maybe it's a technical
problem.
Oh,
yeah.
Okay, sir. Okay, sir. Is it visible right now, sir?
No, right. Nothing. Only your name.
Nothing, ma'am.
Only your name is visible.
Okay, sir. I'm joining again, sir. Okay, sir. Thank you, sir.
Okay. So I move on to next speaker, Dr. Nandini Bhattacharjee.
Thank you, sir. Let me share my screen.
Yes.
Sushma, Could you please?
Yeah. Sure.
Okay, thank you. Is my screen visible, sir?
Yes.
Yes, then.
Thank you, sir. Respected chairperson, sir, dignitaries and esteemed faculty members, I am privileged to deliver our paper before you. The name of our paper is Pivotal Role Played by the Species of Chironomus in Aquatic Food Chain along with the Determinations of Ecosystem Health of Aquatic Bodies. I want to say a few words about chironomids. Chironomids are two-winged flies of the order Diptera and family Chironomidae. They are commonly known as non-biting midges and they are quite similar to mosquitoes but do not bite. They are considered as biotic indicators for detections of the environmental degradations. And most important point is that because of the larvae of the chironomids develop polygene chromosomes in many of their somatic cells. Including those of their salivary glands, the chironomid larvae also attract scientists for exploring riddles of unsolved mysteries of genetics. Polygene chromosomes of dipterans may also be taken as cytological manifestations. The life histories of chironomids consist of the 4 stages: egg, larva, pupa, and adult. So it is a holometabolous insect, and the life cycle from egg to adult ranges from less than a week to more than a year, depending on the species of time of the year. I skip this slide because of time constraint. This slide shows some pictures of chironomids in nature. Now I'm coming to about the aquatic larval lives of this species. Actually, we worked with Chironomus stratipenis. Normally, the aquatic larva life to be the longest durations. It's about 18 days. It actually varies from species to species. In this larva life period, they cover 4 instars. And the 4th instar stage appears to be best for our study of their polygene chromosomes, when the polygene chromosomes are taken to be cytological manifestations. Now, what are the importance of chironomids in the food chain or food webs? The family Chironomidae has an important role in aquatic food webs because it represents a link between the producers. They are playing an important role in the detritus processing of the food chain. Moreover, due to the long life cycle and low morbidity, chironomids integrate various biological processes and may be used as indicators of environmental conditions. And chironomids are a major component of the diet of the many fish species, such as catfishes, carp. Now, what are the significance of the polygene chromosome study from chironomids? Chironomids larvae develop salivary gland polygene chromosome, which make them prospectives for cytogenetic monitoring. And polygene chromosomes are a system in which differential gene activities and its control can be analyzed directly at the level of gene themselves. And under the influence of the pollutant in the habitats where they live, these chromosomes respond exhibiting morphological changes. Right. Now I want to mention a few points about methodologies. We collected samples from the Holdia, Kolaghat, Dhapa, Kolkata, Dankuni and other regions which appear to be highly polluted. And for the preparations of the polygene chromosome from the salivary glands, the penultimate 4th instar larvae were sacrificed to obtain their salivary glands. And we collected egg, mass, larvae, adult flies from these selected zones of our studies in the laboratory. After fixations with 1:3% acetomethanol, we stain this preparations with the 2% acetorsine and then squash to obtain the polygene chromosome. over a glass slides. Beside this, fluorescent staining, we all, as well as DIMSA stainings, were also carried out in our laboratory. And the larvae of the Chironoma species reared in the laboratory were treated with heavy metals such as lead, arsenic, copper, and as well as cadmium in the laboratory environment for the purpose of our induction test. These are the laboratory setup for the rearing of the Chironomies. I want to mention in this regard that this slide shows the 4 polygene chromosomes of the salivary glands of the Chironomid stratipenis. And we characterize the morphological deformities of the larvae, Chironomid stratipenis, collected from the polluted water bodies. These are few pictures taken from our laboratory rearing conditions of Chironomies. These are the 4th install larva of Chironomid stratipenis. This figure shows some main structures of the mentum and the structure of the pectin epipharyngesis of the 4th install larva. Slide 13. Skip this slide. 4 polygene chromosomes are Chironomid stratipenis with normal configurations. They are marked as 1, 2, 3, and 4. Now we showed some pictures. I showed some pictures of these aberrations of the chromosome 1, chromosome 2, which we find in our laboratory conditions. Aberrations 3 chromosomes, aberration 4 in the aberration in the 4th chromosome. Now coming to the point that we also measure the heavy metal concentrations at different collection sites, such as arsenic, cadmium, lead, copper as well. And these are our experimental results, but I cannot explain the total data because of the time constraint. I skip these slides. Now our findings, we observe that mostly polymorphic organizations involve inversions as their cause of origin, and deficiency causes formations of blebs along the chromosome. Among all the chromosomes, the 4th chromosome is mostly affected, as we found in our laboratory experiment. Frequency of the polymorphic structures appear to be more in the regions of higher pollution parameters, and heavy metal exhibits differential effects so far polymorphism is concerned. And now I must say few lines about this. All the areas with high metal concentrations show the presence of the species Chironomas in the habitats, and excessive heavy metal content could not prevent their development in the aquatic bodies. The larvae of the flies also accumulated heavy metals from the environment. This suggests that the heavy metal being toxic to them affected their normal physiologies, which was reflected to their polygene chromosome reorganizations. This is the most important points of our findings. Such an organization of chromatin from the polygene chromosome advocates on the peculiar chromatin organizations in the polygene chromosomes of the flies. Now, I'm coming to the conclusions of our paper. Pollutions in the aquatic habitats affect the chromosomal structures of the chironomid. As we find in the larval conditions, 4 sensor larvae of Chironomid stratipensis, as well as their external morphology, such alterations in the chromosome structure may give some adaptive advantages for their survival in nature. And chromosomes have some weak points to be affected by the external agents like pollutant. And this species, Chironoma stratipenis, may be used as a model to measure the heavy metal pollutions in an aquatic habitat, that is to assess the ecosystem health of the aquatic body while acting as a potent bioindicator. Thank you so much for your patience hearing. I would like to thank the chairperson, sir, and all the faculty members and members of the organizing committee of the Acharya Prafulla Chandra Roy Government College, Siliguri. And I would like to thank my corresponding author, Dr. Porantop Sarkar, faculty members of our Department of Zoology, Rishi Bankim Chandra College. Thank you, sir.
Thank you. Very nice presentation, Dr. Bhattacharya.
Thank you.
Okay, I have just one or two questions. How do you identify this Chironoma stratipensis in others? Do you identify this in Chironomas?
Yes, sir. Identifications have been done, but actually it have been done by my corresponding author, Dr.
Okay.
Purnab Sarkar, who collected the samples and reared in our laboratory conditions. And
So you
then
are working on the diversity. You have also checked the diversity of carcinomas also.
yes, sir. Yes, sir.
Okay, okay. Do
Yes.
you do these carcinomas have also human health effect on human health?
No, sir. I have little
Yeah,
knowledge.
you can check it now. It causes allergy in humans, occupational allergy.
Thank you, sir.
Okay, thank you, thank you. You may leave. May I call Mintu Diashi, Assistant Professor, APJ
Yes,
Abdul
sir.
Kalam College. Okay.
Yes, sir. Yes, sir.
Present your topic. Yeah.
Yes,
So,
sir.
Mintu, you can leave now. You can join later. No problem.
Am I audible, sir?
Okay. Yeah, yeah, Rahul, you are audible.
Okay. Is my presentation is visible, sir?
Yeah, yeah.
Thank you. Thank you.
Yes, sir. Visible, sir.
First of all, let me thank you all for giving me the opportunity. I am Rahul Devnath, Senior Research Scholar at Chemical Ecology Laboratory, Department of Zoology, the University of Burdwan. Today, I am here to deliver a presentation entitled Comparative Performance and Digestive Physiology of Diaphania indica, Lepidoptera cambridii on Trichosan sanguina Q5BTC cultivars. Now, materials and method. Horse plant. Seeds of the three Trichosan sanguina named Baripulong, Polo number 1 and MNS-1 were separately cultivated at the crop research farm at University of Burdwan, West Bengal, India. Now, insect mass culture. 15-star larvae of Diaphania indica were collected from the each cultivar of Trichosan sanguina plant growing at the crop research farm of the University of Burdwan. Now, developmental time, ovulation period, fecundity, and longevity. For each cultivar, newly emerged male and female was paired in a fine mesh nylon net cage containing 10% honey solution for adults feedings, and eggs laid by the females on second day were collected in order to obtain the same aged eggs. 100 eggs were collected for construct life table study. Each egg used as a replicate on each other. Here, APOP stands for adult pre-ovulation period, the time period between the emergence of adult female and her first ovulation. TPOP stands for the total pre-ovulation period, the time period interval from the birth to the beginning of the ovulation. Now, life table analysis. Raw data on the survival, development, and ovulation of all individuals are analyzed based on the S-26 life table theory using the computer program 26MS chart, CHI 2027. Here, SXJ denotes the age-stage specific survival, LXJ denotes the probability of the newly laid eggs surviving to the age X, and FXJ denotes the age-stage specific fecundity, the daily number of eggs laid by the individual at age X. This is the software which I have been used for the data calculation. Food utilization indices. Food utilization indices like GR growth rate, CR consumption rate, RGR relative growth rate were calculated based on the formulas of Walball to assess the feeding efficiency of Diaphania indica. Now, biochemical analysis of leaves. 1 gram of the fresh leaf of respective types were used for the various biochemical analysis such as total carbohydrate, total protein, total lipid, total amino acid, total phenols, and total flavonoids. Also, dried leaves used for the determination of the total tannins and nitrogen. Now comes to the result section. Table 1 represents the developmental time and error longevity of the Diaphania indica on 3 Trichosan sanguina cultivar, where periodal duration highest on the Polo number 1 and lowest on the MNSR1. On the other hand, adult longevity such as females and male insects are highest on MNSR1 and lowest on Polo number 1. Table 2 represents the morphological features of Diaphania indica reared on the Trichosan sanguina cultivar under laboratory conditions. The length and breadth of the adult insect and as well as the larvae highest on the MNSR1 and lowest on the Polo number 1. Table 2 represent APOP, TPOP, ovulation period, and fecundity, where Fecundity was highest on the MNSR1 and lowest on the polo number 1. Table 4 represented the main parameters of the life table parameters like GRR, gross reproductive rate, intrinsic rate of increase Rm, finite rate of increase lambda, net reproductive rate R0, and the mean generation time T. Of diaphragm indica, we are on the 3 Trichosanthis sanguinea cultivar. All of these parameters are showed that highest in MNSR1 and lowest on polo number 1. Table 5 represents the measurement of the food utilization efficiency of diaphragm indica reared on 3 Trichosanthis sanguinea cultivar, where growth rate is highest on the MNSR1 and lowest on polo number 1. Table 6 represents the biochemical analysis of the leaves of 3 Trichosanthis sanguinea, where nutrient properties like carbohydrate, protein, lipid, amino acid, and nitrogen are highest on MNSR1 and lowest on polo number 1. On the other hand, anti-nutrient properties like phenol, flavonol, and tannins are highest on the polo number 1 and lowest on the MNSR1. Now, table 7 represents correlation coefficient of life table parameters of diaphragm indica reared on the 3 Trichosanthis sanguinea cultivar, where immature stages are negatively correlated with the nutrient properties and positively correlated with the anti-nutrient properties, and fecundity positively correlated with the nutrient properties and negatively correlated with the anti-nutrient properties. Also, proteolytic and amylolytic activity highest positively correlated with the nutrient properties and negatively correlated with the anti-nutrient properties. Now, take a look at the enzymatic activity of the fifth instar larvae of defined cultivar Trichosanthis sanguinea. Non-specific amylolytic activity was highest on MNSR1 and lowest on polo number 1. Also, proteolytic activity was highest on MNSR1 and lowest on polo number 1. Now, figure 3 is the A-stage specific survival value or SXG of diaphragm indica feed with the 3 cultivar of Trichosanthis sanguinea. Figure 4, LX, FXJ, MX, and LXMX of diaphragm indica feed with the 3 cultivar of the Trichosanthis sanguinea. Figure 5 depicted that the life expectancy or EXG of diaphragm indica feed with the 3 cultivars of the Trichosanthis sanguinea. Figure 6 depicted that the VXJ of diaphragm indica feed with the 3 cultivar of Trichosanthis sanguinea also. Now, figure 7 represents the population projection of diaphragm indica feed with the 3 cultivar sanguinea cultivar. Now, so that concludes the food quality in the respect of the Trichosanthis sanguinea cultivar influence the diaphragm indica development and fitness. This study suggested that the polo number 1 cultivar was the least suitable for the diaphragm indica. Largest developmental time, lowest fecundity resulted lower intrinsic rate of increase. That indicating lower population growth of the diaphragm indica could lead to the lower subsequent infestation. Thus, use of partially resistant cultivar polo number 1 could enhance biological and this is the
slide is not shifting.
Is this shifting, ma'am?
Slide not shifting.
Yes, shifting. Continue.
A few selected references which I have been used in my presentation. This is the acknowledgement.
Now it's okay.
I am highly thankful to my PhD supervisor, Professor Anandumai Bari, for entire conceptualization as well as the immense support to continue my work. I wish to thank the University Grant Commission, UGC, for providing me the fellowship. Thank you for your kind patience.
Thank you, Rahul. Rahul, one
reference দেয়া আছে তো আমি ফেনলস আর ফ্ল্যাভোনয়েডস হচ্ছে 1 গ্রাম লিফ থেকে প্রথমে এক্সট্রাকশন করেছি, অ্যালকোহলিক এক্সট্রাকশন করেছি, অ্যালকোহলিক এক্সট্রাকশন করে তারপরে তার এস্টিমেশন করেছি, সেটা আমি 1 গ্রাম লিফ হিসেবে
okay, Rahul. Okay. You may leave. Okay, thank you. Nice presentation. Okay,
thank you, sir.
I move on to next speaker. That is Paroma Mitro, research scholar.
Yes, sir.
Yeah.
Present, sir.
Present your topic.
Yes, sir. I'm presenting.
Institution Ecology Research Laboratory, the University of Bardhaman. Our topic is extraction of feed and plant volatile. And showing visible and you may proceed, please.
Am I audible?
Yes. Paroma, you are audible.
Okay. Good afternoon, everyone. I'm Paroma Mitro, a PhD research scholar currently working in the Department of Zoology, the University of Bardhaman. Today I am going to deliver my presentation on the topic Identification of Lathyrus sativus plant volatiles causing behavioral preference of Apis crachivora. At first, let me give a brief introduction about the plant, which is Lathyrus sativus, belongs to family Fabaceae, an annual crop which is acclimatized to diverse harsh environments, and it is resilient to climate changes due to its hardy penetrating root system. Also, the plant is economically important as food, fodder, forage, ornamental crops, and even in traditional medicine, and it is an efficient nitrogen fixer. The major insect pest, Apis crachivora, is a polyphagous plant pest. The aphid serves as a medium for growth of sooty mold, which reduces photosynthesis. Further, the adults and nymphs damages the plant by sucking cell sap, which results in reduction of seed yield. So while conducting the experiments, 7-week-old plants with 25 mature leaves were used for collection of volatile extracts from undamaged, denoted as UD, two types of insect damaged plants, which is denoted as ID50 and ID100, and mechanically damaged plants, that is MD. The protocol followed during volatile collection is depicted in Figure 1, where the charcoal filtered air was pushed at a rate of 2 liters per minute into top of the closed chamber, and the air was pulled at a rate of 1 liter per minute through 4 volatile collector traps consisting of 100 mg of HSEP-Q as an adsorbent. Further, the adsorbed volatile extracts were eluted with methylene chloride. The volatile extracts were fractioned in 3 parts for the use in olfactory bioassays and chemical analysis in GCMS and GCFID respectively. Now the olfactometer bioassays of volatiles were conducted using a Y-shaped glass tube olfactometer with 10 cm stem and arm length and 0.5 cm radius with a 45-degree Y angle shown in Figure 2. Now the dual choice bioassays with adult viviparous Apis crachivora females were conducted towards plant volatile extracts in the following mentioned combinations. Further, the following statistical analyses were conducted where the data were normally distributed as determined by the KS test and the data were homogeneous as determined by the Levine's test for homogeneity of variance. Further, the discriminant function analysis was applied. One-way ANOVA was conducted and the data were subjected to a post hoc Tucky test, and the olfactometer bioassays were analyzed by a chi-square test. Now the results of the olfactometer bioassays and the results of chemical analysis, which is shown in Table 1, where the number of compounds detected in volatile extracts of the following treatment plants are shown as follows. 20 and 19 compounds in undamaged bio and nirmal cultivars respectively. 21 and 20 compounds were identified in ID50 and ID50 bio and ID50 nirmal varieties respectively. Further, 18 and 22 compounds were identified in ID100 bio and ID100 nirmal, and lastly 20 and 23 compounds in mechanically damaged bio and mechanically damaged nirmal cultivars respectively, where benzyl alcohol was predominant, which is followed by p-cymene and 1,3-diethylbenzene. The total amounts of volatile organic compounds were significantly higher in the treatment with 100 aphids per plant, which is followed by in the treatment with 50 aphids and then the undamaged plants for each cultivar. Now the results of the olfactometer bioassays showing that the female showed significant preferences towards the volatile from insect damaged plants of each cultivar when tested against the control solvent. In case of individual compounds and synthetic blends, the female showed responses as well as attraction for each of the mentioned treatments when tested against the control solvent. For example, in undamaged bio, a synthetic blend of 4 compounds showing attraction. In undamaged NEER, a synthetic blend of 5 compounds showing attraction. In case of ID50 bio, a synthetic blend of 8 mentioned compounds showing attraction. In ID50 NEER, a synthetic blend of 11 compounds showing attraction when tested against the control solvent. In ID100 bio, a synthetic blend of 11 mentioned compounds showing attraction. In ID100 nirmal, synthetic blend of 15 compounds and a synthetic blend of 4 compounds showing attraction. Lastly, in case of mechanically damaged bio and mechanically damaged nirmol, a synthetic blend of 3 compounds showing attraction when tested against the control solvent. In conclusion, this study demonstrated that the affixed natural ratios of compound for either a synthetic blend 4 comprising of benzyl alcohol 1,3-diethylbenzene, thymol and 1-hexadecene at similar amounts present in volatile extracts of ID-100 nirmol plants or a synthetic blend of 11 compounds at similar amounts present in the volatile extracts of ID-100 bioplants acted as attractant of Apis crackivora. Therefore, both these blends could be used for the development of baited traps in integrated pest management strategies for controlling the pest Apis crackivora, and the blend 4 with the mentioned ratios could be recommended for use as lures in baited trap development, which will ultimately cut down the use of chemical pesticides which causes detrimental effects for the environment. Here is the published article in Pest Management Science. Here are some selected references. Thank you. Thanks to the organizer for giving me the opportunity to present my research work. Thank you.
Thank you, Parma. Very good work and very nice presentation. So, what next? Do you go for formulations? What you have done?
Yes, sir. The next step is to develop the baited trap in first of all in the greenhouse, then for further.
Okay. Thank you. Thank you. Okay. Next, Ravi Prasad, I think. Ravi Prasad from MLB Government College. Ravi Prasad, are you there? Ravi Prasad,
Yeah,
unmute
just a
yourself. Unmute.
yeah. Just a minute. I am sharing my screen.
You are sharing your from 2 devices.
Yeah, from one mobile only. Whether my PPT visible?
Yes.
Yeah. Yeah. Good afternoon to all of you. Myself, Ravi Prasad, the former student of the MBU Department of Botany, now working at the BSI Jodhpur, and at the same time I am doing my PhD from the MLB Government College, Bhilwara, Rajasthan. And today I am going to talk on a topic that is a very important topic, that is an urgent need to conserve the critically endangered Commiphora vitae in Sariska Tiger Reserve. Sariska is an internationally renowned tiger reserve of the world. It is situated in the Alwar district of Rajasthan, and it is the first tiger reserve in the world where successful reintroduction of the tiger took place. And now I am coming to the main topic about the Commiphora vitae. And this is my study area. I have started working in Sariska since 2016, and continuously I am doing my research work at Sariska. And this is the study area. And some basic facts about the Commiphora vitae, a critically endangered species. It belongs to the order Sapindalis, and the family is the Burseraceae. And the most important, just a minute. Yeah. The most important reason behind its exploitation is the having a number of medicinal properties like expectorant, cremative, aphrodisiac, etc. And at the same time, the plant is very much useful in the rheumatoid, osteoarthritis, obesity, and several other diseases. It lowers the cholesterol level of the blood. That means it is having a number of medicinal properties. And if you will talk about the chemistry, this plant is having the guaiacol 1, 2, and 3, Google lipid, gum resin, etc. And the cause behind the its its reducing number is one of the major reason is that it grows in the tough climatic condition on exposed rocky habitat in dry region. But nowadays most of the rocky habitat in the Aravalli Belt. The problem is that everywhere there is a mining, and as a result, its habitat is totally destroyed. And it is a shrub to small tree, and it is previously it was very much luxuriant in the states of Rajasthan and Gujarat, and also in the arid parts of the Karnataka, Maharashtra, and Madhya Pradesh. But everywhere now they are very much scandal. And now in this slide you can see that the main reason for the exploitation of this plant is an oleoresin injured part of the bark. What greedy people are doing, they are making a very big injury on the bark, and as a result the lifespan of the plant gets reduced. During the survey of last 5 years, I have since 1983 in Sarika Tiger Reserve, there are more than, it was quite abundant in the whole Sarika, but now in the year 2021, after the whole survey, we have recorded its population only at a 3 location, and individuals in the population are varying from 2 to 7 only. So you can imagine the situation here. This is the plant that I have taken the picture of the same plant in 2016, this one. At that time, there was a very less injury, but people are frequently making the injuries on this plant for extraction of the oleoresin, and as a result, the plant become dried. This was the picture taken after the 4 year in 2020, and the same problem. And one major problem is that the habitat destruction, as I have already mentioned, that in Aravalli, there is a huge mining, even in Siliguri and other parts of the India, wherever you are getting the stone material, that is basically mainly from the Rajasthan. And this is some of the basic morphological features. And one main thing is that it is a polymorphous flower. That means it is having a male, female, and bisexual flowers. And in nature, during survey of Sarika, I have noticed that most of the plants in nature are only female plants, and as a result, its number is also not multiplying very much. And this is the fruiting condition and the dried seed. Here you just see when even after the 2 to 3 year old plant, it produces number of seeds, but once the seed fall, there is no germination. The cause is that it's always grow in a phytosociological condition. I will show you next slide. After this thread, I will explain the phytosociological condition. The major thread that I have already explained, that just a second. Yeah, over exploitation and illegal collection even from the protected area. As you can see, this is the highly protected area or tiger reserve, but some illegal activities is still going on. People are collecting the resin and lower percentage of seed germination. Its seed germination is very poor and slow growth rate, and habitat is already lost. And one main problem with this, the reducing number of
Please unmute yourself.
Ravi Prasad, please unmute.
Hello, sir.
But you are not audible.
Sir, you are not audible.
Ravi Prasad, you are requested to unmute yourself.
কানেকশন লুজ করেছে বোধহয়। Cheers.
Ravi Prasad, we cannot hear you.
কানেকশন লুজ করেছে।
Ravi Prasad, you are not audible.
Hello, you are not audible.
Yes, sir. And your screen is not visible. Ravi Prasad.
Mr. Prasad, can you hear me? Mr. Prasad, can you hear me?
নেই কানেকশনে নেই আকাশ।
হ্যাঁ ম্যাম, বুঝতে পারছি, মানে কানেকশন লুজ করেছে হয়তো।
ও চ্যাট করতে হবে।
এই সোমেন,
He's not connected.
Akash, you can send a message to him.
Yes, sir. I'm happy.
Ravi, I can't hear. We can't hear.
ও স্ক্রিনের দিকে দেখছেই না।
কানেক্টেডই নেই।
প্রেজেন্টেশনটা কিন্তু বন্ধ হয়ে গিয়েছে, প্রেজেন্টেশনটাও
কানেক্টেডই না তো।
Ravi, can you hear me? Unmute yourself.
বুঝতে পেরেছেন?
না সঞ্জয়, নেই ও নেই কানেক্টেড।
With the kind permission of chairperson, can we?
Yeah. You can call Sushruba and we can't hear anything.
Oh, okay. We can proceed then to the next presenter, right?
Next presenter, that is Sushruba. I think Sushruba is there, no? Sushruba and
Sushruba, you are ready for presentation?
Yes, sir. Yes, sir.
Okay, please proceed.
Am I audible, sir?
Akash, disconnect.
Yes.
Sir, am I audible?
You are audible but not visible.
Okay, sir. I'm
Ravi Prasad join.
Sir, is it visible? Sir, is it visible?
Ravi Prasad join করেছে।
1 minute, Sushruba. Hello, Ravi Prasad, can you hear me? Sushruba, 1 minute. Ravi, can you hear me, Mr. Ravi Prasad?
Ravi Prasad, you are requested to
Hello?
unmute your. Yes, please.
Yeah. Hello. Yeah.
Actually, the last portion we can't hear anything. So you are muted.
Oh. Shall I present that part once again?
No. It's already late. We are already late. Okay.
Okay.
Okay. You may leave. So
Okay.
you can call the next presenter, Sushruba Chatterjee. Sushruba.
Okay. If you have any question, then you can ask.
Yes, sir.
No, I can't hear anything. I can see or hear anything.
Sir, is it?
Okay.
Is it visible, sir, now?
Yeah.
Okay, sir. Thank you, sir.
Yes, sir.
Yes, Sushruba, please start.
Yes, sir. Actually, my laptop is creating some problems. Okay, sir. Thank you, sir. So, good afternoon to everybody. Myself, Sushruba Chatterjee. I'm a UGC Junior Research Fellow of Calcutta School of Tropical Medicine, and today I will talk about the environmental pollutants and the macrophage. And from the name, we can easily understand this is the study on the effect of. Hello.
Carry on.
I think my screen is just closed. I'm
Okay,
really sorry.
carry on. Carry on.
Sir, is it visible, sir, right now? Because
Visible,
it is,
but you can carry on, no problem.
So just a minute, sir. It's starting, sir. Okay, sir. So the name, we can easily assume that this is a study on the of the effect of environmental pollutants on the macrophage. So we all know that there is a different type of toxic heavy metal are present in the earth naturally, but due to some anthropogenic reasons, the concentration of these chemicals become increasing inside the plant of a body, inside the body of a plant and animal and human day by day. And due to this, they can interact with different type of cellular metabolic pathway and can hamper the normal homeostasis of the body. So in this study, we will focus about the lead and effect of lead and mercury on the body of the albino mice. So from as the previous study also noted that lead toxicity is associated with numerous physiological pathological conditions like the nervous and immune disbalance, cardiovascular diseases and bone metabolism, and also renal and reproductive dysfunction. Also, it was reported that cell death in peritoneal macrophages can be occurred due to the exposure of the lead. So what was our aim and objective? The aim and objective of our study is to detect the normal cytomorphology of the macrophages and also to detect the exposure, any changes occurs due to the normal cytomorphology of macrophages due to the exposure of mercury and lead in the albino mice. So what was our materials and method? At first, we have maintained a number of a group of white albino mice in a clean plastic cages under natural light and condition, and we have divided these mice into 3 groups. The first one is treated as control, and they were left. They are maintained just as a normal diet. And group 2 was recognized as the treated as we have administered mercury chloride 5 mg per kg per day for 2 to 3 consecutive weeks. And group 3 was also recognized as treated too. They were administered by lead acetate as a dose of 10 mg per kg body weight for 21 days. And after that, we have incorporated the PBS buffer saline into the peritoneum of the peritoneum of the mice, and we have aspirate was taken to isolate the peritoneal macrophages. So first the rat was sacrificed, isolation of peritoneal macrophages was done, preparation of smear, incubation at 37 degrees for 3 hours, adherent cells were fixed with methanol, stained with methylene blue, GIMSA and neutral lead. Why we use 3 type of stain? Because the methylene blue and GIMSA are mainly used to study the cytomorphological character of the macrophages, and neutral lead is mainly used to detect the presence of lysosomal content inside of a cell, and also we observed under the light microscope. So the result was like that. The untreated group shows the normal cytomorphological observation, but in case of late treatment, the cells of the cytosol of the macrophages show different type of vacuoles. Sometimes 3 or 4 vacuoles are also formed, and sometimes different number of vacuoles are togetherly form and fuse to form a giant vacuole. This is called a paraptosis. It is also a programmed cell death, but it is it is a programmed cell death that is another type of programmed cell death except the apoptosis. Here, no nuclear fragmentation, no apoptotic bleeding formation, or no involvement of caspase is found. In case of paraptosis, only vacuolation procedure is also found. So in case of mercury treated rat, we found that a pycnotic type of nucleus, that means the nuclei are going to fragmentation and condensation. And as I have mentioned earlier, that we have used the neutral rate to detect the presence of lysosomal content. And in case of control macrophages, we found that the lysosome are intact inside the nucleus, and the cytoplasm is just is white. But in case of treated macrophages, the cytoplasm turns into red. That means the lysosome is leaked, the lysosome membrane is ruptured, so the lysosome is leaked into the cytosol from the nucleus. So the less viability also found. So the previous study also noted that due to the exposure of toxic metal, glucose uptake can be disrupted and glucose regulation can be hampered, and ultimately development of diabetes can be occurred. So we have also published a paper in World Scientific News about regarding this thing, and we have also reported there that due to the exposure of different type of environmental pollutant like the heavy metals, sometimes the macrophages start to increase the pro-inflammatory cytokine secretion, and also they start to kill the beta cells of the pancreas, and ultimately insulin dysregulation can be occurred. They can sometimes cross-talk with the signaling pathway of insulin and can also dysregulate the glucose impairment procedure, glucose tolerance procedure, but further studies also needed. So what was the conclusion? The conclusion was that treated macrophages showed membrane bleeding, rupture of plasma membrane, and loss of viability, and ultimately heavy metal into toxicity may affect macrophage mediated immunity. Also the further study is needed. As a general conclusion, we can easily tell that due to the exposure of different type of heavy metal and environmental pollutant, normal cytomorphology of macrophages can be altered. That may hamper the function of the macrophage, and ultimately different type of disease can be occurred. Diabetes mellitus is one of them, but further studies are in progress to solve this paradox. These are some references, and I would like to acknowledge Dr. Srikanta Guria sir for his guidance, encouragement, and sustained interest. Thank you, sir.
Kusuma, what is the application of your work?
So actually, if we can identify the what was the procedure by which the
lots of works are already have done regarding diabetes. Different types of diabetes are there, gestational diabetes. So
yes,
lots of work. So what is the application? Macrophage, all we know there is a role in diabetes, or macrophage environmental pollution is there. So what is the application of your work?
Actually, sir, the further studies also needed, but if we can identify which signaling pathway macrophage, pro-inflammatory macrophage are crosstalk, if we expose the different type of heavy metals or the environmental pollutant, then it can be work as a diagnostic approach for future further.
Okay, Shosuma, thank you. Thank you for your nice presentation.
Thank you, sir.
Email.
Okay. Chairman sir, only one presentation is left, that is Mintu Desai.
Yes, sir. Yes, sir.
Yes, Mr. Mintu Desai.
Hello.
Hello,
Myself Subhajit Ojha from Darjeeling Government College. I have presented presentation this session. So after that, I think
your name is included in the 3A session.
Yes.
No. Subhajit Shah is not here.
Subhajit Ojha.
Subhajit Ojha is not here. Akash.
Okay. I'm just a minute. I'm finding that part. Okay.
Actually, the PDF I have there, my name is mentioned there. That's why I'm asking.
Okay, Honorable Sir, you just
Sir, excuse me, sir. Sir.
Yeah.
Yes.
Sir, myself, Dr. Momita Sinha from Nistarani College, Purulia. Is my
actually
name
Momita
is
Sinha is
included
after
here?
my name.
Yeah.
Yes, probably you are in 3B section, right?
Actually,
no, sir. It is included in 14 number of 3A section.
Yes.
After Subhajit Ojha.
Yes.
Dr.
Myself
Subhajit
in the
Ojha.
13. After Ravi Prasad, my name is there actually.
After Ravi Prasad, your name
yes,
is there,
yes.
na? Okay.
Yes.
Let me clarify. Just start the Mintu Desai presentation.
Okay, sir. No problem.
Okay, sir. Thank you, sir.
Okay.
We will accommodate yourself into the 3B section, right?
Okay, so I can shift to the next another meet or stay here?
No. You will just click on 3B. Okay.
Okay.
Mohita Madam and Shubhajit sir, you are requested to enter through the another link which is being provided as for 3B section. Okay, I am just reporting to the moderator.
Okay, sir. No problem.
Sir, আমরা কি তাহলে ওখানে জয়েন করব 3B তে?
3B তে, Momita Sinha and Shubhajit Ojha. You are requested to join through 3B section.
Okay, sir.
Okay,
only one presentation left in this section, that is
Mintu Desai.
Yes, please.
Mintu, please.
Okay. Thank you, sir. Sorry, is my screen is visible?
Yes, yes, visible. Continue.
Okay. Myself, Mintu Deasi. My topic is mud crab farming, a promising aquaculture activity in India. Mud crab, also known as the green crab or mangrove crab, belongs to the genus Scylla and occur widely in the estuaries and marine environment along with the tropical, subtropical, and warm temperature in the world. Mud crab constitute the important secondary crop in the traditional prawn and fish culture, and it's become very popular due to its large quality and mid quality and large size. And this crab has the immense market demand, and farmers can earn the good incomes by selling in the local and export market. In Southeast Asia, this kind of farming is carried out in large scale. And in India, basically in the coastal region, that is West Bengal, Kerala, Tamil Nadu, it is practiced regularly. So these are the four basic Scylla species, that is Scylla serrata, Scylla tranquillarica, Scylla parabensine, and Scylla olivacea. These are used for the both commercial fisheries and aquaculture productions. Generally, males attain the 20 to 24 centimeter in carapace length, whereas female attain the 12 to 13 centimeter in their adult conditions. And this is the distribution and habitat of Scylla species. Among these four species, Scylla serrata is most prevalent in the Indian Ocean as well as Indian subcontinent. Except the paramoeciums, three species are well known and well found in the Indian Sundarbans. And Scylla serrata is the most prevalent and abundant among the four Scylla species in India. So now I will just discuss about the different kinds of mud crab fishings. Here the hand picking method is very popular here, and crab fisher just catches this particular crabs by using the primitive indigenous tools and like forceps, scoop nets, baskets, khuli, and chutney jar, etc. And in the hook fishing, farmers can use a J-shaped iron rod, and they just thrust into the crab hole and detect the location of the mud crab. And along with this, there are some single baited line as well as multiple baited lines process for the crab capture is there. In a single baited, just one bait is given, as well as in multiple baited, there are different multiple baited are used. Here some small species of eel fish as well as some trash fishes are also used as a bait. And now I'll just discuss about the different farming. Due to great demand of the mud crabs in domestic as well as export market, commercial scale of its farming is developing very fast along the coastal area in India and neighboring countries. And different kinds of farming systems have been operated in different Southeast Asian countries, like grow out systems. It is also carried on the pond as well as the mangrove. In the crab fattening systems, next one is the box culture systems and the soft shell crab farming systems. So this is the required soil and water quality parameters for the mud crab farmings. Diesel oxygen is greater than 5 ppm, pH is required for the alkaline water, temperature is 25 to 35 degrees centigrade, salinity is 10 to 15 ppt, and like this. And mainly the sandy clay type of soil is most suitable for this kind of farming. And now in this growth culture, basically the young crabs are grown for a period of 6 months, and here it is in ponds. So in the size of the ponds is more about 0.1 to 0.6 hectare, and water depth is about 5 feet. And there are some bath as well as net fencing is much more required just to prevent the crab migration, as well as some paddle wheel aerator are also required for the water circulations. And in the systems, some trash fish as well as some wet feed are given in the pond to feed the crabs. Here after the 6 months culture, Scylla serrata is at an average of 900 to 100 gram, while the females attain 600 to 70 gram with a survival rate of 60%. And here the manpower and technical skill are required for the pond construction, management, feeding, and harvesting. And another advantage of this pond culture is that there are polyculture is also carried out with the different finfish and shellfish species like milkfish, mullets, as well as different kinds of shrimps also, and the benefit cost ratio is very high. Now in the pen culture, the pens are constructed entirely of the wood and plant fencing as well as knitting with the wooden supports, and it is just designed to retain the mud crab farm stocks within a specific area of mangrove. And here the 1 to 2 centimeter mesh of net size is required to prevent the crablets just from their escape. And in the calf fattening system, it is very popular because of its fast turnover, low operating cost, and high survival rate and good demand in the end product. So calf fattening is locally known as the chamber chas in Sundarban. So in particular systems, the adult calves which are considered to be the unfitted for export are cultured in the small brackish water bodies for a short period of 15 to 10 days or maximum 1 month. And it is also carried out on the pond or floating cage or mangrove pen also. And the recommended stocking density is 9 to 10 calves per square meter in cage and 4 to 5 calves per square meter in pen. And nowadays, in recent year, a capital intensive box culture method of crab growth is getting very popular, and here 1 crab per box and are cultured for the 4 to 6 months and attaining 200 to 400 grams for Scylla olivaceae and 300 to 900 grams for Scylla cerevita. And this particular system has an advantage that include the lower maintenance, predictability, easy maintenance, etc. And it is also employed in the indoor systems where scarcity of land and water is found. And these are some modifications as well as some advanced technology that is used in the box culture systems. These are different styles of boxes. These are the vertical recirculatory aquaculture systems, and these are some modifications of submersible box systems that is also used in these systems. And now this is the soft cell crab farming. The soft cell crabs refer to the physiological state of any crab that is just undergo molting to replace their old hard exoskeleton with a new, slightly larger and decalcified exoskeleton.
Into Desai, you are requested to conclude.
And these kinds of farming is very, very popular in Thailand, Myanmar, Vietnam, Malaysia, China, Indonesia also. And here in a large scale, these kinds of farming produce the soft shell crab. In this particular countries, this soft shell crab has a huge demand. And now this is the production and return of mud crab farming in India local market.
Mr. Desai, go to the conclusion slide because
yes,
we are running out.
Yes, sir. This is the live crab. This is the anyone can earn some 80,000 rupees just for 1.1 hectare also. And there are also the major constant also that is the aquaculture is a large commercial scale due to the non availability and inconsistent availability. Hatchery technology has limitations for this poor survival larvae, and lack of good quality of commercial feed, lack of infrastructure, technical knowledge, marketing access, and obviously there are some coastal pollution and mangrove habitat degradation problems. And there are also some research development and awareness, and there are some center for like Rajiv Gandhi Center for Aquaculture or IMPEDA or SIVA or CMFRI. They can regularly research as well as they engage to produce the highly fertile and highly good quality crablets. And this is my conclusion. That is, the mud crab farming is becoming very popular in the coastal district in India, owing to the good demand and better price of the mud crab in the international market. Mud crab has emerged as both potential exportable commodity as well as means of livelihoods of coastal port communities in India. Polyculture of mud crabs might provide an efficient utilization of resource. There is a good opportunity for the sustainable management of the mangrove systems, and this kind of farming can provide the employment opportunities for the fisher folk as a means of alternative livelihoods, and there are enough scope of research and improvements also. And this is my bibliography. And I would like to thank the organizer committee for providing me this platform and present my work, and also I like to give the thanks to the chairperson also. And thank you.
Okay, Mr. Desai, you have taken too much time. Desai, this is your original work or review work?
No, sir, it is a review work. But okay. I am working on the different crafts that is also available from our Chiti Kankadar.
Yeah. Actually, there are lots of papers regarding the mud crab fishing has already been published. Okay, so that is the one thing I can share that those fishermen who are working with the mud crab, they are experiencing some diseases. I think some zoonotic diseases are there in Sundarban.
Yeah, exactly.
Yeah. So,
but I just focus only the mud crab farming and its potential.
Yeah. I know that. I know that. Okay, thank you.
Okay,
very much.
Thank you, sir. Thank you.
So, Akash, I think we have concluded all the.
Yes, sir. You just please conclude the session.
Okay, the session is concluded. All the participants are presented their papers, and some of the papers are very good, and some of they are presented in nice presentations are also. So I would like to conclude this session now. So, what to Akash?
Thanks, Professor Sanjay Poddar, an international scientist of fundamental research in acarology, allergology, ecology, how just mice, feed plant mice, feed allergy, mesodental allergy, these disorders, who was with us in this chair of this session, brushing out all of this extremely big schedule to this program. We, on behalf of the senior organizing committee, APC, in this international web platform, thank you, sir. We hope your gracious presence in future programs organized by our college. Thank you, sir, for your
Thank you.
presentation.
Thank
Thank you,
you.
sir.
Okay, okay. May I leave, Akash?
Yes, sir, please.
Okay, thank you. Thank you.
And this is an announcement for the delegates, participants, and viewers. You are requested to join the program. The next session will start sharply at 4 p.m. So, on behalf of organizing committee, then humble request or to all of the viewers, presenters, and etc. You please join the specific link in the next session that will sharply start at 4 p.m. Thank you.
Okay, thank you, sir.
Okay, sir. Thank you, sir. এবার খাবো 4টার সময় আছে
Good afternoon. This is Rajat Kumar De. Is there anyone on behalf of the organizing committee?
Hello, hello. Hello, sir.
কেউ একজন অ্যাডমিট করাতে থাকো। Good afternoon all. We are at the second technical session of the second day of International Interdisciplinary Web Seminar on modern trends in humanities, science and technology and social sciences for sustainable development. In this session, we have with us Professor Rajat Kumar De, Department of Physics, University of North Bengal, as
Honorable Chairperson of this session. This session is specially designed for multidisciplinary disciplines like physics, chemistry, and related subjects. As it is multidisciplinary in nature, so we maintain the stringency of the program, and to accommodate different discipline paper in this session, I request Professor Rajat Kumar De to be in the chair of this session, and in that case, I request Professor Kartik Chandra De to be a moderator along with our student volunteer. They will help Professor Rajat Kumar De to continue the session. I request Professor Rajat Kumar De to chair over the session. Rajat Kumar De, sir.
Thank you for your kind words. Good afternoon.
Yes, sir. Yes, sir, please.
Can I proceed? Dear colleagues, my dear participants, it's a pleasure to welcome you all to this technical session of the ongoing International Web Seminar 2021. The theme they have set, Science and Technology for Sustainable Development. Today, the list I have been received, we are having 11 oral presentations in this particular session. The time for each talk is to be 8 minutes. Out of which 7 minutes for presentation and 1 minute on question and answer, discussion.
Sir, I want to add some information, if you please allow me.
Yes, yes.
As we have some international speech from invited speaker, so we have a time crisis. So this is an request from the organizing committee to our honorable chair as well as participants to please restrict your presentation only 5 to 6 minutes and 1 to 2 minutes for questionnaires, so that we can conclude the session within the period, so that we can enjoy the international speech. It's an humble request to Professor Rajat Kumar De and all the delegates and presenters. Sir, please.
Sure. That's why I have just told them 7 minutes for presentation and 1 minute for discussion.
Thank you, sir.
Okay.
Thank you,
question
sir.
and
Now
answer.
I hand over the charge of moderation to Professor Kartik Chandra De and Professor Shobhata Ghosh. আমার নামগুলো বলবে, স্যার তো আছেন। Honorable Chair of the session, Dr. Rajat Kumar De, please you may start the session.
Thank you. For participants, I will inform you after 6 minutes to remind you that you will have 1 minute left, okay, during your presentation, and you will be able to have 1 minute for discussion. So 6 plus 1 plus 1, okay. Before the 7th minute starts, I will inform you that you have 1 minute for presentation, and also you will be able to have 1 minute for discussion. Okay. It is a great pleasure to invite the first speaker of today's afternoon session, Tuhin Ghosh, Assistant Professor, Durgapur Government College. Okay, he will be speaking on arabinogalactan polysaccharides containing unica ... in vitro test. Okay, let's get started. Dr. Tuhin Ghosh, please.
Yes, yes. Thank you, sir. Thank you, Professor Rajat Kumar De, for allowing me to speak in this session. Yeah. Is my screen visible, sir?
Yes, yes.
Yeah. Thank
Yes,
you, sir.
sir.
Thank you. Yeah. At the onset, I'd like to thank the organizing committee of Dr. APC Roy Government College for allowing me to speak, and especially Dr. Mohik Sarkar. So I'm happy to be here in front of you to present one of my research work, arabinogalactan polysaccharides containing Punica granatum peels for antioxidant capacity from material characterization to in vitro test. I am Dr. Tuhin Ghosh, Assistant Professor of Durgapur Government College. A little bit of introduction. Natural products have been invaluable as biologically validated platforms for development of new antioxidants. The extracts from natural products containing an emerging class of bioactive compounds, especially polysaccharides, exhibit a large range of properties. The polysaccharide extracted fractions from natural sources have shown in vitro antioxidant activities, but still a vast area remains to be explored regarding the in vivo antioxidant activity of the biomolecules for exhibiting promising antioxidant activity. In this work, we have been working on the edible fruit, that is pomegranate, which is generally sweet in taste, as you all know, and it's available throughout the year in local markets, and the fruit juice is considered to be an essential health benefit refreshing drink. Beside the edible fruit, our main focus was on the hard peels pomegranate, which is generally considered as the non-edible part that is left after the extraction of the juice and is being usually discarded as a waste material in huge amounts. So what we found that there was no report on its macromolecules present in the peels earlier. So we worked on that, and thereby our objective was to identify the chemical structure, that's the chemical profiling of the polysaccharides, which are isolated, extracted from the peels, and the antioxidant capacity of the different active fractions was thereby reported. So what we decided to do is that we used very cheap polar non-toxic solvents following standard procedure which was available. I have a little bit of background on working on carbohydrate chemistry, especially the polysaccharides. We used established bioassays available for the in vitro antioxidant activity, which has the ability to reduce oxidation stress. So I will skip this slide due to time constraint, as already informed by Honorable Chairperson. This is the overall scheme of my work, which I have elucidated in a single slide. And that's the fruit depicted there on top left-hand side, the extraction procedure, and the structural elucidation, that's the chemistry part, which was done by sugar composition analysis, and thereby linkage pattern of the polysaccharides was detected by methylation analysis and Smith degradation if required. Next, what we did was the spectroscopic analysis of the, sorry, I missed the purification fraction, and anion exchange chromatography, size exclusion chromatography was used to determine the molecular weight. After that, what we did was we used simple spectroscopic methods, NMR, IR, UV-Vis, as required, and then the in vitro antioxidant activity on the extreme left-hand side bottom. So this was the overall scheme of the work. This is my extraction method in a single slide. That's the water extract, and then the ethanol precipitated fraction, that is the PF1, and the ethanol soluble fraction was marked as PF2, and the acetone soluble was prepared from the residue was marked as PF3. So as we all know, that size exclusion chromatography is a very important technique for determining the molecular weight, and we did it using cephacryl column, and we detected the molecular weight to be 180 kDa. I'm not going into the details due to time constraint. This is after extraction. What immediately follows is the purification. So there can be a multiple process for, multiplication process for purification, and we use dialysis, we use graded precipitation, especially for this work, and which was very successful. There can be also chromatography, which I earlier talked about. Now, this is the sugar composition analysis. We have a GLCMS support from the University of Burdwan Chemistry Department, and I use that facility because I was a research scholar there before joining this position. So this is the standard sugars which were detected, and from that my unknown sample was detected from the gas liquid chromatography mass spectrometer. This is the result of the sugar composition analysis, and as I pointed out in my topic, that arabinogalactan, so we can see that arabinose was found to a major extent, and galactose, these two, so which is highlighted here. So the linkage pattern has to be detected for polysaccharides, which is very important. So how it is linked? So the structures, I've drawn a little bit of chemistry here. We have used the methylation technique. That's a polysaccharide which was broken down, and then it was methylated, partially methylated. So what is important, that the position which are linked do not get methylated. So the position which is free gets methylated. So this was the science behind the glycoside linkage analysis, and this is the GLCMS analysis of the methylated arabinogalactan polysaccharides. And the probable structural motif of the polysaccharide present in the peels of pomegranate was the 136 linked galactan, and 13 linked galactan was found to a bit terminal arabinose, as well as, yes.
You have
Yes.
1 minute left for presentation.
Yes, I'm completing. Yeah, and this is the methylation analysis was confirmed from the proton NMR spectroscopy. This is the peak for terminal arabinose, and this is for the anomeric proton of 13 linked and 15 linked arabinose. So next, which is followed, that the antioxidant capacity, we use techniques for DPPH radical assay and FRAP assay and hydroxyl. And this is the DPPH assay, which we found that the PF1 fraction was active in all the assays. So the water extracted, purified water extract fraction was active in all the assay compared to the reference used. These are the plots. So finally, the conclusion, the arabinogalactan of 180 kilodalton molecular weight was isolated from the pomegranate, showing promising result to be used as potential antioxidants. The linkage pattern followed as shown in my figure. The polysaccharides was isolated without toxic chemical reagents, so these fractions may be used as a potential as safe antioxidants. Further, as the isolation of these polysaccharides involves few inexpensive and easy steps, it will be of added advantage. Of course, future research in this structure-activity relationship is possible. Before I end, I would like to acknowledge my PhD guide, Professor Vimalendu Rai, Dr. Dipon Adhikari, Hubli Motion College, Durgapur Government College, my working institution, DST Government of West Bengal for Infrastructural Development Funding. These are my two MSc students, Shodham Shai and Ms. Navonita Ray. And finally, the organizing committee, once again, EPC Roy College and UGC-HRDC NBU for providing me an opportunity. Thank you. Thank you. Thank you, sir.
Thank you, Dr. Ghosh, for your nice presentation. Now, the talk is open for discussion. We can allow 1 to 2 questions from participants. Participants, you can raise question for Dr. Ghosh. Okay, in between, I would like to make a comment, Dr. Ghosh.
Yeah.
Does it can do anything as inhibitor of SARS-CoV-2?
Sir, please repeat, sir.
SARS-CoV-2, coronavirus 2.
No, sir. No, sir. I haven't checked that. That's a lot of work. I haven't checked that. It's a separate part, sir. I have worked on the antioxidant capacity, not on the viral study. No, I have not done.
As a non-expert, okay, I have raised the question.
Yeah, thank you,
I
sir.
am not an expert in this field
Yeah, thank
because
you.
pandemic is going on, no?
Yeah.
That's why it is usual. If there is no question from the participants, then we should move on to the next speaker.
Yeah, thank you, sir. I'm leaving.
Yeah, yeah. So our next speaker is Dr. Lovely Sarkar,
Yes,
Assistant
sir, I am
Professor,
hearing.
Siliguri College. You can
A
start your.
very good afternoon, respected chairperson, organizers, and my dear colleagues present over here. Sir, I hope I'm visible, that my screen is visible. It's okay?
Yeah, now your screen is visible.
Okay, sir. So I will be discussing on exposure of pesticides and impacts on its health and soil characteristics. So it's a little bit related to the chemistry, and few part is obviously with the environmental chemistry. We know that professional as well as environmental regulation of chemical pesticide results in an extensive assortment of individuals' health troubles. The present study that reviews the effect of chemical pesticides upon physical condition of the crop cultivators at a particular village, that Rishipur village of Malda district in West Bengal, in view of impact upon the health of the pesticides users by recognizing way and procedures of different types of pesticides applied used.
Dr. Sarkar,
Yes,
please
yes,
put
sir.
your presentation in the presentation mode.
Sir, it is, and I have done it.
Is it the presentation mode?
Yes, sir, I have done it.
Okay.
But
Okay,
I know
continue.
what is. In my device, it is in presentation mode.
Yeah.
Okay, sir.
At least
So I'm
this.
continuing.
Okay, okay.
So for the sampling and data collection, a total of around 104 respondents were selected randomly, and primary data were collected with the help of well-prepared questionnaire based on frequently used chemical pesticides, symptoms of sickness linked to exposure of pesticides, some physicochemical characteristics of the soil of the land used for the cultivation in the study area. And to carry out the study of the soil samples that were collected in the month of July and November of 2018. And a particular site of the agricultural field was selected to study the physicochemical nature of the soil of that area. The soil samples were collected in a clean, sterilized plastic bottles and supplied to the laboratory for the parameters, determination of the parameters like temperature, pH, electrical conductivity, calcium hardness, magnesium hardness, carbonate alkalinity, bicarbonate alkalinity, and chloride content. So I'm not going to that part of the determination of the directly chemical parts. So these are the common pesticides used by the farmers, and the percentage I have shown in the table that mostly in that case they're using perhaps that cartap hydrochloride, and in the sense that only they are not using that only as a pesticide. They sometimes it's very dangerous that they are using it as fertilizers also. So the direct effect I'm coming, that now there are copious evidences of pesticides, chemical that are presence of impending impact to human beings and other living existence, and surplus side effects to the surrounding of the environment. And usually in most of the developing countries, farmers are observed to report their sickness and hospitalization due to application of pesticides. And some symptoms associated to the application of pesticides, in my case, in my studied area, with a higher parasiveness, where the burning sensation, direct using of the chemical pesticides. Skin problem, including irritation, redness, or white patches like breath shortness, eye burning, itching, vomiting tendencies, headache. So there are a few health effects which are directly related to the farmers and related to the chemical pesticides. And it is obviously, it has been proved also that the percentage of farmers, they are affected at a great extent rather than the percentage of farmers who are not directly using the chemical pesticides. So those are not using as those are not actually spraying the pesticides directly in the agricultural fields. So we can just here is a comparison that the percentage of farmers who are directly spraying, those are more affected than those are not spraying, not sprays.
Dr. Karthik De.
Again, it is.
Dr. De.
Yes.
Can you see the successive slides?
Actually, it is not in a presentation mode, I think. Dr. Sarkar was telling that it is in presentation mode, but I think
I am just seeing the first slide. Yeah, now it is working.
Yes, yes.
Now it is working.
Okay, okay.
So now it is the physico-chemical parameters of the soil I have done. So for the physico-chemical parameters, first the temperature I have measured, and it was observed values of the temperature was 26.3 to 22.5 degrees centigrade in July and November respectively. And whenever the pH was measured, it was around between 7.9 to 7.1, that is in July and November. Whenever it was electrical conductivity, that is for in constant, it was that 0.162 to 124. That's the parameter. So all of this we have calculated, and further few studies that have been left, and in future we have a plan that the heavy metals constituents, that concentration of those metals we have to study further one. So next is there is a comparison where we have done that which metals are more in case of July and in case of November, that all the anions, those we have studied, the concentration in July, it is a little bit to the extent it is higher than in comparison to in the month of November. Then if I come to the conclusion part, that in order to realize the physico-chemical characteristics of the soil in the studied area, several parameters were taken under consideration. And the disintegration of organic substances, which is allowed, which is followed by the association of soluble components in the soil with the increase of temperature of the soil. And pH of the soil under this experiment was seen to vary from that 7.1 to 7.9. That indicates that alkaline environment of the soil. So the higher pH was observed in the month of November, depicting development of salinity in the soil. And the higher value of electrical conductivity, that is because of higher concentration of the dissolved ions present in the soil components, and which further supports that the leaching process occurring in the rainy season as well, so in comparison to the month of November. And in the month of November, the concentration of calcium ion and the magnesium cations is relatively lower. That determines less attachment to the organic components as well as soil clay matters. So further, carbonate and bicarbonate anions in combination with the calcium and magnesium form that precipitates as calcium carbonate or somehow magnesium carbonate when the soil solution gets concentrated in the month of November. And in concern with the chloride ion, that is one of the most essential elements regarding plant growth. So here also an overall impact has been studied on the cultivators due to frequent use of a number of chemical pesticides, resulting in direct health effect and also maybe for long-term effect. So it is to be expected that application of chemical pesticides would be applicable with proper awareness. And for this, I have used few references. And last of all, thank you so much to everyone for your great patience.
Thank you, Dr. Sarkar.
Thank you,
You
sir.
have completed within your stipulated time. Now the talk is open for discussion. We can take one or two questions from the participants. If there is no question, I would like to just know from Dr. Sarkar.
Yes, sir.
Did you measure exposure level, pesticide exposure level in human?
No, we have a plan. We have just started it in 2018, and in another work is going on. So we have a plan to study more, including the water quality also.
I see. Where are you doing this research in North Bengal University or Siliguri College?
Yes, sir. From Siliguri College, and obviously with the help of North Bengal University.
I see, I see. Thank you very much for your nice
Thank you, sir.
presentation.
Thank you so much, sir.
So let me move on to the next speaker. Our next speaker is Dr. Sayantani Chatterjee, Assistant Professor, Vijayawada Jyotish Rai College, Calcutta University.
Good afternoon to the respected chairperson, dignitaries, and respected faculties present over here. Sir, am I visible? My screen is visible?
Yeah. Screen is coming. You are audible.
Okay, sir, let me share. Sir, is it visible?
Yeah, it is visible.
Yes, sir. My today's thank you. First of all, thank you for giving me opportunity to present here my research work. My today's topic is resemblance of hydroxy 19-tetraquinone with anti-cancer drug anthracyclines. So, first of all, introduction little bit I'm giving. Nowadays, we all are aware about the fact that cancer has become an epidemic to our society. Recent survey has revealed that this disease has touched every family at least once. So this is alarming and challenging to our society to combat this disease. There are many types of treatments are taken into serious consideration. Chemotherapy and radiotherapy in conjunction with surgery has become a significant approach in this regard. And there are photodynamic therapy and gene therapy are also taken into serious consideration. But in case of the early detection of this disease, chemotherapy is today a significant approach, and there are many types of chemotherapeutic agents are using successfully using in this regard. Anthracycline and its analogs are well known among them. Now anthracyclines has the structure like this. This is a tetracyclic ring structure with a glycosidic linkage to a sugar moiety, and doxorubicin, daunorubicin, epirubicin, ederubicin seen from the slide. These are the well-known chemotherapeutic drug, anthracycline drugs, which are successfully using. We are using successfully. Now let's move on to the mechanism of action. Till today, anthracycline, the exact mechanism of action is not known clearly. There are many types of pathways are suggested. Among them, free radical generation by this anthracycline drug, that is semiquinone formation by one electron reduction, plays a major role in determining cytotoxicity of these drugs in cellular system. Semiquinone free radicals are oxidized by molecular oxygen, forming superoxide radical anion and other reactive oxygen species that are responsible for their cytotoxicity. Another important mode of action is intercalation between DNA strands. This is seen from the slide that this is the DNA structure, double helical structure, and anthracyclines are intercalates between these double helical structures, and thus cell killing occurs. Now, another dominant pathway is topoisomerase enzymes inhibition. The topoisomerase enzyme that unzip DNA during replication. And nowadays, this is a modern technique to poison the enzymes which are important for cell viability. Now let's move on to the limitations of these drugs. Like other chemotherapeutic agents, anthracyclines are also associated with cytotoxic, cardiotoxic side effects. The superoxide radicals, that is the free radicals generated by anthracyclines, are also responsible for their cardiotoxicity. Now, various modifications were tried. Among them, metal complex formation is one of the major among them, and researchers also started to search possible substitute for it. And another aspect is their high cost, which put on a limit on their use. Now, it was found that hydroxy 9-10-anthraquinone, which is the core moiety of anthracycline, and has structural similarity with anthracyclines with regard to disposition of the OH group, And it is also the biochemical, biophysical, electrochemical behavioral center of anthracycline drugs. And it is being much more smaller. The steric hindrance is very much smaller, and can avoid steric interference in crossing barriers, enabling better binding to DNA. And another advantage is hydroxy-9,10 anthraquinones are soluble in water and DMSO and may be used under physiological conditions. And their DNA binding abilities
Dr.
are
Chatterjee, you have
Yes,
last
sir.
1 minute.
Yes, sir. Now, due to time constraint, I have to summarize. This is I have chosen quinalizarin in this regard, and 1,2,5,8 tetrahydroxy-9,10 anthraquinone is quinalizarin, and it has similarity in structure with carbinomycin and novolamycin, and these are the resonating structures. And the physicochemical studies were done with regard to quinalizarin, and pH values, pK values were obtained were comparable with that of anthracyclines, and its metal complexes were also prepared like anthracyclines, and their binding constants are also comparable with that of anthracyclines, and the DNA binding studies were also done. These are the binding isotones with ctDNA. Let's come to the result. Now, binding parameters, from the binding parameters of the quinalizarin with ctDNA, it was found that with increasing pH, there is a decrease in the DNA binding ability. So, in order to increase the DNA binding, metal complexes were prepared, and their DNA binding studies were done, and it was found that there is no decrease in the DNA binding ability with increasing pH, and the pH and the DNA binding ability.
Please move to your last slide, conclusion area.
Yes, sir. Now there are NADH dehydrogenase studies and in vitro topoisomerase studies were done, and cell line studies were also done, and it can be concluded that the DNA binding ability of the metal complexes of anthracyclines are better than that of the parent compound, and quinalizarin and its metal complexes showed comparable cell killing on MALT4 cells, but THAQ probably doing so by superoxide radical formation, and its metal complexes are doing so by topoisomerase enzyme inhibition 1 and 2. And this is less costly and more available, and if it is proved to be anti-cancer agent, then it will be beneficial for our society. Thank you, sir, for your patience. Thank you all. And a part of this study has been published in these two journals, Journal of Physical Organic Chemistry and ACS Omega. I'm thankful to the organizing committee, Acharya Prafulla Government College, and UGC-HUMAN, North Bengal University, and I'm thankful to my PhD supervisor for his cooperation, and also Professor Hemanta Kumar Majumdar and Professor Shushanto Roy Chaudhuri of IICB for their help in doing enzymatic assay and cell line studies. Thank you so much.
Thank you, Dr. Chatterjee. Due to time constraint, we can't take questions from the participants. Sorry for that.
Thank you so much, sir. I'm leaving.
Let me now move on to the next speaker, Dr. Bishwajit Pal. Dr. Pal, please start your lecture. Dr. Bishwajit Pal, Krishnanagar Government College.
Thank you, sir. I am sharing my window. Is it visible?
Yeah, it's coming.
Thank you, sir. Hello, everyone. Very good afternoon. Today, in this web seminar, the topic of my presentation is modeling of complex network of physical phase log loops and the emergent spatiotemporal patterns. Firstly, I would like to introduce about the digital phase lock loop. Digital phase lock loop is basically a feedback control system, and it uses an incoming signal and produces an output signal that is synchronized to the incoming signal. To explain this, we consider a radio transmitter and a radio receiver. There are two clock oscillators, and initially these two clock oscillators are desynchronized. The PLL, which resides inside the receiver, actually synchronizes the clock 2 with the clock 1, which is essential for synchronous communication. These DPLL are observed in most of the digital world, like electronic communication, signal processing, in computer, in microprocessor, memory subsystem, etc. There are several types of DPLL. Among all those types, the simple structure and mostly used DPLL is ZC1 DPLL, called zero crossing DPLL. The block diagram of this DPLL looks like this, and the system equation of this loop looks like this. This equation is basically a nonlinear equation, and K1 is the gain parameter and Chi is the normalized angular frequency of the incoming signal, and phi k is the phase error at the kth sampling instant. This type of equation shows bifurcation and chaos. So this complex behavior is shown in the bifurcation diagram in 1 parameter and 2 parameter plot. The idea of the work is that this type of nonlinear systems are rarely used as an isolated system. Contrary to this, they are actually used in coupled network like internet, satellite communication, FPGA system, computer networks, etc. Now, we are keenly interested to find the aggregate dynamics of those coupled networks, and we are motivated to construct a real complex network with different coupling topologies. There are different coupling topologies which we have worked out. One is local coupling topology, and another is non-local coupling topology. In local coupling topology, a node is coupled with the nearest neighbor only, and in the non-local coupling topology, a node is coupled beyond the nearest neighbor up to some extent. Now, we construct a one-dimensional network of DPLL in the form of shown in the figure. Like this is a schematic diagram. Here, the feedback signal are added to by an adder. The red X actually represent the nearest neighbor coupling signals. The system equation of this system looks like this, and here I is the spatial index and K is the temporal index, and epsilon is the coupling strength. Here we has done some experiment on 256 number of DPLL coupled in such a manner. We find the stability criteria and found several spatiotemporal patterns like synchronized fixed point pattern, Gaussian random pattern, pattern selection, spatiotemporal intermittency, and spatiotemporal chaos. In synchronized fixed point pattern, most of the spatiotemporal dynamics is synchronized to a fixed point, where all the DPLLs are synchronized spatially and temporally. Such dynamics is important for stable operation of the system and for reliable operation of the network. Then we found, as we increase the value of the K1, we found Gaussian random pattern. Here, total spatial extension is subdivided into several domains which are randomly spaced, and
Dr. Pal, you have 1 minute left.
Okay. Then we found several patterns like pattern selection, zigzag pattern, spatiotemporal intermittency, and spatiotemporal chaos. We plot the total phase diagram containing all the different dynamics in the phase space. Then we do the same work with non-local coupling. This is the schematic diagram for that, and this is the system equation. We found stability criteria, and this is the phase diagram. Here the new pattern is the chimera state. Chimera is the incongruous coexistence of synchronous and desynchronous oscillator. Here some oscillators are synchronized while the others are desynchronized. We show the transition from synchronized fixed point.
Please go fast.
Okay. This actually
Go to
transition
your
from
last slide because we have very stipulated time constraint.
Okay. Actually, this is a transition from synchronized fixed point to chimera state. We found some standing wave pattern that changed with coupling range. This is the plot of bifurcation diagram, and the shaded region actually shows chimera state. And the conclusion is that our study guides engineer to construct synchronous coupled network that is much reliable. This is not purely mathematical model. This is actually a real complex model based on some engineering system. The pattern formation, such pattern formation actually reported earlier, but it is the first time on such engineering system like electronic system, and chimera state was first explored in this type of engineering system. And these are the references, and it is published. Some of the part is published into papers, and thank you all.
Thank you, Dr. Pal. This was an interesting work you have presented, but due to time constraint, we cannot take questions from the participants. Thank you once again. Let me now invite Dr. Dilip Pal, the next speaker.
Yes, sir, I am here.
Dr. Pal, you have you will have 5-6 minutes time, okay, in total.
Yes, sir. Yes, sir. Sir, good afternoon, Chairman, sir, organizing committee members, and all others present in the session. I am sharing my screen here. Sir, is it visible?
Yeah.
Sir, the title of my paper is Inflationary Universe with Exotic Fields. The aim of the work is to construct a viable cosmological model. And cosmological model is a mathematical description of the observed universe. Now, the recent astrophysical data obtained from high redshift survey of supernova, cosmic background explorer, and Wilson microwave anisotropic probe predict that the present universe is accelerating. It is also believed that the universe might have come through an inflationary phase in the past. Inflation is permitted with an equation of state P equals to omega rho, where omega is equals to -1. Such a state, however, may be obtained either with scalar field or an imperfect fluid in general relativity. Most successful theory depends so far is based on the dynamics of scalar field with suitable potential which drive an inflation and describe the evolution of the universe. However, the present acceleration of the may be described with omega less than -1. Such an equation of state is not permitted with usual matter in the standard model of particle physics. This modification of the matter factor of Einstein's equation with new field, perhaps the new physics is to be explored. Such condition is attained if one considers exotic fields. Such exotic fields are tachyon, phantom, etc. Focusing on the recent development, I discuss inflation in the universe considering scalar and different type of exotic field. Now, the standard cosmological model based on Einstein's general theory of relativity with perfect fluid assumption. In 1917, Einstein wanted to build a static model of the universe but failed. Einstein modified the field equation, introducing a repulsive term known as cosmological constant. Then Hubble's discovery made it clear that the universe is not static but expansive. Theoretically, non-static expanding universe solution was obtained by De Sitter-Friedman Lemaitre independently without lambda. Now, according to the Big Bang model successful in explaining some thing that is 2.7 degree Kelvin, cosmic microwave background, radiation, expansion of the universe, primordial nucleosynthesis, cosmic abundance of light element, but it fails to explain flatness problem, horizon problem, singularity problem, small scale inhomogeneity problem. Some of the outstanding problems can be resolved satisfactorily by advocating the idea of inflation. Wiener was the first to know the inflationary solution may be obtained in general theory of relativity for an appropriate state tensor corresponding to the matter with properties of vacuum. Then many others are attained in this field, that is for inflationary universe model for inflationary universe that employ temperature dependent phase transition mechanism. Universe expands exponentially according to this Gath model. Universe expands exponentially, that is H is Hubble's parameter, AT is scale factor which expand exponentially. A huge expansion in a very small interval of time is the inflation. And the condition is H dt, integration H dt is greater than 65.
Dr. Final 1 minute.
Okay, sir. Now exotic matter. Exotic matter are tachyon, phantom, Chaplin gas, and accelerating phase of the universe at the later epoch is permitted with an equation of state P equals to omega O, where omega is less than -1. This is the tachyon, and there are conditions for such acceleration considering phantom and tachyon field. For tachyon field, this is the condition. This is Lagrangian, this is density, this is P, and the condition is that the P for accelerating universe, this is the condition. The negative pressure, for negative pressure, psi dot square must be less than -1. And for phantom field, this is Lagrangian P rho, and the condition is psi dot square less than -2v. This is the condition for obtaining accelerating universe in the present epoch. Still, inflationary model are still acceptable cosmological model of the early universe. Every inflationary model can be tested, and many of them have been already ruled out in comparison to the predictions of the observational results. However, it is difficult to reject the idea of inflation. This is in short, and these are the references.
Thank you, Dr. Paul.
Thank you, sir.
We are unable to take questions due to time constraint. Let me move to the next talk. Let me invite Dr. Anurtha Palit.
Yes, sir.
Yeah, let's get started.
Yes, sir. Let me share my screen. Sir, is it visible to you all?
Yeah.
Okay. Thank you, sir, and thanks for the.
You have 6 minutes time in total. Okay.
Okay, sir.
So my title of the presentation is efficient estimation of limit cycle solution of regular forced Rayleigh equation using novel asymptotic structures. Over here, let me directly go to the problem. This is the Rayleigh equation, a nonlinear ordinary differential equation, where this epsilon is actually the main dominating parameter. This is the external force, that external force F, and it has many different applications in different fields of physics and all. Over here, we are actually trying to study the asymptotic behavior of the solution. Asymptotic behavior means the nature of the solution when we take the time tending to theoretical infinity, but that would be actually any arbitrary large time. This Rayleigh forced Rayleigh differential equation can be written as a non-autonomous system, and as time increases, we find that it has an isolated periodic orbit, and this kind of orbit is known as limit cycle. It has unique isolated periodic orbit. We are trying to find out the an approximate value or approximate analytic solution of this kind of limit cycle solution, for which we apply the perturbation method on this kind of forced Rayleigh equation with omega is equal to 1 plus epsilon sigma. Sigma is the detuning parameter over here. These are the intermediate computations in the perturbation method. And in this method, the perturbative solution has this kind of term, which is actually unbounded as time t tending to infinity. So it is actually a kind of secular term. And in order to suppress this secular term, because the presence of this kind of secular term will actually make the solution inconsistent when time increases. So in order to suppress or in order to eliminate this kind of secular term, we apply the renormalization group method. And as a first step in this renormalization group method, we actually introduce an arbitrary time tau in order to split this time difference t minus t0 in this fashion. And then we renormalize this constant of motion by a function, its counterpart a, as a function of t, a complex valued quantity as a function of t. And you see over here, the required solution should be independent of the choice of any kind of arbitrary time. That's why this derivative would be equal to 0. That is the renormalization group condition. And once we apply this renormalization group process, this corresponding solution, we get this kind of solution where r and theta are guided by this kind of differential equations. But the problem is that we do not get sufficient close approximation. Although we eliminate the secular terms, but the solution so obtained is not very close to the exact solution. This is the comparison in between the renormalization group solution, and this is the solid line representing the exactly computed numerical solution. And in order to get close and close solution of good and better approximation, we apply the non-Archimedean extension of the real number system, satisfying this kind of properties known as duality structures. We ultimately get certain kind of control parameter by which we can control the convergence of the approximate solution to the exact solution. And this technique is we are giving the name that an improved RG method or improved renormalization group method. And this is the solution. You can check that the dotted lines are obtained, and these dotted lines, the solution obtained from these dotted lines are actually the improved renormalization group solution. And this solid line is actually an exact.
Dr. Pali, do you have your final minute?
Yes, sir. I've already finished. So this is the result that we obtained, and that's all. From this picture, it is clear that we got a very close enough solution, and these are few references, and these are some papers. This paper has been accepted recently, and this is one of the paper accepted by Chaos Soliton Fractal, written by my teacher, Dr. Indrajit Prasad Datta. So I think I should stop over here. That's all from me. Thank you.
Thank you, Dr. Padit, for your nice presentation. We are still not able to take any question from the participants.
No problem, sir. Thank you.
Let me move to the next speaker, Dr. Kaushik Ghosh.
Yeah, am I audible?
Yeah.
Okay. So I'm quickly sharing my slide. Let me just. I think it's coming.
Yeah. It's fine.
So, let me start then. As you can see, the topic of my presentation is here, theoretical model of differential rotation in brown dwarfs. So myself, Dr. Kaushik Ghosh from Burdwan University, and they are the collaborators, Rinku Bag and Kaushik Bhonja. They are my, Rinku Bag is actually my PhD scholar, and Kaushik Bhonja did this internship in the PG project, as a PG project from Jadavpur University. So let me quickly come to the basics of this particular presentation. As we first want to mention what is about the brown dwarfs. Actually, the brown dwarfs are substellar objects, and their masses are between bottoms of the main sequence stars and the giant planets. So it is actually interlinked. We are having the masses of these brown dwarfs approximately between 13 and 75 to 80 Jupiter masses. And basically, at the very beginning, when the theoretical prescriptions was given for the brown dwarfs, in the 1960s by Professor Shives Kumar, he actually thought that these particular things will not be detected, and that's why he named those things as black dwarf, because the luminosity is very, very feeble. And after that, in 1975, Gilles Tarter, he mentioned that name can be transferred to brown dwarfs, because he thought that perhaps in the later days, this particular things can be somehow detected because there is some kind of advancement in the detection level. And later on, it was detected in the 1990s. Basically, it was first detected in 1995, then 1996 also. And now we are having some number of brown dwarfs in the in the observation detections. And what happens that at the very early stage, if the age is supposed to be less than 10 to the power 8 years, they were actually quite luminous. But as they age, they cool rapidly and become harder to detect. That is the problem. So you can see there is very feeble number. The density at the core is very less compared to the other stars, normal stars. It's around 10 to 10 cube grams per centimeter cube. And if you just think of the temperature at the core, it's still very less. It's around 3 into 10 to the power 6 Kelvin, which is in the other stars possibly around 10 to the power 7 to 10 to the power 8 Kelvins also. And the pressure you can see at the core is something about 10 to the power 5 megabar, so which is less. And what is the motivation and objective of the present work? Actually, we are here trying to find out the differential rotation model of these brown dwarf objects. What is actually visible for the brown dwarfs? The rotation rate decreases gradually from the center to the surface along the radial distance. And it possibly maintains the spherical symmetry, and it also decreases, also the latitude decreases from the equator to pole. So there are two different parameters what we can think of. One is the radial distance, another one is the latitude. And on the basis of these facts, in the present work, a theoretical model of the angular velocity of the rotating brown dwarf has been proposed. And it has been taken as a function of both the radial distance as well as the latitude. And we have taken here 5 different observed brown dwarfs. The names are CHA H alpha 2, H alpha 3, H alpha 6, and type 1 and LP 94420. And we have tried to estimate what can be the changing profiles of these particular objects with the rotation rate due to the combined effect of the latitude as well as the radial distance. So let me come quickly to the theory. First of all, we are coming to the very oldest theory. It was proposed long days back in 1630, and we have still used this one, and we are still using at different levels. It was proposed by Scheiner, and it says something about if you want to find out at certain latitude phi, what is the angular rotation, it is supposed to be something about omega e minus omega p sine square phi, where this omega e is supposed to be the angular velocity at the equator and omega p as at the pole. And by using that particular thing, let me come to the other aspect. Earlier, I have actually done this work along in my PhD thesis, that we have tried to estimate what can be the proposed omega, that is the angular velocity, if we consider the radial distance. And the radial distance is mapped as the psi. You can see that omega xi is actually taken as omega c, some function xi to the power half. Omega c stands for the omega, that is the angular velocity at the core. And using different things, using the Chandrasekhar's model, on the basis of these particular proposition what we have made here, we can think of this particular equation is coming out about these xi. And theta is actually the scaling of the density rho, and it is known as the Lange-Emden function, and it was earlier defined by Chandrasekhar 1939. And using that model, we can find out the relation between this theta and rho by this. And similarly, after this calculation, we can get some kind of theta, which is, of course, a series solution of this one by means of these particular equation 6. And using that particular thing, we are finally getting the equation 8 as this, where we have taken the combination of the xi, which is the radial distance, as well as the latitude. And we have taken the model in this form, a cos square phi plus b sin square phi multiplied by these xi to the power half. And we have made this particular assumptions that omega 0, 0. That means here at the very central core, this is supposed to be omega c. This is the equator, where xi1 is actually the surface, where it is the first 0 of this equation, which is denoted that the surface, and 0 is here still the latitude. And we are getting this particular pi by 2, which is the pole. So you can see omega in and omega p, these are coming back. So after certain particular calculations, these substitutions are made. Finally, we are getting the equation as 15, what we can see here. This is supposed to be in the known forms of omega c, omega e. We are getting this particular form. So xi will be the corresponding radial distance, and phi will be this particular latitude. We can identify what can be the possible omega as a variable of these particular two things. So after using this particular equation, I'm not going to all the details. We have taken care of what can be the increasing and decreasing parameter at different levels. So we have taken the first order derivative and the second order derivative. And we are having also the idea of what is the average angular velocity at different levels. So you can see
Dr. Ghosh, you have your last minute, okay?
Yeah, it's okay, no problem. So we are having this map of these omega. And on the basis of that one, let me come to the results. As we have mentioned that I will be dealing the calculations for these particular 5 different objects. So you can see these are the different 5 objects. And these are the results, what we can have. These are the first column denotes the observed average. These are already observed. And the second is observed radius, what we have already observed. We don't have to detect that one. Finally, on the basis of these observations, we have calculated the estimate average angular velocity of these 5 objects. And later on, we have also tried to identify what can be the possible results. This is actually the profile of the H alpha 2. As we can see, as the latitude changes, as the particular radial distance changes, how the angular velocity is changing. And here you can see different color shades are taken. Actually, we have taken different shades just to identify the ratio between omega and p. Omega p at different levels. We have taken care of different ratio. How this omega p e and omega p, they are different. We can have these identifications. So this is for the second one, H alpha 3. This is for the H alpha 6. This is for trade 1. Almost similar kind of graphs and patterns are coming, only the magnitudes are different. And this is the last one, LP94420. So let me come to the conclusion quickly. The present work describes the theoretical model of differential rotation at different latitudes as well as at different radial distances in rotating ground dwarfs. And we have taken care of these 5 different objects, and their considered and graphical profiles of variable angular velocity are presented for each of those with changing latitudes and radial distances. So I have taken some of these particular elementary references that you can see here, and this is actually the entire presentation. Thank you very much.
Thank you, Dr. Ghosh, for your nice presentation. We can't take questions from the participants because of time constraint.
Okay, it's okay. Thank you very much.
Let me move on to the next speaker.
Dr. Bay.
Yeah.
I want to say one thing. We have 4 presenters left.
Yeah.
So we have to leave this platform for invited speaker who is presenting from USA.
Yeah.
So we have to leave it sharply at 5:30 p.m. So I would like to request the participants, if they complete their presentation between 4 to 5 minutes, not then it will be helpful for us.
Okay.
Continue.
I must convey to the participants, I mean speakers. Okay. Next speaker, Dr. Shukanto Pramanik. So you have 5 minutes now. Okay.
Am I audible, sir?
Yeah, audible. You have 5 minutes. Okay. please strictly adhere with the time constraint or time limit, 5 minutes.
Okay, sir. Myself, Sukanto Pramanik from Siliguri College. I am delivering my lecture on a new 2-parameter lifetime model. Now, first introduction. Modeling of any real-world phenomenon gets complicated. Statistical distributions are important for parametric inferences and also are commonly applied to describe real-world phenomenon. Due to the usefulness of statistical distributions, their theories are widely studied and new distributions are developed. The interest in developing more flexible statistical distributions remains strong in statistics profession. The development of statistical distribution is an old topic in statistics. There are several ways of adding one or more parameters to a distribution function. And addition of parameters make the resulting distribution richer and more flexible for modeling lifetime data. Now, first of all, how to construct my new distribution. The cumulative distribution function of my proposed distribution function is fx lambda theta equals to integration over 0 to fx by 1 minus fx x lambda t, where fx by 1 minus fx, this is an odds function. In this presentation, I choose a particular choice of f lambda x is an exponential distribution with parameter lambda, and f theta x is also an exponential distribution with parameter theta. So hence I call this distribution as odds generalized exponential distribution with 2 parameter lambda and theta. So this is my proposed PDF. Fx equals to lambda theta e to the power theta x e to the power minus lambda e to the power theta x minus 1. I find out some mathematical expressions like measure of central tendency, dispersion, skewness, kurtosis, generating function, mean deviation, conditional moments, etc. Next, I create some plots of this distribution by using R programming. This is the plot of figure 1 and figure 2 are the plot of the probability density function of my proposed distribution. This is the plot of the distribution function, and figure 4 is the mean, median, mode and variance for lambda equals to 0.5 and for different values of theta. Now this is the plot of hazard rate function, reverse rate function, and mean residual life and mean reverse residual life of this distribution. Now here I use maximum likelihood method of estimation procedure to estimate the 2 parameters. This is the likelihood. This is the likelihood function, and this is the normal equation. From the first equation we get the parameter lambda, and from the second equation we get the parameter theta. Here I use the Newton-Raphson method. Now here Monte Carlo simulation study was carried out considering 1,000 times for selected values of lambda and theta. Here is the algorithm. Sample size 20, 40 and 100 were considered, and values of theta were taken as 0.1, 1 and 2 for lambda equals to 0.01 and 0.1 respectively. 2 measures have been done, made by bias and mean square error. Table 2.1 and 2.2 are the bias and MAC of the ML estimator for different lambda and theta. From the table 1 and table 2 shows that the bias is positive in case of our distributions, and table 2 and 3 are also shows that bias and MAC decreases as n increases. In terms of bias and MAC, the parameter lambda and theta of the odds generalized exponential distribution is efficiently estimated compared to that of the other distribution.
Dr.
Now
Pramanik,
data analysis.
please
Yes,
go
sir.
to your conclusion slide.
Okay, sir.
Because we
Just sir, 1 minute, sir.
Yes.
Here I feed this distribution with this data set. This is the strength of glass fiber data set. This is the estimate of the parameter lambda and theta by maximum likelihood method of estimation. This is my parameter value lambda and theta hat. And histogram here, histogram shows that data is positively skewed. The data is negatively skewed. Barreto and Souza fitted this data. Barreto fitted
You have
distribution.
30
I have
seconds
fitted
time left. 30 seconds.
fitted line of our distribution shows that it covers maximum portion of the histogram than the beta feature distribution. And this is the conclusion. Study the new 2-parameter lifetime probability distribution called odds generalized exponential distribution. The advantage is that the distribution has only 2 parameters that are to be estimated, and the proposed distribution has been compared with some standard 2-parameter distribution through simulation study, and the appropriateness of fitting the 2-parameter lifetime model has been established by analyzing a real life data set. Thank you, sir.
Thank you, Dr. Pramanik, for your presentation again. Let me move to the next speaker, Mohammad Ridwanur Mandal from Siliguri College. Please join and start your presentation.
Am I audible?
Yeah. You have
Good
5
afternoon,
minutes time, okay?
Okay, sir. Thank you.
Due to
Good afternoon,
very
respected sales
strict
organizers
schedule.
and
Yeah.
experts. Is the slide visible?
Yeah. Proceed.
Okay. So, my topic is modeling of an eco-epidemiological system with following type response functions. The main points are introduction, formation, etc. Epidemiology, and first of all, mathematical modeling. Actually, nowadays, mathematical modeling is very popular to model biological systems. Interactions among biological community is studied using mathematical modeling, and modeling which deals with disease individuals also in measures, and epidemic modeling is such kind of the biological systems having some disease like infectious diseases. We here consider some differential equations, system of differential equations, and we will deal numerically analyze the transmission of the disease. And we are considering here the disease only on the predator population. And next thing is functional response, which I have already using Holling type functional responses. So there are several functional responses like Holling type, Bennington, D'Angelis, etc. And functional responses actually are those things which deals with the per capita feeding rate or intake rate of the prey by the predator. So this is general model actually. Mostly this one is Lotka-Volterra type model, where X is the representing functional responses, and there may be various kinds of functional responses from Holling type functional response, maybe linear as well as rectangular hyperbolic type, as well as this type of function mx by a plus x square. But in this model which we have considered here, we have considered a generalized Holling type functional response. Here this one is mx by ax square plus bx plus c, and this is the model where x t is the prey population, where y and z are predator population, but y representing susceptible predator as well as z representing infected predator species. Here the coefficients are all positive,
Mondal,
and they have
you have
biological
last 1
meanings.
minute, please.
Okay, sir.
Go to the
So these are some preliminary results, and equilibrium. There are 4 equilibrium points on at which we have discussed stability behaviors like say local stability behavior, global stability behavior, and bifurcation also is there at the coexisting equilibrium point, where there all the species will be existing. To
is it done? We can't hear you.
Okay, sir.
Okay.
So this one is conclusion. The main objectives of this article is to study eco-epidemiological model, where the sound and infected predator population consume the single prey species, and the system is bounded as well as the equilibrium points are stable locally as well as globally, and also there are bifurcations like things.
Okay.
So thank you all.
Thank you, Dr. Mandal. Let me now invite our next speaker, Dr. Rakibul Biswas from Bagpur Rashtraguru Surendra College. You have 5 minutes time. Okay.
So am I audible?
Yeah.
Okay, I'm just sharing my screen, sir. So is it visible?
Yeah. Yes. Yeah. Slide is coming. Yeah. You have 5 minutes, please remember.
Okay, sir. Good afternoon, everyone. Myself, Rakibul Vishwas from Bagpur Rashtrakur Suryandat College, Department of Physics. Today, I am going to give a short presentation on solar heat harvesting. My paper title is Experimental Investigation on Solar Heat Harvesting by Using Paraffin Wax as a Phase Change Material. And in my next slide, I describe the outline of my presentation. So my presentation actually is in 5 parts. At first, we discuss about introduction and then solar energy and heat harvesting and then experimental setup and then result and discussion and last but not the least, conclusion and future scope. And in my introduction part, I shown a picture of pie chart of energy consumption in India. So from this picture, we can say that almost 75% of energy, which is coming from conventional energy source. Recently, the energy demand is increasing day by day, but stock of our resources of conventional energy is decreasing gradually. On the other hand, every day a huge amount of solar energy get reflected from Earth's surface. So it is the high time to think about the uses and conservation of non-conventional energy. And in this slide, I discuss about the solar energy and heat harvesting. Solar energy is the radiation and heat energy which come from the sun. The solar energy that reach Earth's surface is much higher than the energy which used daily all over the globe. In this work, we have devised a system to trap a significant amount of solar energy by using paraffin wax as a promising phase change material. Phase change material are the substance that absorb and release heat energy when they are changing their phases. We can calculate the amount of heat conserved in the phase change material using equation 1, which is Q equal to L into M. Here Q represent the energy stored during the phase change, L represent the latent heat of phase change material, and M represent the mass of the phase change material. And here I shown a picture of different phase change material, and from this picture we can see that paraffin wax has affordable melting point and not much less than melting enthalpy. So that's why we have chosen paraffin wax as a phase change material. And in this slide, I shown a phasor diagram of a phase change material. And we in our setup we have used 1.5 kg of paraffin wax, and we know that latent heat of paraffin wax is 176 kilojoule per kg. So by using equation 1, we can calculate how much energy we can conserve in our setup. That is about 264 kilojoule. And in this slide, we comparing the sensible heat and latent heat of water and paraffin wax. So from this table, we can say that for water, it takes a large amount of energy to melt ice into the water and water into the steam. And so paraffin wax is the excellent material for storing heat. And in this slide, we describe our experimental setup. We have used paraffin wax and dust charcoal to make a composite material which have a good efficiency to conserve heat. And we have also used thermocol box and sawdust to make a setup of heat conservation, because thermocol and sawdust has a very low thermal conductivity, and we have just put in 3 thermometer vertically to take the experimental data. And in this slide we discuss about the result and discussion. So from the figure A, we shown the temperature variation of upper thermometer with time, and figure B and figure C, the
Dr.
temperature
Vishwas,
variation.
you have 1 minute left. Okay.
Okay, sir. Middle and lower thermometer with time respectively. So experimentally, we observed that the rate of increase of temperature is much higher than that of the temperature during the decreasing in the same time interval. And in this slide, we conclude our work. From our previous discussion, we can conclude that this model can be envisioned as a promising contribution to solar power harvesting. Solar power concentrator may be devised using multi-mirror reflection. So multi-mirror mirror position can be turned by solar tracking mechanism, which is given in the lower right side picture. And we can use this model as in our domestic life, water heating, cooking, room heating, different industries and airports and etc. And here is my references, and I would like to thank the Department of Physics, UG, PG, Prabhat Kumar College to giving us such amazing platform to do this project work. And I would like to thank all our respected professors, Dr. Kaushik Shankar Das, Dr. Pradipta Panchadhyay, Dr. Piyush Kanti Samanto, Sir Kaushik Nando to guide us our project work. And thank you very much for your listening, sir.
Thank you, Dr. Biswas, for your presentation.
I'm a student, sir. I'm not the actually doctor.
Oh, okay, no
So
problem.
thank you very much.
Let me move on to our last talk. Dr. Nimolendu Hui from Krishnanagar Government College. Please start your lecture. Dr. Hui, you have 5 minutes.
Okay, am I audible?
Yeah.
Let me present the slide.
Please start.
Yes, let me present the slide. Actually, there is some problem.
Dr. De. Dr. Karthik De. Hello, Dr. De.
Hello.
Your slides are not visible.
Okay.
Moderator.
Hello.
Sir, the slide was not visible.
Dr. Hui,
is facing
your slide is not visible to us.
There is
You can
some.
say something.
Actually,
You can say
hello. Am I audible? Hello.
Yes, you are audible.
Hello. Actually, there is some problem.
No problem. You just say the objective.
Just within 2 minutes.
Within 2
minutes,
because
you can describe your work within 2 minutes. Slides are not essential.
Okay. My topic is. Am I audible?
Yeah. Within 2 minutes you finish.
I am interested to study the collective behavior of interacting dynamical system as it gives rise to several emergent phenomena like synchronization, chimera, oscillations. Mainly, my focus is on quenching of oscillations. Actually, it is necessary where there is 2 type of oscillations quenching at
Okay, continue. We have no time. Dr. Hui.
Honorable Chair, Dr. Day.
Yeah.
Nimolendu Hui left the meeting, so you conclude.
Yeah, he's facing some technical problem from his side. Okay, if you know, Nimolendu Hui is not able to present his. Yeah. Thanks everyone who joining, and thank you organizers for inviting me to chair a session.
Thank you.
Over to you, Dr. Karthik De. Now.
Okay. Professor Rajat Kumar De, an international scientist of high energy physics, astronomy and astrophysics at the Department of Physics, University of North Bengal. He was with us, brushing out all his extremely busy works. I, on behalf of Seminar Organizing Committee, Acharya Government College, would like to thank Dr. De. I hope your gracious presence in the future program organized by our college. I also thank all the participants for their nice presentations. Thank you. So I would like to request the chair to conclude the session. End the session. Thank you, all of you. Next, hello, am I audible?
Hello, sir. Hello.
Yeah. Dear participants, next, an invited talk will be scheduled from 5:30 p.m. Dr. Kaushik Das is the speaker, Department of Physics, University of
Maryland, USA. So before starting, wait.
Okay, thank you very much for inviting me to present my work in this very prestigious seminar series. So I see a lot of noted people here. So let me just share it first, and then I will start in a minute.
Good evening, everybody. This is the second session of International Interdisciplinary Web Seminar on modern trends in humanities, science and technology, and social sciences for sustainable development. In this section, we have two important sessions we already completed. Now we have with us the invited speech from Professor Kaushik Das, Department of Physics, University of Medicine, United States Kingdom, and I, on behalf of organizing committee, welcome Professor Das to the international web platform to deliver speech which will enrich ourselves as well as all the participants, all the delegates, all the viewers of this program in an international level. So I heartily welcome Professor Kaushik Das to deliver his speech. Professor Das, please, over to you, sir.
Thank you very much. So I hope I'm audible to everyone.
Yes, sir. You are clearly audible.
Yeah. And my
Yes, sir.
slides
Yes, sir.
are visible on the screen, right?
Yes, sir. Yes, sir. Slide
Yes,
is
sir.
visible.
Yes,
Yes, please.
absolutely.
Okay, very good. So before I start my talk, let me thank the organizers for inviting me for this talk. This is important platform, and this is an opportunity to discuss a few things that I would like to discuss with you. One of them would be how to introduce curiosity-driven research even in undergrad research curriculum. So that's one of my interests. And before I start the talk, can I request everyone to mute their mic except the organizer so that I don't or nobody hears any background noise.
Thank you, sir.
Yeah, so
Organizer will also put off their mic. Before that,
yes, please. Yeah.
One announcement is there. I request Professor Chandra, Honorable Professor of Episura Government College, to chair the session over the session. That is a simple announcement, and mic will be off. And sir, please proceed. Thank you, sir.
Okay, thank you very much. So today, as I said, that the topic of my talk is curiosity-driven research, and then I will give some examples from microwave plasma to space. Those are some of the research projects that we did in our research lab, and these are examples of undergraduate student research. And not only undergraduate student research, these research the results were featured in international media, and we published this research in many peer-reviewed journals, and I'll talk about that. So the goal is to discuss how we can introduce or properly do undergrad research, and not only just research for research sake, rather it should be a very good quality research, how we can do that. So this is the topic. The first word that we can see on the screen is curiosity, right? Curiosity driven research. So what does it mean by curiosity? So or how do we enhance curiosity? So that should be the first point, right? we can start talking about it, but even without knowing how we can enhance curiosity, it's meaningless to talk about curiosity-driven research. So this picture, a picture tells a lot. It instead of words, if we show a picture, and this picture immediately draws our attention to the involvement, the level of involvement of these kids. These kids are naturally curious. They have found something very interesting, and they are looking into it intensely. And not only that, what are some other components that we can notice from this picture? There is a group. It's a group phenomena. So not only these kids are observing something intensely, they're discussing it, right? And then there would be some conclusion. They could understand something. So there are a few small things. Those are involved in this picture, and those are not so small if we look into it in great details. First thing. This is the ambience, the environment that made these kids curious. So this is nature. One of the examples is nature, and the ambience is a driving force of curiosity. That proper ambience is needed. You need a group of diverse people who can bring different ideas into the study. And then you need discussions, right? You need to discuss and brainstorm and debate. So there would be a debate. Even if looking at this picture, you can tell that some people would, one of the kids would say, I think this is this. Another kid would say, no, I think that is not that. I think this is this. So that debate is a healthy thing. So group, ambience, debate, discussions, these lead to a research, a proper research, if we can translate it into a classroom. And this is not limited to kids only. This is equally applicable to adults, in college level kids, even university level kids. Everybody is prone to these forms. So before we, so this is a psychological fact, how to form this group, how to form this ambience. And all these things are related. It starts from class. It's not. It does not start in the research lab. It starts from the classroom, and then it goes to research lab. Nobody can become innovative scientist without developing their critical thinking ability, without developing their curiosity-driven ability in the classroom. Okay, so let us look at the psychological part first, and then I will come into the research part. So how does one create ambience? Let us look at the next picture. The slide transition is going well. So this is the second slide, and this shows a picture of a restaurant, rather a takeaway place, where the language is not understandable. It's a foreign language. But if you look at this picture, it tells you something. It directs you to do something. What does it direct you to do? It directs you. It tells you that if you see some place like this, you are supposed to go to the front desk. You are supposed to order the food from the list, and the person in the counter would deliver you the food, and you will pay for it, and then you take it away, right? This message is delivered from the ambience, from the architecture. But if you enter somewhere like this picture psychologically tells you that you are not supposed to stand in there. You are supposed to go and take a seat on the table, on the chair, and then somebody would bring the menu card, and then you tell what you would like, and they will deliver the food to your table, and then you pay, and etc. etc. So those two psychological, completely different psychological understanding are just delivered from the architecture, isn't it? So now if we look at this picture, what is the expectation from the students? The expectations, the psychological expectation is that somebody in front of the room, somebody would deliver a lecture like a sermon, and the others who are the students will just look at the professor or teacher intensely, listen to him intensely without uttering a word, and there is no room for discussion, isn't it? So this is the psychological thing. Whenever we enter into the classroom, that is delivered from the architecture, from the environment, from the ambience. This message is delivered to the students that what we are expected to do, just like what I just showed from these two different restaurant phenomena. This is exactly, this message is delivered that you are not supposed to talk, you are supposed to listen. And what did we see from the first slide? What brings curiosity? It brings the curiosity-driven research is driven by discussion, driven by collaboration, driven by brainstorming. Is there any room for those things in these classrooms? No, unfortunately, this is what happens in most of the classrooms. And this is one of the reasons why we cannot innovate, why we cannot think big, why we cannot dream and make some groundbreaking ideas, bring some groundbreaking ideas to the table. Because the message is clear, our education system does not allow us to do that, does not allow us to think freely, and does not allow us to brainstorm and discuss. So this needs to be changed. How can we do this? Creating a right environment. First of all, we need to create the environment so that we can discuss. The students can discuss. The students are not there just to listen and listen to a sermon and take notes only. They are supposed to brainstorm about any ideas that is delivered by the teachers or the professors in schools and in colleges, at the universities, anywhere. It starts from the primary school level, not at the universities. Imagine a person who has never done any sort of brainstorming, who has never thought anything about the applications of the learning process, what they have learned in classroom. A person who has never discussed the topics with his friends and debated it vigorously. How can that person do groundbreaking research? Even if that person is given a lot of money and resources afterwards, up to master's degree, there is no room for this type of brainstorming and critical thinking activities. And then suddenly at the PhD level, students are expected to make great things. Is it possible? Is it rational thinking? It's not a rational thinking, unfortunately. And the West has figured it out long time back. And then they have changed the curriculum from the primary school onwards into the university level. This is a university level picture where I used to teach at the University of Toronto. This is one of the classroom plus lab. So we used to teach in that space. And these tables are like hexagonal tables. Students would sit face to face, and then there would be an experimental apparatus there in the middle of the table. We'd teach something, suppose a theory on, say, oscillation or something, on Newton's law, and then immediately ask the students, do some experiments and check what I am saying is correct or not, and then debate about it. Tell me that you don't believe me. Whatever I teach you doesn't have to be correct, or you don't have to take it like Vedas or Bible. It's not something written in stone. So debate and argue, and unless and until you are convinced that this is what I'm saying is correct, don't take it for granted. Don't take it that this is true. You do it by yourself and check it by an experiment. A theory must be validated by an experiment. Otherwise, it's just a hypothesis, not a theory. We Bengali especially, we know how to do this. And actually, this is in our genes. A few days back, there was a documentary in BBC Travel, and they analyzed the most important part of any Bengali's life, which is adda, right? So, and they gave the definition of adda and how adda shapes our life, common Bengali's life. And there was this strange word, adda, the secret of Bengali conviviality. And see, I'm a Bengali medium student. I never I was never taught in English medium. So this is like Shashi Tharoor type English. I immediately Googled what is conviviality means. Conviviality is the quality of being friendly and lively. So being friendly and lively in discussions, that is the natural quality that we all have. And yet, when we go to classroom, we sit like robots. Is it a natural thing? We are killing the natural tendency of us that is so great that we can talk, we can discuss, and this is the sort of definition that this is adda is a kind of unplanned mental exercise where we not just talk about ourselves and our families, but we go beyond that. It is about ideas and events happening all around us. And this is the goal of science, to understand nature, to understand about ideas and events happening all around us. Not only science and humanities and the social science, you name it, or economics, you name it. It's about ideas and events happening all around us, analyzing them and finding some solution to some problems. This is what group discussion looks like in our common life, daily life. Just look at the intensity of the discussion. This is exactly the same as we saw in the first slide, right? Everybody involved, very involved in the discussion. Everybody very involved in the discussion in a coffee shop, in chai tech. That's what we call here, right? But when you go to classroom, nothing is there. Everything is just sitting, everyone is sitting like robots. So why do we discard that natural conviviality and become robots when you go to classroom, and classroom becomes boring and the learning becomes boring, and we don't get involved in the learning process, and yet we expect we'll do great things in science and social science and economics and everything. Just look at a few great people of Bengal. Satyajit Ray, Amartya Sen, Mrinal Sen, those like people who had groundbreaking ideas. They are the product of coffee house. Remember, if you look there, if you read their statement or their autobiography or anything, when they talked about themselves, they recognized the importance of coffee house, those addas, those free flowing ideas, those debates, in how it shaped their life, right? So this is how we can bring that group discussion, debates in classroom. You break it down in different groups. So this is another classroom, modern classroom, how it looks like. You bring 4 to 5 people in each table. You just rearrange the benches. And you don't need a lot of resources to do that. You do that with your limited resources also. And where is the teacher? The teacher is here. Teacher is the back of the room. You teach something, you teach it on the whiteboard, project it on the screen, and then throw a question in between the classes, in between the discussions, and ask questions to the students. And most of these questions are open-ended, and let them discuss and debate among themselves. And then ask them, one student from each group, what is their idea about that question? That's how you bring discussions and debates in classroom. And in the West, all the modern classrooms are just like that. This is one of the examples that I was involved. we designed this new classroom, and this is a press release from University of Toronto, and they were very proud of that new practical rooms. And just let us read the first line. The first thing you notice when you walk into one of the new physics practical rooms is the hum. Hum is the noise of the discussions, the students discussing freely, just like the hum you hear in a coffee shop, or the hum you hear in chai tech, or the hum you hear in a bazaar discussion. So that lively discussion you are creating in the classroom. And I was involved in there in designing this classroom. It became immensely successful, and now it is one of the flagship thing in the University of Toronto. So this is what we did, and this is how the teacher's role, the professor's role here is to facilitate discussion, right? So that's what we tend to do. Now, critical thinking. What is critical thinking? Critical thinking is the ability to understand, apply, analyze, evaluate facts or principles. This is very, very important. If we don't let the students discuss and debate and brainstorm, then we are not letting the people become critical thinkers. Why? You know, this is what happens if you don't let the critical thinking process seep into students' mind. Then we become intellectually growing, and intellectual growth, it's a cartoon. It says progression from ignorant certainty to intelligent confusion, and this is so true. We just memorize a few facts, and we don't analyze it, or we cannot use those facts or those analysis in our day-to-day life, or solving a problem in our day-to-day life. This is one example. What I don't understand is how they get cubic centimeters in round bottles. That's a joke. But anyways, so in modern pedagogy, this is one of the most important rubric the educators use to measure whether the students' thinking ability is going from the lower order thinking to higher order thinking. How? First, the students learn to remember, just like memorizing Newton's laws. Then the next step is whether you understand it or not, those laws. If you can apply those laws in solving problem, whether you can analyze it, whether you can evaluate it, and then final stage is whether you are able to create something new using what you have learned so far. So these are critical components of critical thinking. So you remember first. Remembering is the first step. Understanding is, suppose you are solving a few book problems, and mostly our level of thinking remains in here unless and until you let the students become free to think, free to analyze critically, free to debate. So our full understanding of any subject matter remains here in these two steps, and then application, analyzing, analysis, evaluation, and creating. Those are higher level thoughts. So first thing is formative assessment. You assess the students' progression in class in real time. There are some methods. Today's talk is not, we cannot discuss everything in today's talk. It takes a series of talks to talk about each of the components. Formative assessment is extremely important. Group discussions, guided discovery that we talked about in the first few slides, that you give the students some hands-on tools and let them explore whether your theory is correct, whether what you are telling them is correct at all or not. Hands-on activities followed by innovative research projects. Unless and until the students go through these processes, they cannot be innovative researchers. So these are gradually, these are scaffolding process, just like a ladder. If you want to go up, you have to step on the lower levels, and then you gradually move on. So these are the steps. So group discussion, discovery, guided discovery, hands-on activities. Then you bring them to innovative research projects. So it starts in classroom. It starts in teaching labs, and then eventually it goes up to the research lab. So when we talk about innovative research, it's you cannot do it with a simple recipe like a cookbook. Just like you know how to make dal, how much water is needed, how to fry this moong dal, and then pour water at some point of time, what ingredients you need. Although this is not easy. I mean, the dal making is also not easy. I mean, I shouldn't have given this example. I mean, very difficult to make Mayer Hater dal, right? So we all know that. So still, science is not a simple recipe in a cookbook, and learning is not memorizing facts or tests. What we need to do, we need to climb up the ladder to go to the top level. We need to achieve all these capabilities and skill sets to create something, to be able to create something. So then only this Make in India will be possible, right? So if we want to be innovative, we need to follow these steps. Okay, so first thing, what we did in our labs after going through those processes, we created some authentic discovery-based laboratories, and we brought these in classroom. In authentic discovery-based courses, we created courses to encourage students to get immersed in research. These are one semester course, two 1 semester courses. We created two authentic discovery-based labs. In the first course, they would do all the background research, read. We'll choose a topic, and then the students will read at least 10 research papers, peer-reviewed research papers, and then write a report. And in the second part, they will do hands-on interdisciplinary research. So that was how we did it. Experiencing the thrill of an original scientific discovery can be transformative to students. I don't have to tell this. You know, everyone knows that a discovery is life-changing. The discovery doesn't have to be huge all the time. Even when my first excitement, personally, I can tell you, when I discovered that how this lebu ras, this lime juice or lemon juice, can make this phase transition in hot milk from milk to milk solid, dudh theke chhana kata. That was the first experiment I did when I was a little kid, and then I became curious. To me, I was so little at that time that I couldn't differentiate between talc and salt, like lemon juice and salt. So I thought, if lemon juice can do this, can I test if salt can also do that? And I won't encourage someone to do that, but I did that experiment with like a bulk of milk. When যখন আমার মা বাইরে গেছে রান্নাঘর থেকে, তখন আমি হচ্ছে এতখানি নুন নিয়ে ফেলেছি, আমার ফার্স্ট এক্সপেরিমেন্ট। And I don't want to talk about the results now, because the results were visible in many places. Okay, so that was the hands-on experiment. Only a few courses offer authentic research experiences. Increasingly cutting-edge discoveries require an interdisciplinary participation. This is important. Interdisciplinary participation is important. Why? Because every discipline and the students in every discipline has their own unique perspective. A student of chemistry looks at the same problem differently than a student in physics or mathematics or biology. So everybody brings their own perspective to the table, and that becomes really a vibrant research community. So what we try to do, we bring like one chemistry student, one physics students, one engineering student or mathematics student, and group them together and give them one single topic, research topic, and let them evolve, let them bring their own expertise, their own level ability of thinking, own thinking pattern into the problem. And then we do weekly discussions, and then the final research project that can be presented. We will create a final research project, we'll present it in a national meeting or published in a scientific journal. And this is where we are also lacking. We don't, for our undergrad students, we have very limited resource, and not only resource, very limited place where they can present their work. So I'll come into this part in the at the end of my talk, where we are trying to create a consortium in India, especially in Bengal, where a few colleges have come up together and we have proposed that we can create a consortium to host an annual undergraduate research symposium where students can present their work, and I'll come into that. Okay. So how we are talking about the topic of our talk was from microwave plasma to space research that we have done in our lab, our undergrad students have done. So where from it started? It started from some curiosity-driven research, obviously. We saw that some people have done some research on organic solar cell. Yes, it is organic solar cell. They extracted from the pigments from dark-colored fruits and berries, and just like jam, you can see here, this is jam or beet, and these pigments are called anthocyanin, and this anthocyanin is very very I would say it's a solar active material that it absorbs sunlight and it gets activated with sunlight. So we extracted these anthocyanin from these dark colored fruits and berries that is available here, and then we use titanium dioxide as a semiconductor using normal protocol. This is known thing, there is nothing new, and people have done it before. So we created these, we were redoing the experiment, and we created solar cell from these things. And this is very easy method, and you can do that in your at your home also. So this, and then we analyze them, and this is the setup done by my students in the parking lot. You can see the solar cell is there, and they measured the efficiency of the solar cell, which was low, obviously, because this is not done in a clean room environment, or this is a very crude method. We did it just to check the principles, that whether it works or not, and how it works. And then it worked perfectly fine, as expected. Then we became curious. You know that we use these titanium dioxide semiconductor in making those solar cells. And then we became curious if we can replace this titanium dioxide by zinc oxide, which is a very similar semiconductor, whether it works or not. There was no report, almost no report in any of the papers. So we said, let's do it. Let's change it to zinc oxide, and then let's see whether efficiency becomes greater or lesser. And surprisingly, the solar cell didn't work. And it didn't work at all. There was no signature of any current or voltage generated by the solar cell. And then we became further curious why this is happening. And we started dismantling everything, and we started checking every single component of it. What we found that zinc oxide changes its resistivity in not only in UV light, rather in visible light spectrum also when it is mixed with ethanol, ethyl alcohol. So we analyzed it, and we did detailed analysis of those things, and then it became we published it in 2018, and the resistance switching memoristic hysteresis in visible light activated adsorbed zinc oxide thin films. And this became one of the most read paper in 2018, and this is like Nature Research sent us this data, and they congratulated us for becoming one of the most important papers, most read paper in 2018 from this Nature site, nature.com site. So this is how it worked. Now, when we did that, we wanted to check whether this effect, this zinc oxide resistance switching, whether this is a surface effect or bulk effect. And one way to analyze that is by creating plasma, or by treating the material with plasma. And what is plasma? First of all, we didn't have a plasma reactor at that in our lab at that time, so we couldn't do that. But first of all, what is plasma? Plasma is the fourth state of matter, and this is the definition of plasma. Some people would say that this is the fourth state of matter. Why it is fourth state of matter? Because normally we talk about three states of matter: solid, liquid, vapor, right? Solid, liquid, gas. So if you take an example like solid, say ice, yeah, and if you add some heat, what is heat? Heat is a form of energy. If you add some energy to ice, it melts. It changes its phase to liquid phase. And then if you further add some energy to it, to the liquid phase, which is water, then it becomes gas, which is vapor, water vapor, right? Now, so if you add some energy to a state, it changes its phase, and if you keep on adding energy to the gas phase, what we get? You'll get a new form, which is a new phase, which is called plasma, where all the electrons are separated from their bonds, and all the bonds are broken, and then it's a soup of negatively charged electrons and positively charged nucleus, and that soup of gas, ionized gas, is called plasma. That's why it is called fourth state of matter. Plasma is not like something special, it's ubiquitous, it's everywhere. Lightning is a plasma, sun is a plasma, plasma TV, neon light, even fire is a plasma, aurora is a plasma, interstellar gas is plasma, so plasma is everywhere. There is huge application of plasma, fusion research, rocket thrusters, semiconductor processing, material science applications, astrophysical plasma. There are tons of applications. So we wanted to create plasma because we didn't have a plasma generator in our lab at that time. So how do you create plasma? You normally take two plates, metal plates, and you generate huge amount of electrical field and in between them, and then the electrical field, and then you need some primary source of electrons. In that electric field, the electrons will become very fast and accelerate, and then it will bombard with other atoms. Those are present in the gas, in the tube, in between the plates, atoms or molecules, and then they would bombard on those atoms or molecules, and then there will be secondary electrons coming out. They will knock out electrons from the outer shells. And those knocked out electrons will again accelerate because of the electric field, and then it will create tertiary electrons and so on and so forth. And within a moment, you will get a bunch of ionized nucleus and ionized part of the gas and the electrons. It becomes plasma, and this is what it glows here. So this glowing thing is plasma. We wanted to check now microwave that almost everybody has, and it is available everywhere, including our lab also. So microwave delivers the same electromagnetic energy, and that's how you get your cup of tea hot in a microwave. How does it occur? It microwave delivers oscillating electromagnetic field, and the oscillating electric field oscillates the water molecule in the cup, water molecules in the cup, and then when they oscillate, they collide with each other, and that collision becomes is manifested as heat in the cup. So that's how the microwaves work. So we thought, if microwave is a viable source to deliver energy, why can't we create plasma in a microwave if we deliver energy through microwave? And we did that. We had this hypothesis that we'll take some water vapor in a gas, we'll take a tube or a flask, and then we'll create some vacuum in the tube, and then we'll just water vapor molecule will be everywhere. And if we have right pressure of the tube or the flask, then we should be able to generate plasma in there. So that was our hypothesis. So microwave plasma, so this is electrical field. You see E is the electrical field, and if you have a dipole here, then the dipole, any undergrad physics student would know that a dipole would try to orient itself in the direction of the electric field, because these positive charges will be attracted towards the electric field. The negative charges will be repelled in the opposite direction of the electric field, and it will align itself. So now, instead of a steady electric field, if the electric field is oscillating, just look at these arrows, then what would it do? The dipole would start to oscillate also. Or if it overshoots, then it will start to rotate or spin. And this is, if it spins vigorously, because microwave frequency is very high, and then because of this rotational kinetic energy, an electron may pop out, right? That's just the centrifugal effect, centrifuge. The way a centrifuge works, it moves the heavier part of the molecule in one side, and the lighter part would be the other side, and there is a mass distribution of the water molecule. And also, if while it is rotating, if it hits some other molecule, it can knock out an electron. So that could be the primary source of electron. And once the primary source of electron is created, it will start to accelerate both the positive ion in the direction of the electric field and the negative ion in opposite to the direction of the electric field. They would start to accelerate, and then it would knock out electrons, secondary electrons from other molecules. Exactly the same way we have seen the plasma generation in a tube, and this is how the plasma will be generated again. So this is the cascading effect will occur, these plasma. So how could you do that? You can change the pressure of the bottle, and then you see that when there is the pressure is low, we have observed that there is no plasma generated in the bottle, and when the pressure is in the sweet spot, we have plasma generated. When the pressure is too high, there was no plasma generation. So that's what we have seen, we have observed in our lab. And it became a big news that plasma in a jar. MIT Technology Review featured our work as how to turn a kitchen microwave into a plasma etching device. Not only that, they said that it could be a game changer. So plasma, but an easier way to make and control plasmas could change all that. So it says that it could be a game changer in the field. So, and this is done by undergrad students. This is again just the curiosity-driven research. If we can direct it properly, they can do wonders. It was published in American Journal of Physics. This is free to read. I'd suggest you guys to look at that. Go and this is freely downloadable also. It became editor's pick, editor's choice. Some of the only a few selected papers from American Journal of Physics they make editor's pick. And this is freely downloadable. So please check plasma generation using a kitchen microwave oven, and you can put my last name DAS and American Journal of Physics. It will pop up in Google. You can read that. Now, where is space coming into here? When we did that, okay. Can I get 3-4 minutes extra? I know I have to. Yeah. So when we wrote that paper, we had a comment that when the water molecules are rotating very fast, it may flip flop, and that was predicted by some understanding of solid body rotation, and the referees didn't like it. Referees said that how can you say that the water molecules spinning very fast will flip back and forth? And we said we can demonstrate that. But how can you do a free moving, freely rotating particle on Earth? Because in gravity, it will fall down, right? So we decided to design an experiment and to go to space. That's what we did. We created an experiment. We did a parabolic flight, zero gravity experiment, and we flew. I personally flew with that in that zero gravity flight. And let me show you. I don't know whether I can run it or not. So, rotating solid bodies behave strangely in space. This is our experiment in collaboration with MIT. And this is a nut opened in International Space Station, and look how it behaves when the solid body rotates freely. This is a dancing T handle in the International Space Station. And then if we move forward, we created a toy water molecule. You see this rotating water molecule there, and then in zero gravity we let it go. That is, we let it become freely rotating, and we were trying to observe what we can observe. What is the dynamics of that toy water molecule in space? In a few seconds, you'll see that this is detached, and you see this is again flip-flopping. So let me see if I can show you a greater video. This is me in explaining this. So this is the water molecule here, and this is in zero gravity. You see this, now it would be detached, and this is rotating, and then, and this is flipping. You see that flip-flop behavior you can analyze analytically, not just experimentally. Okay, so, and then this is the end of my talk about how you can do it in undergraduate level. But in the context of India, what we can do, what the pressing needs of the society, that topic we can bring into the undergrad students. That what we can do, how we can use our understanding in physics and applied science and humanities and social science together and solve a big problem of the society. The understanding, if it is only kept in solving test problems and cracking engineering exam or just some job interview, it's not enough. Where is the connection between solving a societal problem using our understanding that we are getting in schools and colleges? So that's what we identified a few problems like grand challenges: water, energy, environment, food. food security. That is very important in the context of India, like how to desalinate water. India is surrounded by water, and yet in Bangalore, in Delhi, in Chennai, you have to buy water, drinking water. So what's the problem? Problem is desalination. India is surrounded by water, but those are seawater, those are ocean water, then those are like salty water. Can we develop a very simple method to desalinate that water in a low cost method, low energy consumption method, low carbon footprint method that can be used to desalinate this huge amount of water and solve that drinking water problem? This could be one issue. One of my friends worked in there. He was my lab mate when he was at MIT. Solar steam generation by heat localization, and then this topic was also written in a Bengali web portal, which is biggan.org.in. I'm one of the editors of that science portal. This is an online science journal, you may say. Every article is written here. Whatever is written here is peer reviewed, and we send them to the experts in that field, and then we get every article gets reviewed, and then we publish it. We try to publish very cutting edge research and both in education research and physical science research in this portal. So I would encourage everyone to visit biggan.org.in. Some of the initiatives we have taken, the formative assessment that we talked about but we didn't discuss, you can go to Biggan, and we have developed a method. It says Poranor Obhinabo Paddhati Rongin Cardar Madhyame. This is done for the school and college students who are not very like fluent in English. They can read it in Bengali. Even school teachers can implement these techniques in their classroom, school and college teachers. So I'll encourage everyone to visit biggan.org.in and register there. All the new articles will be delivered to your mailbox. We are working with Contai Science Academy and several schools to implement education research-based techniques in classroom. We have started summer research program to solve local problems with local talents, started modifying and modernizing textbooks from class 8 to 8 onwards. So let us made it a movement. Let us, all of us join in, and then it would become a movement of the society. It's not like done by only a few bunch of people. Okay. So just like a few minutes ago, Rakibul gave this talk on solar heat storage by using phase change materials. This is one of the topics we have chosen as local problems to be solved by local students. There is solar power related smart device using artificial intelligence, designing biodegradable materials for practical applications, and graphene preparation from pencil lead. So all of these are cutting edge research, and our students are doing it. And this is what I just said, and this is one of the slides with which I want to end my talk. All these things are done by undergrad students. We have got the cover page article in applied nanomaterials, current applied physics. We got numerous awards. These students have been absorbed by NASA. Many students are going to Ivy League schools to do their PhD, and I would like you guys to replicate that in Indian context also. And if you need any help, any discussion, anything that you think would be beneficial for Indian context, we'd be happy. From biggan.org.in's point of view, we'd be happy to assist you in any ways we can. Okay, so thank you very much for your attention, and I'd like to have any question that arises from my talk. Thank you.
Thank you, sir. So it is open for discussion, and anybody can put question on the topic. If there be no question, sir, I have one question.
Sure.
In Indian context, how this kind of ambience can be produced?
Yes. So that's a good question. Actually, just this week we had a meeting. We here begin.org.in. We are acting as a catalyst, and we are trying to form a consortium. Not we, rather we as the catalyst. We had a meeting with St. Xavier's College and then Contai Science Academy and I think PK College, Prabhat Kumar College, and some professors, very interested professors, they came in and they are trying to make a group to start this year-long undergraduate research program. And the resources will be shared, and they are trying to start an undergraduate research symposium annually every year. So if you can join in, and I'd encourage all the colleges to join in, then it will start to snowball. Actually, it will start to grow, and then once it reaches the critical mass, it will become an annual event, and then you can write like joint proposal, collaborative proposals from DST or CSIR. And one of the suggestions was to create a maker space kind of space, like a small room where all the necessary tools, like small tools, would be kept in every college, so that the students can explore and create things, like a few drill machines, one laser cutter, one 3D printer, a few Arduino kits. Those are necessary things, and those, if we can keep those things in proper place in an organized manner, students can come in, use them, and design something, and then leave again. Next day they would come and do things. So it needs to be done. Somewhere we need to start. Right?
Thank you, sir. Thank you, Dr. Kaushik Das, Professor, Department of Physics, University of Maryland, and he delivered a talk on the topic curiosity-driven research from microwave plasma to space. And during the course of his discussion, he covered that the group debate ambience, these are very much important in research, for innovative research. He visualized how ambience is important to develop psychological understanding, development of critical thinking. Creating right environment starting from primary level is very important for innovative research. Group discussions, debate, or in Bengali words, Adda is very much important. As Professor Dash discussed, in Adda we feel very much free and become very much curious and creative, and in classroom we do not become so much creative. So ambience is very important. So we have to prepare the ambience and apply, and also we design some steps for making ambience and innovative research, and applying these type methods. He developed solar cell, organic solar cell, and he got success, and he published this in the journal, Nature journal. And finally, he suggested to apply the method to solve the social problem. I think the discussion is very much important and relevant to this present scenario. I would like to thank Professor Dash.
I think there is one raised hand, Johin Ghosh.
Yes, sir. So can I speak?
Yes. Sure. Yeah.
Sir, yes, sir. I am Vishwajyoti. Hello.
Hello. Yeah. Okay, please
Sir,
go ahead
just a
and
minute.
say one by one. Yeah.
Sir, thank you for your cooperation. You are here today in this platform, and I also thank our Deputy Director, Sir Sanjeev Bandyopadhyay, sir. So UGC HRDC who actually give your number and communicate with you. We communicate with you, and I thank to Sanjeev sir. Without Sanjeev sir, I can't communicate with you. It's a very pleasure that you are present in our this international seminar. Thank you, sir. Thank you very much from our side.
This is an honor. This is an honor, and this is an opportunity for us who are outside India to serve and contribute to Indian society and academics, right? And I must thank you for giving me this opportunity. So
Okay,
yeah,
sir. Okay.
yeah. So let us take this question. Johim Ghosh, you are trying to say
Yes, sir.
something.
Sir,
Yes,
you
please.
were saying that the proper substitute for titanium dioxide was zinc oxide. That was used in the solar panels, and we know
Right.
that sulfuric acid is used in battery acids, and it has
Right.
similar kind of activity. So why we can easily use sulfuric acid instead of all zinc oxide and titanium dioxide, I guess.
Yeah, actually, it needs some sort of material, solid state material, and zinc oxide and titanium dioxide. Both of them are large band gap semiconductor. So you need some semiconductor material to create those solar cells. So I'd suggest you Google it and read some of these dye-sensitized solar cells. This is very interesting topic. Google dye-sensitized, DYE, dye-sensitized solar cell, or DSSC. You will get a lot of information, and keep your curiosity alive. Why not sulfuric acid? So this is how new things come in. Okay.
Oh, thank you, sir. So, and there was one more thing, phosphoric acid. When we treat zinc oxide with phosphoric acid, I think it forms like a cement-like structure material. I think when we treat zinc oxide with phosphoric acid, so we use phosphoric acid as as a substitute.
I don't know. We haven't tried that, but yeah, definitely you can try.
Okay, thank
Try
you, sir.
it. Try it. Use
I'm not a
your
science.
hands.
I'm not a chemistry student. I was.
But anyways,
in class
talk to one of the chemistry professors. There is no such
Thank you,
rule
sir.
like you cannot do it. Anyways, I think our time is up, and yeah.
Thank you, Professor Kaushik Dash.
Thank you very much.
এত সুন্দর একটা স্পিচ মনে হয় আরেকটু শুনি, কিন্তু সরি, বিকজ উই হ্যাভ টু কোপ আপ আওয়ারসেলফ উইথ দ্য টাইমিংস। থ্যাংক ইউ স্যার। ভবিষ্যতে আমরা আমন্ত্রণ করব, আপনি আমাদের সাথে থাকবেন এবং এরকম প্ল্যাটফর্মে আপনাকে পাবো এই আশা এপিসিরা গভর্নমেন্ট কলেজ করছে। আপনি ভালো থাকবেন, সুস্থ থাকবেন। থ্যাংক ইউ স্যার ফর ইউর, I think, excellent presentation and super presentation. Thank you. Today's international interdisciplinary web seminar on modern trends in humanities, social science, science and technology for sustainable development. We came almost at the end of the session or end of the 2 days program. Now, I like to request Dr. Moyuk Sarkar, Honorable Officer in Charge, as well as Honorable President of this 2-day International Interdisciplinary Web Seminar, to deliver his valedictory speech. After that, we will have some discussion about the feedback, link, certification, etc. Participants, viewers, delegates, and all the persons, students here in this international web platform are requested to be with us, please. Over to you, sir, Dr. Mohit Sarkar. So, a very good evening to all of you, all the participants of this 2 days interdisciplinary international web seminar on modern trends in humanities, science and technology, and social sciences for sustainable development. We are glad to inform you that all the presenters have successfully presented their paper in this international web seminar. I congratulate them. And more than yesterday, that in the 23rd of September, we have presented 65 papers in humanities and social sciences. And today, on 24th, we have presented life sciences paper 31 and physical sciences paper 29. So more than 125 papers have been presented in this web seminar, and there was a very outstanding papers we have saw in the different fields of environmental degradation, in the directional changes, in the green technology, in the human-computer interactions, astronomy, astrophysics, mathematical modeling. So lots of that one, particularly in the socio-cultural attributes of the North Bengal. We have the papers from the Bengali department. So Bangla Sahitye Mahamari Udbhav Uttorun. So basically, within a very short period, we have conducted this web seminar with the UGC-HRTC, and it's a overwhelming responses from all over the participants throughout our country and outside our country. So I thank all of you, particularly the abroad speakers, Professor Salma Akhtar and other dignitaries. Particularly, they are very well spoken about that matter, but nowadays in the socio-economic conditions we have highlighted from the Bangladesh, and then in the last speaker, there's Dr. Ghosh, Dr. Das, he has spoken on different aspects of this, particularly in the field of physics. So all the participants have presented the papers. Due to some technical errors, we might have not presented in a little bit, but we have tried our level best, because this is the first conference in our college with the help of the jointly organized by the UGC-HRTC, and that is fully complete in the digital platform due to this COVID-19 pandemic. So thank you all about this matter. So we basically, main thing is that one, that particularly already has been the feedback links and everything has been circulated, and I will hand over my mic to our joint secretaries, particularly Mr. Vishwajyoti Roy and our Akash Chaudhary. They have done a tremendous job to perform this program successfully, and we had a good teamwork. Without this teamwork, it cannot be possible to do these sorts of web seminar. We are trying our level best to do that part, and thank you. There's Kaustav Rai is one of the major assistant professor in our college, who has guided us for this digital platform, and students who have been participated in our this web seminar. They have guided particularly for this YouTube link, and it has been circulated to the YouTube channel. So basically the overwhelming responses, and I am obliged that one that everybody has everybody will have presented this type of seminars, and we'll try to improve ourselves. Please feel free to fill up the feedback form, and automatically we'll get in touch with you, and after that we'll send you the certificates and everything, and matter will be, I'll hand over this matter to our joint secretaries, Akash Chaudhary. He will tell you about the details of that. Okay, thank you all, and very happy Puja and happy Deepavali to all of you in advance. So enjoy the Puja with maintaining all the COVID protocols, and because we know that the third wave is going to come or not, we don't know, but still it will be, will get vaccination, and probably that government is trying our level best to open our this higher educational institutions. I hope so that it will be open soon, and we'll meet physically in this period. Okay, thank you everybody for your patience hearing, and that you have done a tremendous job, particularly in this period. Thank you everyone, and I'm handing over it to our joint secretary Akash Chaudhary for the confidence session. Thank you. Thank you, Dr. Mohit Sarkar, Honorable Officer in Charge and President of this program. Today's International Interdisciplinary Webinar or web seminar on modern trends in humanities, social sciences, technology, and science and technology for sustainable development. There were major two sub-themes: socio-political, cultural, and economic dynamics for sustainable development, science and technology for sustainable development. A colorful inauguration program was there, which are decorated with a number of international personalities. Honorable Vice Chancellor Professor Subiraj Bhattacharya, North Bengal University, delivered his motivational speech in the session. I, on behalf of Organizing Committee, APC Government College, thank Professor Subiraj Bhattacharya, Honorable Vice Chancellor, North Bengal University, Professor Subhash Chandra Roy, Dean of Science, Professor Anjan Chakraborty, Honorable Director, HRDC MBU, Dr. Mosharraf Hossain, Assistant Director, HRDC MBU, Dr. Sanjeev Bhattacharya, Deputy Director, HRDC MBU, Dr. Shantanu Chakraborty, ex-officer in charge, APC Government College, and all the speakers describe the relevance of the topic of the seminar to the recent need of the society. We had with us the eminent international speaker like Professor Salma Akhtar, Department of Sociology, Dhaka University, Bangladesh, Professor Shabani Shaha, Lincoln International Business School, University of Lincoln, United Kingdom, Professor Habibur Rahman, Head of the Department of Sociology and Social Work, Dhaka, Bangladesh, Dr. Madhumita Manna, Honorable Additional Director, Education Directorate, Government of West Bengal, Professor Indrajit Pal, Disaster Preparedness, Mitigation and Management Section, Asian Institute of Technology, Thailand, Professor Kaushik Das, Department of Physics, University of Maryland, United States of America. The program was beautifully decorated with the invited speaker from these international speakers. In the first day, that is day 1, 23rd September 2021, there are two sections for technical session for presenting the papers regarding humanities, literature, social sciences. As already YC sir, that is President sir, described the details. I will not go through that part. About 65 fundamental research work from different section was presented. In the second day, that was designed for life science and physical science and chemical science and mathematical science. There are about 31 papers from life science, including zoology, botany, microbiology, immunology, ecology, environmental biology, etc. And 29 papers from physics, chemistry, mathematics, computer science, biotechnology, etc. So many discussions are there in this multidisciplinary seminar. Episcopal Government College Seminar Committee feels that it is a successful in terms of the aims and objective of the seminar. It was aimed to lab-to-land dissemination, transfer of the knowledge from lab to land to find out the cause of the problem and find out the way of solving the issues with sustainable development in northern part of Himalayan foothill, that is Tarai and Duars. In that case, we had with us Honorable Chairperson Dr. Bhaskar Goswami, Department of Economics, Bardhaman University, Professor Swati Bhattacharya, Department of Philosophy, Krishnanagar Government College, Dr. Kaushik Dan, Department of Economics, Episcopal Government College, Professor Dalia Bhattacharya, Department of History, University of North Bengal, Dr. Swapon Sarkar, Honorable YC, P.R. Thakur Government College, Dr. Sudesh Lama, Department of History, University of North Bengal, Dr. Sanjay Poddar, Professor, Department of Geology, Bardhaman University, Dr. Amal Kumar Patro, Department of Geology, Vivekananda College, Kolkata, Dr. Rajat Kumar De, Honorable Professor of Physics, University of North Bengal, Dr. Vishwajit Sinha, Department of Chemistry, University of North Bengal. On behalf of Seminar Committee, Episcopal Government College, Akash Chaudhary and Vishwajit Roy, the Joint Secretary of the program, thanks warm wishes to all the dignitaries, all the chairperson, all the international speakers to continue or to control and to regulate the program. After all, you are the delegates, you are the presenter, you are the viewers of this program who are the basic section of this program. The seminar, international seminar, we are at the first stage and for the first time we are organizing this part. If any troubles regarded with the web, internet or any other technological problem, on behalf of seminar subcommittee, we expect that you will never mind about the matter. In the near future, we will meet again in this type of platform or in this type of seminar. Thank you very much. Now come to the oral presenter. You will have a feedback from all the participants, all the dignitaries. You can feel that feedback form and it is designed for all. But for only oral presentation, the feedback form must be sent through the Telegram group, and we assure you that all the certificates of participation along with the details of your name, designation, paper title will be reached to you after 15 days. Telegram group will continue. We are active in the Telegram group. If any person, if any presenter did not get any his or her certificate, they can contact me by phone after 15 days. We will send the certificate of presentation into your mail, which from which you fill the Google form. And I again thanks all of my colleagues, all the members advisory committee, honorable professors of our college, from outer college, from universities, HRDC, and all parts of section, non-teaching staff for their kind cooperation, which ultimately enable us to continue the program successfully. Thank you all for your nice presence and nice togetherness. We will expect that togetherness in future. Thank you, APCDA Government College, Siliguri, Matigara, West Bengal, India. Thank you.